Audio recording for this meeting has begun.
Good morning and welcome. I would first like to remind everyone to please mute your line when you're not speaking. For media and press, the FDA press contact is April Grant. Her email and phone number are currently displayed. My name is Jorge Garcia and I will be chairing today's meeting. I will now call the first session of the September 22-23 of 2022 meeting of the Oncology Drug Advisory Committee to order. Dr. Xixia Shen is the designated federal officer for this meeting, and she will begin with introductions.
Thank you, Dr. Garcia. Good morning. My name is Xixia Shen, and I am the designated federal officer for this meeting. When I call your name, please introduce yourself by stating your name and affiliation. We'll first start with ODAC members. Dr. Garcia.
Jorge Garcia. I'm a GU medical oncologist and the chief of solid tumor oncology at University Hospital's Simon Cancer Center at Case Western Reserve University in Cleveland, Ohio.
Dr. Kun.
Good morning. My name is Pamela Coons. I'm a GI oncologist and director of the GI Cancer Program at Yale Cancer Center in New Haven, Connecticut.
Dr. Liu.
Good morning, everybody. My name is Chris Liu. I'm a GI medical oncologist and associate director for clinical research at the University of Colorado Cancer Center.
Dr. Madan.
Good morning. My name is Ravi Madden. I'm a medical oncologist specializing in GU malignancies at the National Cancer Institute.
Mr. Mitchell.
Hi, I'm David Mitchell. I'm the consumer representative to the ODAC. I am the founder of Patients for Affordable Drugs, and I'm a cancer patient myself.
Dr. Roscoe.
Good morning. I'm Ashley Roscoe. I'm an associate professor in the Division of Hematology at The Ohio State University and medical director of the Oncogeriatrics program.
And Dr. Song.
Good morning. My name is Anthony Song. I'm an associate professor of medicine in the Division of Hematologic Malignancies and Cellular Therapy at Duke University.
Thank you. Next, we'll move on to temporary voting members. And Dr. Hummels.
Good morning. This is Balazs Holmes here. I'm a thoracic oncologist, and I'm also Associate Director for Clinical Science at the Montefiore Einstein Cancer Center in the Bronx, New York.
Dr. Harrington.
Good morning. This is Dave Harrington. I'm a biostatistician at Dana-Farber Cancer Institute and the Harvard School of Public Health.
Mr. Pantelis.
Good morning. I'm Jim Piantellis. I'm a patient representative, Navy veteran, and 17-year survivor of stage 3B non-small cell lung cancer.
Dr. Sheila.
Good morning. I'm Catherine Sheila. I'm a thoracic medical oncologist at the University of Maryland Greenbaum Comprehensive Cancer Center in Baltimore, Maryland.
Dr. Thomas.
Hey, I'm Thomas. I'm a clinical investigator, thoracic oncologist focused on lung cancers, mostly at the National Cancer Institute.
And Dr. Woolman.
Good morning. I'm Scott Waldman. I am the chair of the Department of Pharmacology, Physiology, and Cancer Biology. I'm an internist with subspecialty in clinical pharmacology, and my research focuses in GI malignancies.
Next is the acting industry representative to the committee, Dr. Krause.
Yes, good morning, everyone. Albert Krause. I work in cancer drug discovery and development. I'm currently with Pfizer Corporation.
Thank you. Finally, I would like to introduce FDA participants. Dr. Pazler.
Hi, I'm Richard Pastor, and I'm the Director of the Oncology Center of Excellence here at the FDA.
Dr. Beaver.
Hi, I'm Julia Beaver. I'm a medical oncologist and Chief of Medical Oncology in the Oncology Center of Excellence at FDA.
Dr. Singh.
Hi, this is Harpreet Singh. I'm the Director of the Division of Oncology 2 here at the FDA, which houses thoracic cancer.
Dr. Dresner.
Hi, this is Nicole Dresner. I am a cross-disciplinary team lead in the Division of Oncology 2 at the FDA.
Dr. Melino.
Hi, this is Justin Malino. I'm a medical oncologist and clinical reviewer in the Division of Oncology 2 at the FDA.
And Dr. Fori Zirkobak.
Hi, I'm Jeanne Fori Zirkobak. I'm a clinical pharmacologist at FDA.
Thank you all.
For topics such as those being discussed at this meeting, there are often a variety of opinions, some of which are quite strongly held. Our goal is that this meeting will be a fair and open forum for discussion of these issues and that individuals can express their views without interruption. Thus, a gentle reminder, individuals will be allowed to speak into the record only if recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the Government in the Sunshine Act, we ask that the Advisory Committee members take care that their conversations about the topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with the FDA about these proceedings. However, FDA will refrain from discussing the details of this meeting with the media until its conclusion. Also, the committee is reminded to please refrain from discussing the meeting topic during the break. Thank you. Dr. Shishia Chen will now read the conflict of interest statement for the meeting. Dr. Chen.
Thank you, Dr. Garcia. The Food and Drug Administration FDA is convening today's meeting of the Oncologic Drugs Advisory Committee under the authority of the Federal Advisory Committee Act, FACTA of 1972. With the exception of the industry representative, all members and temporary voting members of the committee are special government employees, SGs, or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of this committee's compliance with federal ethics and conflict of interest laws covered by but not limited to those found at 18 U.S.C. Section 208 is being provided to participants in today's meeting and to the public. FDA has determined that members and temporary voting members of this committee are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflicts when it is determined that the agency's need for its special government employees' services outweighs his or her potential financial conflict of interest, or when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Related to the discussions of today's meeting, members and temporary voting members of this committee have been screened for potential financial conflicts of interest of their own as well as those imputed to them, including those of their spouses or minor children, and for purpose of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, CARDACs, teaching, speaking, writing, patents, and royalties, and primary employment. Today's agenda involves the discussion of new drug application NDA 215643 for poziotinib tablets, submitted by Spectrum Pharmaceuticals Inc. The proposed indication used for this product is for the treatment of patients with previously treated locally advanced, or metastatic non-small cell lung cancer, NSCLC, harboring HER2 exon 20 insertion mutations. Select patients with NSCLC for treatment with poziotinib based on the presence of HER2 exon 20 insertion mutations using an FDA-approved test. This is a particular matters meeting during which specific matters related to Spectrum Pharmaceuticals NDA will be discussed. Based on the agenda for today's meeting and all financial interests reported by the committee members and temporary voting members, conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208 B3 to Drs. Olav Hummus and Ashley Roscoe. Dr. Hummus' waiver involves his employer's research contract for one study funded by competing firms. This study is funded by a competing firm, and Dr. Hummus' employer receives between $0 and $50,000 per year. Dr. Roscoe's waiver involves her employer's research contract for one study funded by a competing firm. This study is funded by Innovent Biotherapeutics, and Dr. Roscoe is not aware of the funding amounts being provided to her institution for the study. The waivers allow these individuals to participate fully in today's deliberations. FDA's reasons for issuing the waivers are described in the waiver documents, which are posted on FDA's website at www.fda.gov/committees/committees/human-drug-advisory-committees. Copies of the waivers may also be obtained by submitting a written request to the agency's Freedom of Information Division, 5630 Fisher Lane, Room 1035, Rockville, Maryland 20857. Or requests may be sent via fax to 301-827-9267. To ensure transparency, we encourage all standing committee members and temporary voting members to disclose any public statements that they have made concerning the product issue. With respect to FDA's invited industry representative, we would like to disclose that Dr. Albert Kraus is participating in this meeting as a non-voting industry representative, acting on behalf of regulated industry. Dr. Kraus' role at this meeting is to represent industry in general and not any particular company. Dr. Kraus is employed by Pfizer. We would like to remind members and temporary voting members that if the discussions involve any other product or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all other participants to advise the committee of any financial relationships that they may have with a firm at issue. Thank you.
Thank you, Dr. Chen. We will now proceed with FDA introductory comments from Dr. Nicole Dresner. Dr. Dresner?
Good morning. I am Nicole Dresner, an oncologist and cross-disciplinary team leader at the FDA. I will refer to Spectrum Pharmaceuticals as the applicant for the remainder of the presentation. FDA is bringing this application to the Oncologic Drugs Advisory Committee to enable public discussion of the poziotinib drug development program and our significant concerns regarding the overall risk-benefit assessment. The applicant is seeking accelerated approval of poziotinib 16 milligrams once daily for the treatment of patients with previously treated, locally advanced or metastatic non-small cell lung cancer harboring HER2 exon 20 insertion mutations. Poziotinib is an oral inhibitor of pan-epidermal growth factor receptors, including HER2. As this application is being considered for accelerated approval, I will first discuss the provisions of the Accelerated Approval Program. The ZENITH-20 study provides the primary efficacy and safety data in support of the application. After presenting the top-line results, I will provide a high-level overview of the 4 major review issues contributing to the FDA's risk-benefit assessment. The efficacy of poziotinib does not demonstrate a meaningful improvement over available therapy given the low overall response rate and limited duration of response. The safety data for poziotinib demonstrates a high rate of toxicity with a substantial incidence of drug interruptions and dose reductions. The dose was not adequately optimized, and the confirmatory trial has yet to enroll any patients. Finally, I will present the discussion topics and voting questions for the ODAC committee. As you will hear in the subsequent FDA presentation, the major review issues were discussed with the applicant prior to submission of the NDA and throughout poziotinib's development. Beginning in 2017, we informed the applicant that their plans for dose optimization were not adequate to support a registrational program. When the applicant presented their top-line data at a pre-NDA meeting in late 2020, we requested that additional data on dose optimization be provided in the NDA, but felt that the application was fileable. Given that 1 year elapsed between the pre-NDA meeting and submission of the NDA, our concerns were once again expressed in July 2021, and the applicant committed to providing additional data at the time of NDA submission. Our formal review began in November 2021, which has confirmed and magnified our several concerns. According to the Code of Federal Regulations, accelerated approval may be granted to a drug that is intended to treat a life-threatening disease and has an effect on an intermediate endpoint that is reasonably likely to predict clinical benefit. Accelerated approval is available only for products that provide a meaningful therapeutic benefit over existing treatments, and further investigation of the drug to verify its clinical benefit is required. FDA guidance interprets a meaningful therapeutic benefit to be an improvement in efficacy and/or safety in the context of available therapy. Given the residual uncertainty associated with the drug approved under the accelerated approval pathway, for regulatory purposes, the Division of Oncology has considered available therapies to be marketed drugs with traditional or regular approval, or those that are considered standard of care. FDA guidance also states that if post-marketing studies are required as part of the accelerated approval provisions, it is anticipated that the confirmatory trial should be underway at the time of an accelerated approval action. I will now review the ZENITH-20 study design. ZENITH-20 is an ongoing, multi-cohort, dose-finding and activity estimating study of poziotinib in patients with non-small cell lung cancer with EGFR or HER2 exon 20 insertion mutations. Patients enrolled in cohort 2 provide the primary efficacy data for this application. The primary endpoint is overall response rate assessed by independent central review. Secondary endpoints included duration of response. Data from cohort 5 was also included in the NDA to provide supportive evidence for the adequacy of dose optimization and included patients treated at a range of doses. I will now describe the major risk-benefit considerations for this application. First, we assert that the efficacy of poziotinib, as demonstrated by the limited response rate with poor durability observed in the primary efficacy population, is not improved over available therapy. For patients with non-small cell lung cancer who have received both prior platinum-based chemotherapy and an immune checkpoint inhibitor, available therapy includes docetaxel in combination with ramucirumab with a benchmark ORR of 23%. Anti-PD-L1 therapies are considered available therapy if not previously received and are associated with lower ORRs with more substantial durability than what is observed with chemotherapy. Trastuzumab deruxtecan, a HER2 targeting antibody drug conjugate, received accelerated approval last month for the treatment of patients with HER2 mutated non-small cell lung cancer, an indication which would include the patients who comprise the primary efficacy population in this application. The drug demonstrated a response rate of 58% with a duration of response of 8.7 months, both considerably greater than what was observed with poziotinib. A randomized trial of trastuzumab deruxtecan to confirm its clinical benefit is well underway. The ORR for poziotinib-treated patients in the primary efficacy population was low at 28%, with poor durability demonstrated by a median DOR of 5.1 months. Of the 25 responders, 24% had a response lasting more than 6 months. Similar results were observed in the subgroup of patients who had progressed on both prior platinum-based chemotherapy and an immune checkpoint inhibitor. Importantly, 1/3 of the patients did not receive prior treatment with an immune checkpoint inhibitor, which would be considered an available treatment option for these patients. The second key issue for discussion is the high rate of toxicity observed with poziotinib at the proposed dosage. In FDA's subsequent presentation, we will describe the highly toxic safety profile seen with the 16 milligram once daily dose, reflected by approximately 80% of patients requiring treatment interruption and over half requiring dose reduction. The overall toxicity profile is driven by very high rates of diarrhea, mucositis, and rash, each occurring in over 70% of patients. In addition, our review details fatal events of pneumonitis observed at this dose level. It is still unclear whether alternative doses would improve the toxicity profile associated with poziotinib. The third key issue for discussion is the inadequate optimization of the poziotinib dosage throughout the development program. You will hear from our clinical pharmacologist that the 16 milligram daily dose may not represent the optimal regimen. The applicant studied other doses in very limited patient numbers, resulting in similar response rates with overlapping confidence intervals. Given the toxicity profile of the 16 milligram daily dose, further dose exploration is warranted. The final key issue for discussion is the delay in confirmation of benefit. To verify the clinical benefit of poziotinib, the applicant has planned a randomized study of poziotinib 8 milligrams twice daily versus docetaxel in the second-line setting, the same population for which they are seeking accelerated approval. The primary endpoint is progression-free survival, and the targeted benefit is 2.5 months. However, despite ongoing discussions with the FDA about the need for a confirmatory trial beginning as early as 2020, enrollment was not opened until well after submission of the NDA, and no patients have been enrolled as of this month. Furthermore, the selection of poziotinib 8 milligrams BID as the dosage to be tested in the confirmatory trial is incongruent with the potential approval of a dosage of 16 milligrams once daily and with the applicant's assertion of improved efficacy at 16 milligrams once daily. Finally, it may not be feasible for this planned study to be conducted given the recent approval of trastuzumab deruxtecan in the same space. The risks of treatment with poziotinib at the proposed dosage must be considered in the context of its potential benefits in a rare patient population with few available therapies. FDA's risk-benefit assessment presents layers of uncertainties when the application is considered in its totality. The efficacy results from Zenith 20 are not improved over currently marketed second-line therapies. If granted accelerated approval, this would be the least effective targeted therapy for lung cancer approved to date. In addition, there was a high rate of toxicity at the proposed dosage. The applicant failed to adequately explore various dosages throughout the development program, resulting in disparate dosages being investigated in Zenith 20 versus the planned confirmatory trial. It is still unclear whether the 8 milligram twice daily dose is optimal. Finally, given that the confirmatory trial is not yet underway, the risks of severe toxicities and marginal efficacy may be borne by patients for years pending results of the randomized study. I will now present the discussion topics and voting question for the advisory committee. We ask that the committee discuss the overall risk-benefit of poziotinib 16 milligrams once daily given the following: its limited response rate with poor durability, high rate of toxicity, inadequate optimization of the currently proposed dose, and the delay in confirmation or refutation of benefit. We would like the advisory committee to vote on the following question: do the current benefits of poziotinib outweigh its risks for the treatment of patients with non-small cell lung cancer with HER2 exon 20 insertion mutations? Thank you.
Thank you, Dr. Dresner. Both the FDA and the public believe in a transparent process for information gathering and decision making. To ensure such a transparency at the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages all participants, including Spectrum Pharmaceuticals LLC's non-employee presenters, to advise the committee of any financial relationships that they may have with the sponsor, such as consulting fees, travel expenses, honoraria, and interest in the sponsors, including equity interests and those based upon the outcome of the meeting. Likewise, FDA encourages you at the beginning of your presentation to advise the committee if you do not have any such financial relationships. If you choose not to address the issue of financial relationships at the beginning of your presentation, it will not preclude you from speaking. We will now proceed with presentations from Spectrum Pharmaceuticals Inc.
Good morning, Mr. Chairman, members of the committee, FDA representative, and members of the public. I'm Francois Lebel, the Executive Vice President of R&D at Spectrum Pharmaceuticals. I first would like to thank you for the opportunity to share the data supporting our application for an accelerated approval of poziotinib for the treatment of patients with non-small cell lung cancer harboring HER2 exon 20 insertion mutations. I would also like to thank patients and their families, investigators, and their staff who have supported our efforts to develop this drug and made it possible for us to be here today. Poziotinib is an oral, irreversible tyrosine kinase inhibitor, or TKI. Patients with HER2 exon 20 insertion mutations urgently need effective and safe therapy. Our presentation today will demonstrate that poziotinib is clinically effective and safe in a patient population who currently has no approved oral treatment options. In early 2021, the FDA granted Fast Track designation for poziotinib in our proposed indication, shown here. Our clinical development program consists of 22 studies in more than 1,300 patients, representing the largest data set in this rare disease. We extensively evaluated doses in 7 studies at multiple dose levels from 0.5 to 32 milligrams per day. In addition, we conducted our positive pivotal study at 16 milligram QD. For our presentation today, we will focus primarily on our ongoing Study 202, also known as Zenith 20, which is a global, open-label, multi-cohort phase 2 trial. Pivotal efficacy data comes from Cohort 2 in previously treated patients. This is the largest study ever conducted in patients with HER2 exon 20 insertion mutations. The study met its pre-specified endpoint. Supportive efficacy data in the second-line setting comes from Study 202, Cohort 5, and an investigator-initiated study conducted at MD Anderson Cancer Center. Supportive efficacy data in first-line setting comes from Cohort 4, which has also met its pre-specified primary endpoint. Taken together, the clinical data supports approval of poziotinib under the accelerated approval pathway. Our clinical program was designed in accordance with FDA guidance and meets the criteria for accelerated approval. First, non-small cell lung cancer HER2 exon 20 insertion is recognized as a rare and life-threatening disease. Second, poziotinib provides a meaningful advantage over available therapies with an overall response rate of 28%, exceeding all available therapies. Please note that as highlighted in the FDA's briefing document, trastuzumab deruxtecan is not considered available therapy. Third, pozi demonstrated substantial evidence of efficacy in the pivotal Cohort 2, exceeding the pre-specified ORR, which is likely to predict clinical benefit. And finally, a randomized confirmatory trial, Study 301, is currently underway to confirm the clinical benefit demonstrated in this patient population with a futility analysis in 2024. There are 4 key points for discussion today. Patient needs new treatment options now. Poziotinib addresses the unmet need, and the ORR exceeded available therapies and met the pre-specified primary endpoint. The safety profile is typical of the TKI class, with high rates of diarrhea and rash, with a low rate of permanent discontinuation. These AEs are familiar and handled routinely by the medical oncology community. In addition, poziotinib shows a low rate of fatal pneumonitis. We must point out that extensive dosing study has already been done for this rare disease, including 7 studies comprising over 404 patients. The 16 milligram QD dose met the primary endpoint with protocol-allowed dose modification. Finally, the confirmatory study is actively underway with 8 milligram BID dosing as agreed with the FDA. We remain committed to working with the FDA if an alternate dose warrants further evaluation. A futility analysis will be conducted within 2 years. With that background, let me take you through the agenda for today's presentation. Dr. John Haymack, head of Thoracic and Head and Neck Medical Oncology Department at MD Anderson, will present the unmet need and the mechanism of action of poziotinib. Dr. Gajanan Bhatt from Spectrum will review the efficacy data. I will then return to present our safety data. And Dr. Mark Sosinski from the Advent Health Cancer Institute will provide his clinical perspective as an experienced thoracic medical oncologist and investigator in our program. We have additional experts here with us today to help answer your questions. All external experts have been compensated for their time. Thank you. I will now turn the presentation over to Dr. Haymack. John?
Well, thank you and good morning. I'm John Haymack, and I'm professor and chair of thoracic head and neck medical oncology at MD Anderson. Over the past 2 decades, I focused my career on caring for lung cancer patients, conducting clinical trials, and running a laboratory that studies new targeted therapies for HER2 mutations and other subgroups. I'm here today as a clinician and an investigator with firsthand experience using poziotinib to treat these patients with a high unmet need. HER2 or ERB2 exon 20 insertions are a targetable oncogenic driver, and they're therapeutic targets in non-small cell lung cancer. HER2 mutations as a whole comprise 2 to 3% of non-small cell lung cancer cases. Among these, the majority, up to 86%, or about 2,300 cases per year in the U.S., are specifically exon 20 insertions, which is the population we're discussing today. Importantly, this is the only major tyrosine kinase subgroup for which there is no oral tyrosine kinase inhibitor currently FDA approved. This is due to the structural challenge in developing inhibitors of this particular mutated kinase domain. I'll briefly describe the challenge in developing TKIs for these patients. The ribbon diagram shows the crystal structure of the HER2 kinase domain. HER2 exon 20 insertions cause the P loop of the protein, shown here in green, as well as the alpha C helix in turquoise, to be pushed inward, reducing the volume of the binding pocket and resulting in steric hindrance, making it hard for most TKIs available in the clinic to bind. For this reason, TKIs active for tumors with amplification of wild-type HER2, like neratinib or lapatinib, are not effective here. Poziotinib is able to overcome the steric hindrance largely because of 2 features: the small halogenated terminal group and the flexible amine linker that enables the molecule to fit into the binding pocket. Because of this structure, the molecule is able to more potently inhibit these insertions. Here we show viability curves for the 6 most common HER2 exon 20 insertions. The further to the left, the greater the potency of inhibition. As you can see, poziotinib is an order of magnitude or more potent than other HER2 TKIs currently in the clinic, such as neratinib or lapatinib. All of these drugs also have some activity against wild-type EGFR, which leads to the predictable, reversible class effects, such as rash and diarrhea. Other TKIs have been tested in the clinic for this population, and the clinical data is consistent with the preclinical observations. In the first 4 rows are trials that specifically tested TKIs for HER2 exon 20 insertions. And as you can see in the highlighted text, response rates are quite low, ranging from 0 to 12%. So this really highlights that TKIs currently available in the clinic for other purposes are not effective against HER2 exon 20 insertions. Given the lack of efficacy of the current HER2 TKIs, what are the treatment options for these patients after first-line platinum doublet therapy? This data here is from the FDA briefing document. As you see, docetaxel has a low response rate and a median PFS of about 3 months. While this is not highly active, it is the most relevant standard, and for this reason has been used as the standard comparator for phase 3 studies in platinum refractory non-small cell lung cancer patients. Docetaxel has also been combined with ramucirumab, and there a response rate of 23% was observed and a median PFS of 4.5 months. Given its toxicities and the comorbidities of the non-small cell population, ramucirumab is only used in a minority of cases. Finally, I'll point out that trastuzumab deruxtecan received accelerated approval last month. However, as noted in the FDA briefing document, it is not considered available therapy from a regulatory standpoint because it is approved under the provisions of accelerated approval. Now, what about immunotherapy? Checkpoint inhibitor plus platinum doublet chemotherapy is now the approved first-line treatment and the NCCN recommendation for patients with wild-type EGFR and ALK, including those with HER2 exon 20 insertions. Furthermore, it's worth noting that EGFR and ALK mutant tumors, HER2 mutant tumors like them typically are poorly responsive to checkpoint inhibitors, likely due to their low PD-L1 and TMB levels. This is illustrated by the data from the 3 largest cohorts reported for checkpoint inhibitor monotherapy for HER2 mutant tumors. In all 3 cohorts, checkpoint inhibitors provide a very short PFS ranging from 1.9 to 3 months and objective response rates from 7 to 8% when given as monotherapy. For patients who didn't receive checkpoint inhibitors in the first-line setting, this table shows the efficacy of the overall population and the HER2 mutant population where lower response rate and median PFS were observed. So to summarize, docetaxel and not checkpoint inhibitors is the relevant comparator for second-line therapy, both because checkpoint inhibitors are used as first-line treatment now with platinum doublets, and because they are not effective as second-line monotherapy in this population. I mentioned before that in addition to oral TKIs, the other targeted approach are HER2 antibody drug conjugates like trastuzumab deruxtecan. These drugs have distinct mechanisms and distinct toxicities that are relevant for selecting the most appropriate therapy. The TKIs are oral, they inhibit the kinase directly, and toxicities are predictable related to inhibiting wild-type EGFR. HER2 ADCs are intravenous, and their toxicities, like neutropenia, are related to their chemotherapy payload. Now, ILD pneumonitis has been a concern with trastuzumab deruxtecan, with a rate of 26% ILD pneumonitis seen in the DESTINY-01 lung study published in the New England Journal at the 6.4 milligram per kilogram dose. After modification of the dose to 5.4 milligrams per kilogram and limiting patients with certain pulmonary risk factors such as history of severe COPD, asthma, pulmonary embolism, pleural effusion, pneumonectomy, or pneumonitis, a rate of 12% was observed in the package insert. But it highlights that patients with certain pulmonary risk factors common in lung cancer patients may not be suitable for the drug. Resistance occurs through distinct mechanisms, which likely explains why poziotinib appears to retain its activity after HER2 ADCs. The benefits of having mechanistically distinct drugs such as these recently has been seen with the approval of amivantamab, a bispecific antibody for EGFR exon 20 insertions, as well as mobocertinib, an oral TKI for the same population that was recently approved with an objective response rate of 28%. So to summarize, what is the current treatment landscape? First-line therapy typically consists of platinum doublets with or without checkpoint inhibitors. And as per NCCN recommendations, a HER2 ADC, such as trastuzumab deruxtecan or trastuzumab emtansine, is an option, but some patients may not be suitable due to pulmonary risk factors. For these patients, the standard option would be a docetaxel regimen with the low response rates and chemotherapy-associated toxicities. Given these available options, as a clinician who treats lung cancer patients for a living, I would consider an objective response rate of 15% and a median PFS of greater than 4 months to be clinically meaningful in the second or third line setting for these patients, given the available options. With this in mind, you can see that there is an important unmet need for more effective second-line therapies after platinum doublets, particularly for patients who are not suitable for trastuzumab deruxtecan or who prefer an oral regimen. And there's also a need for a third-line option after a HER2 ADC or docetaxel regimen. As you'll hear from Dr. Bhatt, poziotinib is an effective oral treatment option with a favorable benefit-risk profile that addresses these unmet needs. I will now turn the presentation over to Dr. Bhatt.
Thank you, Dr. Hemack. Good morning. I'm Gajanan Bhatt, Senior Vice President of Clinical Science at Spectrum. I'm pleased to review the efficacy data that demonstrates the clinically meaningful benefit of poziotinib. Data from Study 202 provide the primary efficacy to support poziotinib in the proposed indication. I'll focus on the pivotal cohort 2 first, and then supportive evidence from cohort 5. Additionally, I'll share supportive data from an investigator-initiated trial at MD Anderson Cancer Center, which also investigated poziotinib in the refractory setting. Cohort 2 was an independent study with a pre-specified primary endpoint and patient population. Patients were required to have NSCLC harboring HER2 exon 20 insertion mutations and be previously treated for locally advanced or metastatic NSCLC with at least one systemic therapy. Patients were required to have at least one target lesion per local investigator using RECIST 1.1 criteria. All patients received poziotinib 16 mg once daily for up to 24 months. The dose could be reduced in 2 mg increments if necessary in the presence of toxicity. Now let me talk about the study endpoints. The primary efficacy endpoint was the objective response rate, defined as the proportion of patients with a confirmed complete response or partial response. Response assessment was based on the central radiographic review by an independent review committee. Dr. Haymack has just summarized the literature for efficacy for the currently available therapy in second-line NSCLC HER2 mutations. Based on the literature and per the FDA discussion, an observed ORR of 30% with 17% as the lower bound for 95% confidence interval was considered to represent clinically meaningful efficacy in this study. The ORR was evaluated in the as-treated population. Secondary endpoints included disease control rate, duration of response, and PFS. Next, let's look at the patient disposition. A total of 90 patients were treated and one patient was ongoing at the time of the November NDA 120-day safety data update. The primary reasons for discontinuations were disease progression reported in 58% of the patients. Demographics and other baseline characteristics were representative of the published literature for this patient population. The mean age was 60 years old and 2/3 of the patients were younger than 65 years. The majority of the patients were female, white, and non-smokers with an ECOG status of 1. Next, let's look at the prior therapy data. Patients in this study were heavily pretreated with a median of 2 prior lines of therapy. 39% patients received at least 3 prior lines of systemic therapy at study entry. 68% had received prior immune checkpoint inhibitor therapy, and 28% had received at least prior HER2 targeted therapy, including trastuzumab and T-DM1. I will now present the summary of efficacy. Cohort 2 met the pre-specified primary endpoint of ORR in the as-treated population. Based on the independent central imaging review using RECIST 1.1 criteria, the ORR was 27.8%. The lower bound of 95% confidence interval was 18.9%. That exceeded the pre-specified criteria of 17%. This ORR demonstrates clinically meaningful efficacy compared to the reported literature of available therapies. The disease control rate was 70% in this study. In this slide, a waterfall plot shows the tumor reduction due to poziotinib in cohort 2. Poziotinib demonstrated anti-tumor activity with 74% experiencing tumor reduction during the treatment. This Kaplan-Meier plot shows the duration of response in the 25 responders. The median DOR from the initial response to disease progression or death was 5.1 months. The duration of response at 6 months was 24%. The Kaplan-Meier plot in this slide shows the PFS. The median PFS was 5.5 months and the PFS at 6 months was 38%. Here is a forest plot presenting ORR in subgroups. The vertical line in the center denotes the overall study ORR. Across patient subgroups of demographics, baseline characteristics, and other subgroups, we observe mostly similar efficacy. Now let's look at a few of these in more detail. Poziotinib shows activity in patient subgroups regardless of prior lines of therapy. Although the study was not statistically powered for stratified subgroup analysis, it's noteworthy that for patients who had received 3 or more lines of prior therapy, the ORR was higher at 37%, and the median DOR and PFS were similar to that of the overall study. Now, this slide shows the summary of efficacy by types of prior therapy. ORRs were generally similar between subgroups regardless of types of prior therapy, including patients previously treated with checkpoint inhibitor or HER2-targeted therapy, except for TKIs where the ORR was 50%. Efficacy analysis was also performed for the 14 patients with stable brain lesions that were identified at baseline. In this subgroup, the ORR was 28.6% and the median DOR and PFS were 5.1 and 7.4 months respectively, suggesting poziotinib activity in the brain. Here, we have the quality of life data from cohort 2, showing mean change from baseline in symptom scores. Note that in this analysis, a decrease in mean symptom score reflects improvement in symptoms. Lung cancer-related symptoms like cough, pain, and dyspnea all showed a trend towards improvement over time, while diarrhea worsened initially but stabilized over time. Let me move to the first of 2 supportive efficacy studies, which is cohort 5. Cohort 5 includes 95 patients previously treated with HER2 exon 20 mutations across dosing schedules. I will now focus on the efficacy data for the 10 previously treated HER2 exon 20 patients who received 16 milligram QD. This slide shows the cohort 2 on left as a reference, and cohort 5 results are shown on the right. In cohort 5, the ORR was 40%, median DOR was 6.5 months, and median PFS was 7.3 months. Although number of patients is small, results of ORR, DOR, and PFS in 16 milligram QD are supportive of the primary efficacy from cohort 2. Next, I'll review the second supportive efficacy study. This was a prospective investigator-led study conducted at MD Anderson in 27 patients previously treated with a platinum-based therapy. This waterfall plot shows the best change from baseline in tumor reduction in 30 patients, out of which 27 were in second line. A majority of patients showed tumor reduction consistent with results from cohorts 2 and 5. Here, I am showing the overall survival data from cohort 2 and the MD Anderson study. Cohort 2 was not designed for the OS follow-up, and there was high censoring since patients were not followed once disease progressed. However, the median OS ranged from 15 to 17 months, which compares favorably to historical standard in platinum refractory treated with docetaxel that ranged from between 6 to 10 months. In addition to the studies I just reviewed, Spectrum has also conducted a phase 2 study of poziotinib in treatment-naive patients with NSCLC HER2 exon 20 mutations. The primary endpoint of ORR in cohort 4 provides additional poziotinib efficacy as first-line treatment in patients with HER2 exon 20 mutants. The ORR in the as-treated population by independent central review was 45% treated with 16 milligram QD. Median duration of response was 5.7 months and median PFS was 5.6 months. 74% patients achieved disease control rate. Next, let's look at the waterfall plot. As you can see here on this slide, there was again tumor reduction in a majority of patients. In summary, in pivotal study 202 cohort 2, poziotinib with 16 milligram QD dose met the pre-specified primary endpoint and demonstrated clinically meaningful efficacy in heavily pretreated patients of NSCLC HER2 exon 20 insertion mutations. The efficacy was seen across all subgroups. These results were further supported by cohort 5, where poziotinib demonstrated consistent response observed in 16 milligram QD dosing arm. Further support comes from MD Anderson Cancer Center study, where poziotinib demonstrated anti-tumor activity in refractory patients, supporting the findings of pivotal study in this heavily pretreated population. In cohort 4, first-line patients who received 16 milligram QD poziotinib showed high response rate of 45%, meeting the pre-specified primary endpoint. In conclusion, poziotinib showed consistent and reproducible efficacy in the proposed patient population. Thank you very much. Now I will turn the presentation over to Dr. LaBelle.
Thank you. Poziotinib is second-generation TKI. Like other drugs in this class, has toxicities related to wild-type EGFR receptor, and as a result, on-target adverse events are expected. Although these AEs are typical of this class and managed by clinicians every day, one should not minimize the burden on patients. The proposed labeling will allow clinicians to inform and guide their patients on what to expect and what measures can be taken to mitigate adverse events, allowing patients to derive benefit from this drug. Let's review the data. We've studied poziotinib in more than 1,336 patients in our clinical program. Our primary safety data supporting the proposed indication was Group 1 from our briefing document, which included 482 patients treated with poziotinib 16 milligram QD or 8 BID, regardless of EGFR or HER2 receptor or line of therapy. I should point out that in the FDA briefing book, the safety population is restricted to patients treated with 16 milligram QD, a group of 368 patients. In spite of this difference, the safety results are similar. Today, I will present safety data from Group 1 as well as Cohort 2, which is the basis of our NDA. Cohort 2 patients were dosed with 16 milligram QD and therefore are included in Group 1. This table shows poziotinib exposures based on dosing data recorded in patient diaries. The duration of treatment showed a large range with a median of 113 days. The relative dose intensity was 72% of the planned dose to be received. Any day without a reported dosing level or with a missing dose diary entry was counted as a dose interruption. Median time to first dose reduction was 36 days, and first dose interruption was 16 days. The most commonly reported adverse events were generally on target and are typical class effect of second-generation TKIs. The most frequent AEs included diarrhea, rash, stomatitis, and paronychia. Adverse events were predominantly associated with either skin and subcutaneous tissue or GI disorders. Similarly, grade 3 events were on target with rash, diarrhea, and stomatitis occurring most often. Grade 4 adverse events were uncommon. Generally, events were manageable by following study protocol, prophylactic recommendations, and management plan or site-specific institutional protocols. Now, turning to serious adverse events. As expected, serious adverse events of dyspnea and pneumonia were observed most often in these advanced lung cancer patients. Importantly, SAE rates for rash and diarrhea were relatively low, occurring in 3% of patients or less. There were no serious events of pneumonitis or ILD in Cohort 2. Individual adverse events leading to treatment discontinuation occurred at low rates. Rash was the most common cause in 4% or less of patients. Treatment-related AE leading to permanent discontinuation was 12%. Pneumonitis or ILD of any grade was seen at a frequency of 1.1% in cohort 2 and 3.3% in group 1. There were 16 patients in group 1 who experienced an event of pneumonitis or ILD, and most events were assessed as related to poziotinib. There were 4 fatal events on pneumonitis at a rate of 0.8% and 3 in 16 milligram QD and 1 at 8 milligram BID. Importantly, there were no fatal ILD cases in cohort 2. We have proposed language in the warnings and precautions section of the label to address common drug-related toxicities. Based on event severity, we are recommending to withhold dose, reduce dose, or permanently discontinue. A detailed plan is outlined in the briefing document. Off-target adverse events were uncommon. There were no clinically significant abnormal ECG in cohort 2, and no patient had QTc prolongation. There were no clinically meaningful change in cardiac parameters. In summary, the safety profile of poziotinib is similar to second-generation EGFR TKIs. The most common adverse events were on target, included rash, diarrhea, and stomatitis. Most AEs were reversible following study protocol, recommended measures. Importantly, pneumonitis ILD, a potentially life-threatening adverse event, was rarely observed in the total data set. Of note, our previously treated patient population with HER2 exon 20 insertion, with only 1 patient with grade 1 pneumonitis in cohort 2. Now let me briefly review the dosing rationale for 16 milligram QD. The FDA briefing document mentioned that various doses needed to be adequately explored. However, we have conducted 7 studies with 404 patients, concluding that 16 milligram QD is the appropriate starting dose. Preclinical allometric scaling in mice projected 15 milligram QD as the safe and effective dose in humans. We then explored continuous and intermittent dosing ranging from 0.5 to 32 milligram per day. Our phase 1 studies determined the maximum tolerated dose of poziotinib to be 18 milligram continuous daily or 24 milligram QD 2 weeks on, 1 week off dosing. As a result, 16 milligram QD was selected for an investigator-led study at MD Anderson and for our pivotal trial. In addition, we conducted a randomized dose-ranging study in cohort 5 that confirmed 16 milligram QD as the appropriate starting dose. Recently, we have communicated to the agency our willingness to consider conducting additional dose optimization post-approval. And when we look at efficacy and safety across dose in cohort 5 for HER2 exon 20 patients only, we can see that the 16 milligram QD arm has the highest proportion of responses compared to the other dosing arms. We also investigated 10 milligram QD and 6 milligram BID dose. However, they were deemed to have lower performance. Now let's examine the tolerance of various dosing arms. Grade 3 or higher related AEs were higher in the 16 milligram QD arm, especially the on-target side effects of rash, in line with the higher efficacy of 16 milligram QD. The proportion of patients who needed to reduce or interrupt their dosing was not much different across dose levels. Overall, the efficacy and safety data support 16 milligram once daily continuous dosing for poziotinib. Following extensive dose exploration in this rare patient population, 16 milligram QD is a safe and effective starting dose to address an urgent medical need in patients who need therapeutic options. Now let me turn to our confirmatory study. The study has been initiated to confirm the clinical benefits seen in cohort 2 as required for an accelerated approval. The study is designed to enroll 268 patients with previously treated non-small cell lung cancer harboring HER2 exon 20 mutation in up to 150 sites globally. Patients are being randomized 2-to-1 to 8 milligrams of poziotinib orally administered twice daily versus docetaxel. Based on data available at the time of discussion with the agency and on the evolving PFS data from cohort 5, 8 milligram BID was chosen as the poziotinib starting dose. The primary endpoint is PFS, and the study includes a futility analysis planned for 2024. The study was originally designed based on the results from cohort 2 and stage 1 of cohort 5 dose-ranging study. Stage 1 analysis of cohort 5 showed 8 milligram BID had similar efficacy and possibly better tolerance. The dose for the PMR was determined after discussion with the agency. Given the urgency to start the confirmatory trial, the agency and Spectrum decided to initiate the trial at 8 milligram BID. 16 milligram QD remains safe and effective, but the 8 milligram BID shows a trend to slightly better tolerability. We remain committed to working with the FDA to amend the protocol if necessary. Here is the current snapshot of the study status. Spectrum has targeted 448 sites across 29 countries. A total of 97 sites have been qualified as of August 22. Based on the enrollment projection, we expect a futility analysis to be completed in 2024 and final analysis in 2026. In our presentation today, we've addressed the key points raised by the FDA and showed that the benefit of poziotinib outweighs the risks in a patient population who currently has no approved oral treatment options. Thank you very much for your attention, and let me turn the podium to Dr. Sosinski. Mark?
Thank you, Dr. LaBelle. I'm Dr. Mark Sosinski, and I'm a thoracic medical oncologist and the Executive Medical Director of the Advent Health Cancer Institute. I have 30 years of experience treating patients with lung cancer and was an investigator in Study 202. Patients with lung cancer harboring HER2 exon 20 insertion mutations are in desperate need for additional treatment options, and poziotinib would be a welcome addition for these patients. Treating these patients has become increasingly complex, and I am here to share my clinical experience. Let me begin by sharing an example of the anti-tumor activity of poziotinib in a patient from cohort 2. As you can see on the screening CT scan, the patient had a liver metastasis as well as abdominal wall metastases. On the bottom row, after receiving poziotinib, there is a clear and significant reduction in measurable tumor with durability of the response lasting 9+ months or 11 cycles. There isn't a patient in my clinic that wouldn't welcome access to a drug that would potentially induce a similar response. Lung cancer consists of multiple molecular subgroups, some of which are harder to drug than others. Patients have benefited from having multiple targeted options available for these subgroups, especially if they are mechanistically distinct. As an example, as Dr. Haymack pointed out, amivantamab, a bispecific antibody, was approved for EGFR exon 20 insertion mutations. Shortly thereafter, mobocertinib, an oral TKI, was also approved with a 28% overall response rate in the pivotal study, similar to poziotinib. Likewise, for KRAS G12C mutations, in the randomized confirmatory study presented last week at ESMO, sotorasib demonstrated a 28% overall response rate in a median progression-free survival of 5.6 months. Keep in mind, poziotinib had an overall response rate of 28% in the heavily pretreated group and 45% in the treatment-naive group. These are different molecular subgroups, so results shouldn't be compared to one another, but it illustrates that for hard-to-treat subgroups, such as these, the response rates are not only higher than docetaxel, but also demonstrate meaningful clinical benefit. In terms of risk, EGFR TKI adverse events have been routinely managed for well over a decade or so as part of the day-to-day supportive care carried out by our oncology nurses and oncologists in practice. We can see that poziotinib's safety profile, shown in blue, is in line with a number of these TKI agents, including the recently approved mobocertinib and dexamethasone. Based on the data here and my clinical experience with this drug, the on-target adverse events are both predictable and manageable. Compared to the other options of chemotherapy and HER2 ADC treatments, the lack of myelosuppression and its consequences, absence of cardiac toxicity, and a very low rate of ILD provide an option that may make it a treatment of choice in selected patients. As I reflect on what I've heard today, these data are particularly important to me. Poziotinib showed a meaningful advantage over available therapies in the second and beyond lines of treatment. Let me remind you that there are no currently approved drugs for third-line patients who have exhausted all options, including docetaxel. Note that in this heavily pretreated setting, poziotinib showed an overall response rate of 37% with a median progression-free survival of 5.5 months. I believe that for these heavily pretreated patients with no other approved options, the benefit of pozi is clear. Let me illustrate this in the context of the treatment landscape. Earlier in the presentation, Dr. Haymack shared the current treatment landscape and unmet need for patients diagnosed with advanced HER2-positive exon 20 insertion disease. Patients need more options and more time. With the data that you've seen today, poziotinib would be an option at many points in the journey of the patient. This could be an option after HER2 ADC treatment or as a second-line option in patients who prefer oral treatment or who have pulmonary risk factors that make them not suitable for trastuzumab deruxtecan. And it even has activity after docetaxel as demonstrated. In all of these cases, the approval of poziotinib would give my patients more options and potentially thus more time. So in conclusion, poziotinib represents an important advance for patients with HER2 exon 20 insertion mutations. The data you've seen today demonstrated poziotinib has clear efficacy in this molecularly defined population of patients with an urgent unmet need. It has manageable safety profile similar to a number of other currently approved FDA EGFR TKIs. Lastly, poziotinib would fit into a number of different treatment scenarios we face in the lung cancer clinic every week, either immediately after first-line chemo or after the other subsequent lines of therapy. I would welcome poziotinib as an option for patients with this disease. Thank you.
Thank you. We will now proceed with the FDA presentations from Dr. Justin Molyneux and Dr. Jean Fourie-Silkerback.
Good morning. I am Justin Molyneux, medical oncologist at the FDA. I would like to acknowledge all the members of the multidisciplinary review team. The applicant is seeking accelerated approval of poziotinib for the treatment of patients with previously treated advanced non-small cell lung cancer harboring HER2 exon 20 insertion mutations. As you have heard, there are several major review issues for this application. The efficacy, as demonstrated by the low overall response rate and poor duration of response, does not represent an advantage to patients over current therapies. The safety profile for poziotinib demonstrates a high level of toxicity requiring a large number of dose reductions and drug interruptions. You will hear from clinical pharmacology that the applicant did not adequately justify their dose selection and prematurely moved forward with the 16 milligram daily dose. The confirmatory trial has not yet enrolled any patients, which will significantly delay confirmation or refutation of the drug's benefit. I will describe the FDA's risk-benefit assessment based on the major review issues. The discussion and voting question for the committee will follow. First key review issue is the limited response rate with poor durability observed in the primary efficacy population. Patients with HER2-mutated non-small cell lung cancer who progress after first-line therapy may be treated with either chemotherapy or docetaxel plus ramucirumab, which offers a response rate of 23%. Single-agent immunotherapy yields response rates of up to 20% with substantial duration of response of at least 16 months. This is an option available to patients who did not receive immunotherapy in the frontline setting. Trastuzumab deruxtecan was recently granted accelerated approval specifically for patients with HER2-mutated non-small cell lung cancer. The approval was based on single-arm data with a response rate of 58% and a median duration of response of over 8 months. Key demographic information for Zenith 20 is shown. We would like to highlight that roughly 1/3 of patients were not treated with immunotherapy prior to enrollment. Therefore, immunotherapy is considered an available treatment for these patients, as discussed on the previous slide. As you have heard, poziotinib yielded an overall response rate of 28% with a lower limit of the confidence interval at 19%. For patients who received both platinum chemotherapy and immunotherapy, the overall response rate is unchanged. The median duration of response was 5.1 months, and of the patients that responded, only 24% had a durable response lasting 6 months or longer. Poziotinib's duration of response is considerably lower than all other recent targeted therapy approvals. We do not consider these efficacy results to demonstrate a meaningful benefit over currently marketed therapies. To put this in context of current therapies, this slide shows that poziotinib yields a similar response rate to docetaxel plus ramucirumab. The durability of response to poziotinib is similar to that of single-agent chemotherapy. For the 1/3 of patients enrolled in the primary efficacy population who did not receive immunotherapy, single-agent pembrolizumab or nivolumab offers a modest response rate and up to 17 months of durability. Thus, the FDA asserts that the efficacy of poziotinib does not represent a clinically meaningful benefit over current therapies. Patients experienced high rates of toxicity with poziotinib at the 16 milligram daily dose. The applicant states that the safety profile of poziotinib is similar to other drugs in class. However, in our assessment, poziotinib is more toxic than other tyrosine kinase inhibitors for lung cancer, especially at the 16 milligram dose. 8 of 10 patients experienced grade 3 to 4 adverse events. Similarly, over 80% of patients required a drug interruption and almost 60% of patients needed a dose reduction. You will hear from Dr. Fori Zirkelbach that these frequent treatment modifications may impact the overall efficacy of poziotinib. The applicant states that poziotinib may provide an alternative for patients who cannot tolerate antibody drug conjugates such as trastuzumab deruxtecan. in part due to the known risk of pneumonitis associated with these therapies. However, there were serious pulmonary events, including severe cases of dyspnea, pneumonia, and 8 cases of pneumonitis in patients treated with poziotinib. Patients also experienced serious events of diarrhea and acute kidney injury. In the overall 16 milligram daily population, 26 patients, or 7%, had fatal adverse events. Included in the 7% are 3 patients who died from pneumonitis. There were other fatal respiratory events, including respiratory failure and pneumonia. Given the rate of severity of pneumonitis and other pulmonary toxicities, poziotinib's safety profile does not represent an advantage over other treatment regimens. Our FDA analyses show that the rates of fatal adverse events seen in patients who received poziotinib are 3 to 5 times higher than those seen in recently approved targeted therapies. Patients treated with poziotinib experienced high rates of rash, diarrhea, and mucositis. Almost 50% of patients with rash experienced severe symptoms and required drug interruptions. A quarter of patients experienced grade 3 or 4 diarrhea. Almost 1/3 of patients with diarrhea required drug interruption, and 1 out of 5 patients with diarrhea needed a dose reduction. And while it is known that TKIs cause both rash and diarrhea, in the next few slides, I will show you how poziotinib compares to other recently approved drugs in class. For example, the incidence of rash in patients treated with poziotinib was 92%, which is substantially higher than most other approved therapies. Similarly, rates of diarrhea with poziotinib are also high relative to other targeted therapies for non-small cell lung cancer. I would like to point out that the applicant has brought up mobocertinib a few times, though with this drug, although with similar ORR, had a substantially longer DOR, an ongoing confirmatory trial, and did not have the dosing issue seen with poziotinib. In multiple analyses of cancer and therapy-associated symptoms, diarrhea has been found to be highly associated with decreased healthcare-related quality of life and with social functioning. It is important to note that according to the CTCAE grading scale, grade 2 diarrhea is defined as an increase of up to 6 stools per day over baseline, and grade 3 indicates hospitalization. Patients with cancer often cite low-grade chronic diarrhea as having a negative impact on their quality of life. In Zenith 20, 82% of patients experienced grade 1 to 2 events of diarrhea, with 1/4 of patients requiring hospitalization due to grade 3 to 4 diarrhea. Almost 1/3 of patients interrupted treatment, despite treatment interruption not being recommended in the management algorithm until grade 3 diarrhea occurred. This may indicate that lower grade diarrhea may have been considered intolerable by the patients, resulting in therapy modification. Patient-reported outcomes, which may have assessed patients' perceptions of the tolerability of the adverse events observed, were inadequately collected in Zenith 20. The applicant used 6 questionnaires to collect patient-reported quality of life and lung cancer symptoms in a sparse schedule as listed on this slide. Because of the infrequent and incomplete collection of patient-reported symptoms, tolerability of poziotinib is not clear. Specifically, the severity, duration, and trajectory of notable side effects such as diarrhea, stomatitis, and rash were not adequately captured. In terms of efficacy, No meaningful conclusions can be made as there was no pre-specified PRO hypothesis and the majority of patients did not provide a PRO response after cycle 3 due to attrition. Overall, the applicant did not assess patient-reported outcomes in a frequent and comprehensive way to better characterize the tolerability of poziotinib. The third key issue is the inadequate dosage optimization throughout the poziotinib development program. You will now hear from Dr. Jean-Faurie Zirkelbach, FDA Clinical Pharmacology Team Leader, for further discussion of this topic.
Good morning. I am Dr. Jean-Faurie Zirkelbach, a clinical pharmacologist at FDA. FDA's concerns regarding dose optimization date back to 2017 when the trial was initiated. We recommended studying a lower daily dose within the efficacious range of 12 to 16 milligrams daily due to the lack of information differentiating the 16 milligrams once daily dosage from alternative dosages. When FDA reviewed the top-line data in 2020 and 2021, we reiterated our concerns regarding efficacy, safety, dose selection, and the delay of confirmation of benefit. In an effort to accelerate the confirmatory trial, the applicant moves forward with a different dosage of 8 milligrams twice daily for their randomized trial. However, FDA continued to reiterate the need for additional data to support the proposed dosage. Initiating the trial absent these data was at the applicant's risk. Clinical data at the proposed dosage show that poziotinib has marginal activity with a high rate of toxicity. Limited data are available for other dosages, and it is uncertain if alternative dosages can maintain effectiveness and improve tolerability. Therefore, additional dosage optimization is still warranted. As of today, FDA's clinical pharmacology team does not have sufficient information to determine the optimal poziotinib dosage. In a preliminary study in patients with advanced tumors, dose selection was based on a maximum tolerated dose approach. Dose escalation occurred at doses of 12, 16, 18, and 20 milligrams administered once daily. 16 milligrams once daily, as shown in the highlighted box, was selected as the applicant's recommended dosage. Because the patient cohorts were so small, for example, only 3 patients were enrolled in the 12 milligram cohort, there were very limited safety and preliminary activity data available. These insufficient data were not adequate to identify differences in safety or activity within this dosage range. Thus, the available clinical data did not support selection of 16 milligrams once daily for the registrational trial. The applicant's recommended dosage of 16 milligrams once daily was investigated in cohort 2 to support the proposed indication. Based on FDA feedback, alternative dosages, including once daily and twice daily regimens, were later explored in cohort 5. The evaluation of safety and effectiveness for these alternative dosages is still ongoing. In the next few slides, I will summarize the available data from these alternative dosages, along with the exposure response analyses for safety and effectiveness. You are aware that the ORR of poziotinib at 16 milligrams once daily in the proposed patient population was 28%. In looking at the ORRs and the confidence intervals for the alternative dosages being investigated, the response rates again appear similar to that of 16 milligrams once daily. The confidence intervals are wide and overlapping, presumably due to small patient numbers. Though the applicant did provide additional data at alternative dosages as requested, the patient numbers remain insufficient to make a determination regarding the optimal dosage. Preliminary exposure response analyses do not support a dosage of 16 milligrams once daily compared to the alternative dosages. Exposure response relationships for efficacy are inconclusive due to the limited data at dosages other than 16 milligrams once daily. However, when we look at safety, we see that higher average concentrations are associated with a higher risk of grade 3 or higher treatment-emergent adverse events and adverse events leading to dose reduction and treatment discontinuation. Thus, while we know that higher dosages do yield greater toxicity, it is not known if alternative dosages will yield comparable efficacy to the 16 milligram daily dose. Given the high rates of interruption and dose reduction at 16 milligrams once daily, the relative dose intensity was only about 12 milligrams per day. This means that the average patient only received 12 milligrams once daily, or 75% of their prescribed dose, largely due to toxicity. The first dose interruption occurred at approximately 29 days following treatment initiation and lasted about 8 days. Given the elimination half-life of poziotinib is approximately 6 hours, no drug would be found in the systemic circulation within 2 days after withholding poziotinib. It is uncertain whether these prolonged interruptions to manage toxicity have an impact on efficacy. Most patients enrolled in Cohort 2 received a reduced dose within the first month, as illustrated by this graph. The dark blue shaded area shows the relatively short treatment duration at the proposed dosage. The black dashed line shown here marks the 6-week point after initiation of poziotinib. As shown by the dark blue shaded area, at this point, less than 50% of patients remained on 16 milligrams once daily. The black dotted line, shown here, marks 24 weeks or 6 months after initiation of poziotinib. As shown by the lighter blue and orange shaded areas at this point, most patients received dosages of 12 milligrams daily or less. The red shaded area shows the fraction of patients who did not receive poziotinib due to treatment interruption. In FDA's review of the applicant's clinical pharmacology package, we identified significant areas of concern regarding the lack of dosage optimization. Given that the applicant has provided insufficient data over the clinically relevant dose range, we cannot determine if alternative dosages may provide acceptable efficacy and an improved toxicity profile. Therefore, we continue to assert that the applicant failed to adequately justify their proposed dosage of 16 milligrams once daily. I will now turn your attention back to Dr. Malino to complete FDA's presentation.
The final review issue is a significant delay in confirmation or refutation of clinical benefit. As a reminder, in the applicant's planned confirmatory trial, patients will be randomized to either poziotinib 8 milligrams twice daily or single-agent docetaxel, with progression-free survival as the primary endpoint. They are targeting a 2.5-month PFS event benefit, which may not be clinically meaningful. The trial has not enrolled any patients as of this month and is not slated to read out until 2026 at the earliest. Patients could be exposed to a highly toxic drug with unverified clinical benefit for at least 4 years. Now let's consider this application within the framework of the FDA's risk-benefit assessment. The FDA considers the totality of evidence when making risk-benefit determinations. We recognize that this is a rare population with limited therapeutic options. However, given the marginal ORR and limited DOR, FDA asserts that poziotinib does not represent a meaningful therapeutic benefit when compared to current therapies. Poziotinib has a high level of toxicity with high rates of grade 3 to 4 AEs, serious AEs, fatal AEs, dose reductions, and drug interruptions. The dose proposed for marketing is not currently optimized. In addition, there are no definitive plans for alternate dose exploration beyond 8 milligrams twice daily. The selection of the 8 milligram twice daily dose for the confirmatory trial is incongruent with the approval of a 16 milligram daily dose. Finally, initiation of the confirmatory trial is significantly delayed and places patients at undue risk. We know that accelerated approvals without a confirmatory trial initiated at the time of approval require several more years to verify or refute clinical benefit than those which already have a confirmatory trial underway. The recent approval of trastuzumab deruxtecan in the same space may make it infeasible to conduct a proposed confirmatory trial, further delaying confirmation or refutation of benefit. I will now present the discussion topics and voting question for the advisory committee. We ask that the committee discuss the overall risk-benefit of poziotinib 16 milligrams daily given the following: its low response rate with poor durability, high rate of toxicity, lack of dosage optimization, and significant delay in confirmation or refutation of benefit with a randomized trial. We ask that the committee vote on the following. Do the current benefits of poziotinib outweigh its risks for the treatment of patients with non-small cell lung cancer with HER2 exon 20 insertion mutations? Thank you for your attention.
Thank you, Dr. Malino. We will now take clarifying questions for the presenters, Spectrum Pharmaceuticals Inc. and the FDA. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak, and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and end of your follow-up question with a that is all of my questions, so we can move on to the next panel member. So let's just go to the committee. You know, we have heard the FDA presentation, and we have heard the applicant presentation as well. And I would like to open the group for us to ask the questions to specific people. So let's start with Dr. Liu.
Hi, everybody. This is Chris Liu. This question is for the FDA, and I just want to clarify the status of trastuzumab deruxtecan for this. It's been brought up in the presentations, and we've seen the overall response rate, but we need to know if this is considered to be an available therapy or not, just in terms of how we view the current data.
Hi, this is Harpreet Singh, Director. I will take that question. Yes, let me I agree that there is a lot of discussion around trastuzumab deruxtecan. And before I answer your question directly, there is a point of clarification from a lot of the data that the sponsor cites in terms of rates of ILD and pneumonitis. They're actually citing, particularly in Dr. Haymack's presentation, toxicity data from the higher dose, the 6.4 mg per kg dose. We did not approve that dose. We asked the company, much like we asked Spectrum, to conduct a dose-finding study comparing the 6.4 dose to a lower dose because of the toxicities we were seeing. They did do that study. We did receive the results of that study during the course of our review, and thus we approved a lower dose, 5.4 mg per kg dose, which had substantially less ILD and pneumonitis, pulmonary toxicity in general, and maintained a 50% or so, 50 to 60% response rate. So I'd like to make that point of clarification because I did feel that that particular toxicity information comparing the two question about availability of therapy. Available is a regulatory issue. And we don't necessarily want the committee to, make this too complicated. The point is that Enhertu or trastuzumab deruxtecan is clinically available to providers. Yes, it is under accelerated approval. This means that from a regulatory standpoint, when we are comparing available therapies, we are not looking at that 60% response rate as the bar to beat, so to speak. However, I think this has implications in terms of the confirmatory trial, which both Dr. Dresner and Dr. Malinow point out. How feasible is it, will it be for Spectrum to conduct this confirmatory trial given that trastuzumab is available to providers with a 60% response rate? So I think that is really, how this comes into play. But then back to even if you are considering it from an FDA regulatory perspective, which we're not asking you, the committee, to do, that's our role, we still assert that poziotinib does not represent a meaningful advantage, even if you take trastuzumab off the table. So I hope that answers your question, but I would like to bring in Dr. Julia Beaver if she'd like to contribute anything to that.
Yes, thanks. Hi, this is Julia Beaver. Exactly as Dr. Singh stated, for the purpose of regulatory purpose of available therapy as defined in our guidance, that is therapies approved under regular or traditional approvals or set standard of care. Those are regulatory distinctions we will consider. We'd like the committee to view this from a clinical standpoint, from risk-benefit, and as Dr. Singh mentioned, we want the committee to consider if the availability of Enhertu will impact the feasibility to enroll patients on the confirmatory trial. We also note that the sponsor mentioned a number of times potential sequencing of their drug, potentially even after a trastuzumab-based ADC, which is not something that we are aware of has been studied. So we do not feel that that particular indication would be appropriate. Thank you. That is all.
That answers my question. Thank you so much.
Thank you. Dr. Madan, you have a question?
Yes, hi, Ravi Madan, NCI. So I have a question for the sponsor regarding the unmet need. So there's assertions that the trastuzumab is perhaps not available for patients with certain pulmonary conditions and that poziotinib would be an option for those patients. Does the sponsor have any data in those specific populations demonstrating safety and efficacy?
Thank you for your question. I'd like Dr. Amack to address this question.
Yeah, and with that, I'll also bring up something that Dr. Singh had mentioned earlier. In my presentation, I mentioned the doses both at the 6.4 originally in the Destiny-01 study where 26% was seen, and then with the dose reduction to 5.4. Now in Destiny-02, and this data has not been presented publicly for us to review, so we're basing it on the package insert. But for Destiny-2, they did two things. They lowered the dose from 6.4 mg per kg to 5.4, but the eligibility were also changed. So here from clinicaltrials.gov is the eligibility criteria for poziotinib versus the trastuzumab deruxtecan in this space. And so you can see poziotinib has a standard grade 2 pneumonitis or high pneumonitis exclusion, which is seen in all studies in this space. For trastuzumab deruxtecan, they said not only a history of non-infectious interstitial lung disease requiring steroids or the other things, or suspected ILD or pneumonitis that can't be raised out, but they also said other conditions that put you at pulmonary risk, including pulmonary emboli within 3 months of the study, severe asthma, severe COPD, restrictive lung disease, pleural effusion, etc. Now in our study, we know 17% of patients had pleural effusion as an example. We know that COPD and restrictive lung disease are common in this population. And we know that if you take the lung cancer population as a whole, 19% have some history of pneumonitis in the past, often restricted pneumonitis. So the point here, and I know this meeting isn't about trastuzumab deruxtecan, but it's to say that what they've done in the Destiny-02 is exclude a substantial number of patients that have pulmonary risk factors that are extremely common for our lung cancer population, including the 17% in our study that did have pleural effusions treated with poziotinib.
So just to clarify, do you have any safety or efficacy data in those 17% of patients?
We haven't broken out the 17% with pleural effusions. We do have data for patients treated with prior HER2-directed therapies. As was mentioned, the response rate was 24%. Median PFS was similar to the overall population, 5+ months. And specifically in patients that were treated with TDM1, which was the HER2 ADC that was available at the time. Again, trastuzumab deruxtecan wasn't available during the poziotinib study, so there isn't a history of many patients. There was, I know at least some there on the study, but we had 6 patients treated with TDM1. The response rate in that group was 33%. And you saw the response rate with prior HER2 TKI. So obviously the study wasn't powered for these rare subgroups, but the message was that activity was consistently seen regardless of prior immunotherapy, HER2-directed therapy, and so forth.
Okay, but just to clarify, in the unmet need discussion, the patients with these pulmonary conditions, you don't have data showing safety or efficacy.
Right. One could search for that data, but I'll say that the trastuzumab deruxtecan data hasn't been reported publicly, so we don't know all the different conditions. But if we
I was asking about the poziotinib data.
Right. Right. Okay. But we could pull out the pleural effusion subgroup, for example, to look for efficacy.
Okay. One other question along the same lines is, there's this kind of implication that an oral medication would be more accessible, even though it's a novel agent that's, under special approval. it would be more accessible than an IV agent that's administered every 3 weeks, although it still require access to probably, a pretty cutting-edge oncology service. Is there any data that actually demonstrates that oral agents are more accessible to patients than an IV agent?
Yeah, I'm going to ask thank you for your question. I'm going to ask Dr. Sasinski to address that. Thank you, Dr. Sasinski. I'm not aware of any specific data with regard to that. I can tell you my clinical experience, since in the field of lung cancer, a number of the targeted agents are obviously oral agents. And it's not unusual when you have a situation where you have an oral and an IV after patients have received first-line IV therapy that they kind of want to break from the IV experience. So my clinical experience is that patients often, very commonly do favor the oral route of administration because of the convenience. I view many of these situations as a plan for sequential therapy. So whether you do one first, the other one, that sort of thing, from a clinical point of view, we have those sorts of discussions, and patients often prefer the oral route that's more convenient. Thank you.
I thank the sponsor for taking time to answer my questions. That is all.
Thank you. Dr. Waldman, do you have a question?
I do. This is Scott Waldman, Thomas Jefferson University. I have two questions. First question is for the FDA. And this plays off of a little bit off of Dr. Liu's question that was asked before. I understand that trastuzumab deruxtecan doesn't set the bar for response rates and duration of response. I understand that. But can we consider it that it's part of the armamentarium that's available to the questions, to the patients that we're considering here? In other words, I understand it doesn't set the bar, but is it available when we consider what's available and what gaps poziotinib might be filling? Can we consider this agent available to the patients?
Hi, this is Harpreet Singh, Director. I will take that question, and I understand that this particular point is causing a bit of consternation, so I will take this, and then I will ask Dr. Richard Pazder to come on and maybe clarify any fine points. But I just want to make one comment on the last question about oral therapies being available and being preferred regimens. I want to make 2 points about that. Yes, certainly patients may prefer oral options. However, I would say that first, they would not prefer an oral regimen that is inferior to what is available IV, whether we're talking about a RAM, docetaxel, or an IO, or to get to this point about trastuzumab. And secondly, the sponsor did not adequately collect patient-reported outcomes data, as we highlighted. And so we know that chronic, daily, low-grade, and with this drug, high-grade toxicities like diarrhea and rash really contribute to a negative or, contribute negatively to quality of life. Back to this question about what is available. Yes, you may consider trastuzumab as available to providers. It is available to you, the oncologist, in clinic every day. And what we are asking ourselves, and you must ask yourself, if you were presented with a patient with this rare mutation and you have the option of pulling this ADC off the shelf, which has 2 other indications, is known to providers, versus enrolling them on this confirmatory trial, which we are telling you the dose may not be optimized, and the toxicity, and the response rate, and that the comparator is single-agent docetaxel, what would you choose? And I think that's something we're asking the committee to consider when we think about the context of what's available to you in the clinic. Let FDA, worry about the regs and the bar. So in answer to your question, yes, I do believe you should consider this as available to you as providers, because it is. Dr. Pazder, would you like to add any further comment to this?
Well, let me go through what the guidance actually says. And I was partly responsible for putting this in guidance is that we would consider in making a regulatory decision, okay, that available therapy is the approved therapies that we have and not those under accelerated approval. So that's in FDA guidance. I think the reason why we have brought this up, the issue of this particular drug, here again is the issue of having a trial that has not been yet initiated. Is it conceivable really to do this trial? I think that was one of our major points of view. Here again, at the end of the day, if you could see our question that we're asking you to vote on, we're not asking you whether or not the drug should be approved. We're asking you at this point in the context of what is out there, does this represent basically a positive risk benefit for patients here again. But in making that regulatory decision, the FDA will not consider basically the drug under accelerated approval as available therapy. Okay?
Okay. Again, Scott Waldman, I have one more, and thank you for the answers. I appreciate that. I have one more question for the sponsor, Spectrum. There's a disconnect between the dose of poziotinib that you're going after in the confirmatory study, 8 milligrams BID, and the dose that you are seeking approval on, 16 milligrams QD. It's clearly a disconnect. Can you address the disconnect? Because I didn't get it from your original presentation. How those two facts are congruent with each other. They seem incongruent to me.
Yeah, sure. Thank you. We believe that dose is clear. It's 16 milligrams per day given in the QD or 8 milligram BID. I think the differentiation between those two from an efficacy point of view, there's a tremendous amount of overlap. So one cannot really claim one better than the other. However, upon, additional information
this is Dr. Singh. I cannot hear you very well, and I'm getting word from my team that this is universal. I really do want to hear this response. Could we try to work on the audio?
We believe that the audio has been a problem. We missed part of the comments from Dr. Pazdur.
Dr. Level, we still cannot hear you really well. You're breaking up. Dr. Chen, do we know if he's reconnecting?
Hi, Dr. Garcia. This is Shija Chen. Just a moment. Let me chat real quick. Thank you for all your patience.
I'm looking forward, this is Dr. Garcia, I'm looking forward to getting back to in-person meetings sometime soon. This is the virtue of technology, which still is pretty lumpy for all of us.
Okay, the sponsor should be reconnected. Thank you.
Yes, this is Dr. LaBelle. Can you hear me now, Dr. LaBelle here?
Yes, please proceed.
Okay, thank you. So as I was saying, I don't know when broke off, so I'll start from the start. So we believe the dose is quite clear in 16 milligram per day.
is the dose that was shown to be safe and effective in cohort 2 and was supported by 2 independent studies. We did, the body of evidence clearly over time has evolved, and it seems like the 8-BID would have the same activity and potentially a slightly better adverse event profile. So there were extensive discussion with the agency. I think there was at least 5, including as late as March of this year, in terms of arriving at the final dosing for the PMR. There was obviously, there's an urgent need to be addressed, and also we wanted to be in compliance and initiate the study. So after many discussions, the agency and us agreed that the 8 milligram BID would be the way to go forward. I would additionally mention that we have, in part of my formal presentation, that we're certainly open to additional discussion if another dose or schedule would be identified. But we believe that patients needed that option as soon as possible.
So this is Scott Waldman again. Can I ask a follow-on clarifying question of the FDA, please?
Please go ahead, Dr. Waldman.
Thank you. Thank you very much. So hearing that, this is a regulatory question. Is it the case that for an accelerated approval of a drug, if the accelerated approval is at 16 milligrams q.day, and the confirmatory study is at 8 milligrams BID, does that confirmatory study support the accelerated approval of the original dose? Again, I'm dealing with this incongruity, and I neither understand it from a drug development perspective nor from a regulatory perspective. Sorry. Apologies.
Yeah, this is Harpreet Singh, Director. Thank you for the question. So let me just respond briefly to the sponsor, because I do feel, although Dr. Fori-Zirkelbach did state in her presentation, let me reiterate, we did not come to any formal agreement about the 8 milligrams BID dose. We had very limited data, and we told the sponsor that it would be at their own risk to move forward with this incongruent dose. The safety ER analysis, exposure response analysis conducted by our clinical pharmacologist shows fairly comparable safety profiles between the 8 milligrams twice daily and the 16 milligrams once daily. I think a lot of this speaks to the sponsor kind of rushing this development program and trying to take catch-up steps, whereas the steps should have been taken slowly and methodically and appropriately early in development. Basically you're asking the question, how will FDA deal with a different dose in the confirmatory trial versus the dose which was studied? Frankly, it's a bit of uncharted territory. I don't think it's a question we need to deal with today, but certainly if the accelerated approval is granted, if then the confirmatory trial is conducted, if it is a positive study with this different dose, then we will be faced with a labeling challenge, obviously a point of confusion for patients and providers. But I don't necessarily find this to be the most problematic issue that we're faced with. I don't know if that answers your question directly. I'm going to call for backup from my team if anyone would like to add anything to that. But I don't think that's kind of the major issue about whether or not, whether there's a different dose in the confirmatory trial versus the accelerated approval. It is an issue. But I think that if everything lined up, we would manage that later, where we would probably, look at adding all of this information to the label, making a decision at that point with the data in hand. Right now, we cannot answer that question. We frankly do not have enough data from the 8 milligram BID dose
to kind of sanction it, so to speak, as an approvable dose moving forward. So give me one moment. Let me.
Yeah, hi, this is Rick Pastor. I just want to emphasize to you that your confusion is well warranted, okay? Because what this really represents is poor drug development. Obviously, before somebody launches a large phase 3 trial, they should have confidence in what their dose is. And the dose optimization should occur beforehand. What would we do if the large phase 3 trial, if it can accrue, and that's a big if, is negative? Is it because they chose the wrong dose? We have a whole program here at the FDA on dose optimization, and this just points to one of the problems that we have here. When one attempts to launch a phase 3 study without adequately looking at what the dose is and having confidence in it. I've said this before, and I'll say it again. Proceeding with a drug development program when you don't have a well-founded dose is literally building a house on quicksand here. And this is one of the problems. Your confusion regarding this point is well taken.
This is Scott Waldman. I appreciate the enthusiastic discussion, and I'm finished with my questions.
Thank you, and thank you to the FDA for their answers as well. Next will be Dr. Thomas. Do you have a question?
Hey, my name is Thomas, NCI. So maybe just to follow up on the previous question, another area that I found, quite a disconnect and a lack of congruence is the phase 3 study. Is the confirmatory trial really underway? I mean, the FDA documents seem to indicate that, it's much delayed, but the sponsor's discussion seems to suggest that it's well underway, although patient is not enrolled. Where is that at this point? Is there any, some of the concerns were shared about enrollment? Is that going to be a problem? So that's sort of one question.
So thank you for the question. Let me bring up the slide. So yes, the study is underway. When you do, I'm sure you can appreciate, and many members of the committee would understand, when you do a large global randomized controlled trial, it takes time to get things going. And, if you go to average academic center, for example, in the U.S., it can take as much as 6 months to get through the various committees. And it can be in other places as long as a year. So we have a body of experience here. We have done the largest collection of patients with exon 20 mutation in HER2 and others globally. We have, and we are connected again with these sites. I believe there's 50 of them. And they're very enthusiastic about getting going. We have 3 sites open right now. We're going to get close to 30 in the next few months. And we're targeting many more sites. We have, different from what I have on the slide here, we actually have 107 sites qualified. We are filed in various countries. We recently got approval in Korea, where we plan to have 8 sites, and we're in various stages of activation of the trial. Our best enrolling site, if you look at the top 5 enrolling sites in the past, are all coming in, and they're in various process of approval at their site. So this study is very much underway. We acknowledge there's no patient right now, but if you look at the curve on the right, we never expected patients probably at this stage, and we're on track. Clearly, we understand there's a bit of faith here involved, but we've done these studies before, and so we remain very optimistic, and we have good progress, and we're committed to carry out this study.
Thank you. Thank you, Dr. Lerner. I think Dr. Basler has a question or a comment. Dr. Pazdur?
Dr. This is Harpreet Singh. May I be permitted to respond to that, or is that acceptable?
Yes, please, Dr. Singh. Go ahead.
Okay, very quickly, I just want to say that, I think it's a great question from Dr. Thomas, and while there are no patients enrolled, it takes back to this, feasibility issue, whether or not patients in the United States would be feasibly enrolled in this trial, given what providers do have in their armamentarium, which we've discussed at length. And so if this study is completely offshored, so to speak, or completely done ex-US, this would be, antithetical to the entire oncology community statement and commitment to diversity and having a patient population that's reflective of the U.S. Patient population. So I do think that this is an important point. It's kind of an extension of this question. You hear the company talking about opening sites in basically in other countries. You must ask yourself why they must do this, and if we would get any U.S. patients in this study. So I just wanted to add that small point. Thank you for allowing me.
Thank you.
Chairman, can I?
Dr. Pazdur,
Yeah.
you have a comment?
Yeah, the company might be happy. We're not, okay? And let me make this real clear. And I'll refer you to an article that we just published in the New England Journal that came out yesterday, last night, on this whole topic. It's called the on-ramp and off-ramps of accelerated approval. And it has important numbers in there for you to consider, and I'll give you some of these. The issue here is we want companies to come in and have a comprehensive discussion with us very early on regarding what their plans are for accelerated approval and what their plans are to confirm their study, okay? And those in guidance, okay, FDA guidance that has been there for more than a decade, okay, clearly state that it is anticipated that these trials should be ongoing. And by ongoing, we mean accrual of patients to the study at the time of the accelerated approvals. To give you an idea, and let me quote some of the numbers out of that paper, which shows the importance of having the studies actively accruing patients. If the confirmatory trial was ongoing at the time of the accelerated approval, which was 66 actions, the median time to conversion was 3 years. If the confirmatory trials had not been initiated, it was 5 years. However, if you take a look at the trials that did not demonstrate or were not converted, it is very impressive that if the trials were ongoing, it's 3.8 years. However, if they were going, it was 3.8 years, and if they weren't, it was 7.3 years. So a significant delay. The other important point is not only the delay, but can the trial be done? And this is where we have gotten into problems with accelerated approval, is that sponsors agree to these studies and then they come back in a year and say, oh, we don't have sufficient accrual, and then we're looking at alternative trial designs. So really the point that we want to get across is that we want early discussions on these trials and them to be ongoing. And it's really only fair to the patients because you're really putting patients significantly at risk. And you're also putting your development program at risk because many times response rates will not really project what the true benefit of the drug is, and that will only be seen with a randomized study that looks at overall survival. In other words, these response rates may not be good correlates or surrogates for overall survival. So you could actually be abandoning drugs just looking at response rates. But the point I want to get across is really we want to have these trials ongoing, and it really is to the detriment of patients not to have these trials ongoing. And this has been in the FDA guidance for many months. And I will give you some additional numbers. We took a look at this. If one takes a look at the accelerated approvals over the past 3 or 4 years, it's probably 85% of these accelerated approvals have ongoing trials. So industry has heard us loud and clear, and this trial is obviously, or this application is not consistent consistent with many of the current trends that we're seeing with other sponsors, including the other drug that they keep on bringing up in HER2 had a trial that was actively accruing patients. I hope this helps.
It does. Thank you, Dr.
Chairman.
Roger.
Mr. Chairman, can I try to clarify 2 points that were made by the two last speakers?
Yes, you can. Please identify yourself so we know who's talking.
Yeah, sure. Dr. LaBelle from Spectrum. I just want to clarify where are the location of the sites. So the plan is to have about 20% of the sites in the U.S. We already have 3 open in the U.S. and we anticipate those are going to open pretty fast now over the next couple of weeks. And then separately, we're very aware to maintain and go in countries where the practice of medicine is not identical to the U.S., of course, but very much in line so that the data can be representative when we get it. As to the feasibility, we have 2 points to make. One, the first one is we clearly have been able to recruit the largest collection of HER2 exon 20 patients than anybody else, even though we're a small company. And then the last point is, given that there potentially is, well, there, you know, an available or approved drug in the market, and we think actually that it may enhance recruitment in the U.S. and obviously ex-U.S. the drug is not necessarily approved. Actually, and therefore there would be no real competing group there.
Yep.
Dr. Aimak, do you want to make an additional comment?
Yeah, to give a clinical perspective on this and expand on it, just to clarify, because I'm not sure this came through earlier. In the randomized phase 3, patients with prior trastuzumab deruxtecan or trastuzumab emtansine or any HER2 targeted therapy are eligible for the confirmatory phase 3 study. And that's a really important point because I've heard this raised as an important issue about the feasibility, but actually those patients are included. So in the same way that those patients were eligible for the poziotinib study, they were eligible for the confirmatory study. Now the reason this is important is when we have a brand new disease space, you know, up until this last month, there was no clinical reason that mandated checking for HER2 mutations for patients with lung cancer. But as of the past month, now with the drug approved, this means the standard of care will be to check for HER2 mutations. So the number of HER2 mutant patients we expect is going to dramatically rise, and many of those will be treated with prior trastuzumab deruxtecan or trastuzumab emtansine. And so that we believe will increase the total number of patients eligible for the clinical study. So given that those patients are eligible, I think this confirmatory study meets a really important unmet need for what happens after a HER2 ADC, whether patients receive it or don't receive it. If I could just make one more sort of clinical comment, you know, I've heard some points raised about, you know, the size of the different studies in terms of the dose modification. And to give a clinical perspective here, this is by far the largest study ever in this rare population. In fact, it's more than twice as large as any other study for HER2 exon 20 mutations that I'm aware of. So for studies like this, the amount of dose optimization done, I'm not aware of anything comparable. But from a clinical perspective, I think that our management would be similar with 8-BID or 16-Q-day. I think the differences from a clinical perspective in how we manage those will be similar. Patients, like with drugs we use like dexamethasone or afatinib, both of which the majority of patients get dose reductions in the randomized studies and in clinical use. We explain to patients that during the first 2 to 8 weeks they'll have side effects. We expect that they commonly will need dose modifications. And then we prepare them for that. And so this is not something that's unexpected. And just to remind people, with afatinib, FDA-approved, or dexamethasone for lung cancer, the majority of patients require dose reduction, but this doesn't prevent us from using it effectively. We prepare patients for this, we dose reduce where needed, and we're able to manage that clinically. So from a clinical perspective, you know, we think it's more important that patients have the ability to use the drug and let clinicians modify it as they're used to do it than delaying it for a while additional dose optimization is happening.
Thank you.
Thank you, Dr. Hymak, for your clinical insights. In the interest of time, we're just going to move on. We have a few additional questions from the committee members. Next will be Mr. Pantelas.
Yes, thank you. This is Jim Pantelis. I'm a patient representative. And I've got a question, I've got two questions for FDA. I understand the FDA's assertion that poziotinib may not present an option over existing and available therapies, but many lung cancer patients tend to look at survivability in more of an iterative fashion. As such, I found the proposal of use in the second and third line treatment reflected in the applicant's slide CO73. If we can put that up, that would be really helpful. I found that slide compelling. My first question is, can the FDA comment on this slide, including its validity and proposed use for the drug? And second, where will trastuzumab fit into this slide from the FDA's perspective?
Okay, this is Harpreet Singh. Thank you for the question. Let me just address 2 points that you raised. First, you say that, you know, FDA is asserting, which we are correctly, you are, in that we are asserting that poziotinib does not represent a meaningful advantage to patients over what is currently available. That is the legal requirement in the Code of Federal Regulations for accelerated approval. This is different than a regular or traditional approval in which the drug just has to be better than what it was compared to. Here, in the single-arm setting, it must be clinically either efficacy or safety-wise represent an advantage to patients over what they have available. So we do not believe this to be true based on what we've shown you. Now, your question about trastuzumab, I certainly understand how one could find this compelling. However, it is inaccurate and misleading, and I'll tell you why. And sponsors are very, sponsors often present slides that look like this. But the applicant is proposing basically to insert poziotinib post-trastuzumab. So, you know, as a sequencing, as part of your regimen for a patient who has this rare mutation, that poziotinib could come after trastuzumab. The fact that they're even, you know, placing it after, I think is indicative that they believe that most providers would reach for that ADC first. But I digress. So let's go back to the sequencing issue. This is not a population in which they studied. This is not the indication which they are seeking. They are seeking an indication for poziotinib post one prior therapy. They are not seeking an indication for poziotinib post trastuzumab. If they were to conduct a trial in which all patients receive trastuzumab and then poziotinib and then present us with that data and we're seeking that indication, the conversation may be different. But here you see the sponsor try to make the case for indications they simply have not studied. And therefore, we cannot consider this argument in our risk-benefit assessment. Dr. Beaver, is there anything you'd like to add to this?
Hi, Julia Beaver, FDA. Just to summarize the main points, for accelerated approval, the drug needs to represent improvement over available therapy. For that line of therapy it was studied in. And in this case, the available therapies are clearly taking Enhertu off the table. The available therapies, docetaxel, ramucirumab, and also includes IO, because not all patients in the poziotinib study presented for accelerated approval received that therapy. Therefore, IO is considered available therapy. We cannot invent new indications without data demonstrating benefit in those populations. And as such, it's purely hypothetical that poziotinib would have benefit in different lines of therapy or in different sequencing. Thank you.
Thank you so much.
Thank you. We have a few minutes for a couple of questions before the break. Let's go with Dr. Harrington. And please remind the committee members, once you have completed your question, if you can lower your hand, that would be great. Dr. Harrington?
Thank you. I have a question and a comment. I understand the values and the weaknesses of accelerated approvals, and clearly there's some uncertainty here regarding best to proceed with accelerated approval. Perhaps the FDA could clarify for me, if accelerated approval is not guaranteed, can the sponsor proceed, I believe they can, with their phase 3 randomized trial?
Yes, they may. Whether, them conducting this randomized trial is independent, we would hope, of whether or not accelerated approval is granted. I will quote Dr. Pazner here. Accelerated approval was designed and created for the benefit of patients. They are not meant to be a financial incentive or any incentive for drug companies to rush drugs to market. So we encourage and promote, and we sincerely hope that the sponsor, aggressively pursues this randomized trial to really demonstrate if there is true benefit of this drug in a time-to-event endpoint in a randomized setting. Absolutely, yes, they may. The two are independent of each other. Thank you.
Thank you. I think we're going to probably have one final question. We have to get to break. Dr. Halmos?
Hi, Chef. As a thoracic oncologist practitioner myself, actually sitting in clinic, I definitely recognize the unmet need for this patient population. Indeed, these are typically younger patients with limited, if any, benefit from immunotherapy. So that probably is not a fair comparison here, as highlighted before, and very limited benefit from second-line chemotherapy. Even if you consider trastuzumab deruxtecan, I think poziotinib, a TKI, oral TKI, could certainly be a potentially attractive additional choice. Very different mechanism of action, likely no cross-resistance, and reported CNS activity. I also really appreciate the tremendous effort it has taken to collect this large and unique experience in this orphan disease, and recognize that the study met its efficacy endpoint with some long-term beneficiaries, and actually carefully read the patient testimonial submitted to the FDA demonstrating the desire to have access to this option. However, certainly one has to be concerned about practically each patient facing significant toxicities. As much as these are anticipated and familiar ones to the clinician, after decades of EGFR TKI experience, this suggests a very narrow therapeutic window. There actually may be a perfect dose across the board but just not be definable. So my questions to the sponsor are there actually identifiable molecular or clinical features that could highlight populations with a higher chance of response benefit? And similarly, any clinical or PK features that would help identify patients at higher risk for excessive toxicity, where the potentially approved dosing clearly would not be advisable or safe in order to help enhance the risk-benefit profile. And if approved, what efforts will the sponsor take to further study these critical issues in real-world patient populations, understanding that the confirmatory study will take a long time to provide results, if at all feasible, which is indeed in question. Thank you.
Thank you for your question, Dr.
Absolutely. So this is Dr. Janelle Gray from Moffitt Cancer Center. I certainly understand the concerns that have been raised. And if, as a reminder of what was just presented by Dr. Hayma, we know in clinic that we are very familiar with taking care of patients with dose reductions, dose adjustments, managing these toxicities. You can see here for the second, third generation TKIs, and again, I'm showing data here that is in addition to what was included in the FDA data, that the slides that were just presented. You can see that over 50% of patients with afatinib, dexamethasone, drugs that we have treated over years and years in clinic and have a lot of experience, familiarity with, that these patients require a dose reduction. There's actually lower dose reductions with dexamethasone than there are with poziotinib. The discontinuation rates across these medications are similar. And in the MD Anderson study, interestingly, that only one patient required a discontinuation from medications in the poziotinib study. Another thing I want to just share with you is that, as clinicians, we are also very familiar with treating toxicities, right? This is across rash, this is across diarrhea. We do these every day in our clinic. This is my life every day in clinic. We look at poziotinib compared to some medications like mobocertinib with neratinib. The grade 3 or higher diarrhea rates are the same. We've been doing this already, so it's 21 to 22%. When in clinic, what do? I set expectations with my patients. I let them know that there is a big clinical team working together in partnership with them, for them to get this done. These patients, they have nurses, they have nurse navigators, they have my physician assistants, they have me to help them through this. And those expectations, I talk to them what's going to happen in those first 2 to 8 weeks. I let them know that we may need to adjust doses. We may change your medication dosing. We may talk about your dietary changes. We may prescribe more medications to take care of these toxicities. And that eventually we'll get to a point where we're stable and we feel comfortable. To your point about the education, I definitely want to allow the sponsor, when these drugs post-approval, we certainly know that there's going to be management of these toxicities that are going to be outlined in the package insert. There's going to be a vast amount of education, not just to patients, but also to providers. What resonates with me when a new drug gets approved is, what have I done before? Where have I seen this medication previously? And then I can say, okay, I know how to manage diarrhea from TKIs. This is no different. We also know that in the setting of recent approvals, that these medications, when I look at them and look at their outcomes, okay, it's very hard to think that these medications have similar overall responses, that I'm going to have to deny a patient access to poziotinib. We know the lung cancer burden. We recognize, as I heard on this, that access is something that's also very important. And the thought of delaying patients' access to a drug that could be potentially efficacious to them is something that is very, very challenging. And from my clinical perspective, we need this medication approved now for our patients and allow us, the clinicians who do this every day in clinic, to manage these toxicities that we've been doing for years over and over, and we feel comfortable doing this. And with that, I'll ask Dr. Straczynski.
Oh, okay. This is Dr. from that was a very long response. FDA has to respond to this, to the safety. We cannot move on to efficacy until FDA responds to safety, because there was some highly misleading information there. We have some backup slides that refute this information. And also, single-arm time-to-event endpoints were shown, PFS, which we cannot consider, were misleading, particularly for mobocertinib. So FDA, can you please pull up our backup slide? Dr. Malino, do you have the slide number?
Dr. Singh, I'm going to give you a few more minutes to address those comments. In the interest of time, we're going to have to take a break because we have a pretty busy agenda after the break. But please go ahead and finish your comments.
Okay, if we can't pull the backup slide, which I'll ask Dr. Malino to tell us, which is the slide that compares all the recent lung cancer FDA-approved targeted therapies and their efficacy. Mobocertinib keeps getting invoked. Yes, this had a similar response rate, but the duration of response was in excess of, I believe, over a year. It was quite long. Here we are. Thank you. 17 months for mobocertinib. There was no dosing issue with mobocertinib, so we were not struggling with the safety profile. It was established. It was clear. And, the sponsor keeps talking about how they have amassed the largest database in this rare mutation. Thus, I find it unfortunate that they have missed several opportunities at several steps along the way to optimize this dose. If they had so many patients with this mutation, they should have moved forward either or both
to adequately optimize the dose in larger patient cohorts rather than 3 patients per cohort as they did, or they should have initiated randomized trials. This is a fatal flaw in this development program, and we really needed to show this data in a different way. Look at this duration of response of mobocertinib. You cannot look at single-arm PFS data. It is not interpretable. So I will close with that.
Thank you, Dr. Singh. And in the interest of time, Dr. Roscoe, apologize. We probably will have the ability to have an active discussion in the group after the OPH. We're going to just take a quick break. We have to come back at 11:40 for the open public hearing. So take 6 minutes and we'll reconvene at 11:40. We will. We will now begin the open public hearing session. Both the FDA and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationship that you may have with the sponsor, its product, and if known, its direct competitors. For example, this financial information may include the sponsor's payment of your travel, lodging, or other expenses in connection with your participation in the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. The FDA and this committee place great importance in the open public hearing process. The insights and comments provided can help the agency and this committee in their consideration of the issue before them. That said, in many instances and for many topics, there will be a variety of opinions. One of our goals today is for this open public hearing to be conducted in a fair and open way where every participant is listened to carefully and treated with dignity, courtesy, and respect. Therefore, please speak only when recognized by the chairperson. Thank you for your cooperation. We have 8 speakers, so will speaker number 1 please begin by stating your name and any organization you are representing for the record?
Yes, hi. Thank you for giving me time to speak. My name is Susan Johnson, and I have no financial connections to any drug company, and no one is paying me for my testimony today.
This is Shi Jiao, the DFO. Ms. Johnson, we cannot hear you. Can you try it again, please? Thank you.
Oh, yes, I'm sorry. Thank you for giving me the time to speak. My name is Susan Johnson, and I have no financial connections to any drug company, and no one is paying me for my testimony today.
Mrs. Johnson, do you have any statements? We can see your PowerPoint slide, but we don't hear you.
Yes, can you hear me now?
Yes, we can hear you now.
Oh, okay. Are you ready for me to speak?
Please proceed.
Oh, sure. Thank you. And you heard the part, I have no financial connections to any drug company.
We did indeed.
You did. Thank you. Okay. My husband was a poziotinib patient for a little over 13 months, and he did so well on this drug with an initial reduction of over 90%, so much so that he was able to return to work full-time at a job that he loved. He was working in a classroom with 5th grade students. And this drug allowed him to do the things and to visit the places that he loved the most. He was able to travel to places like Hawaii, Alaska, and hike in these beautiful places. Even close to home, there was a great hike that upon reaching the peak, you could actually view the ocean. He was determined to make it to the top soon after starting poziotinib, and he did just that. And he was able to go out and have his lunch with friends and family daily. That was one of his goals once he was able to retire. And he was able to live life as normal as you can get with a stage 4 terminal cancer diagnosis. And were there side effects? Sure. They were very minimal for him, thankfully. And as long as you work with your clinicians, they'll work with you, and they'll help you to get through them. So definitely these side effects can be managed. Poziotinib offers a chance at a normal lifestyle, probably similar to pre-diagnosis, and that's how we felt. And for 2 years, we traveled out of state, getting on an airplane each month, and it was nearly, I think, 23 months to receive this drug. It was that important to us. And thankfully, it later became more easily available. And as we talked to many cancer patients and many folks that had the opportunity to try poziotinib, and we let them know that poziotinib is a targeted drug that will focus on a particular mutation. And as an angel buddy and a volunteer patient navigator for exon 20 and HER2 patients for the last 5 years, I've had the opportunity to share our amazing poziotinib experience and let them know that poziotinib will focus on their particular mutations. And another thing was my husband, who started out teaching students for 38 years. He ended up a huge teacher of adults and advocating for and praising poziotinib for letting him live an amazing life. And another thing that I thought was amazing was that my husband weighed 180 pounds, and he was on the full dose the entire time. And at the same time, a very slight woman weighing much less, she too was on the full dose, and they were both receiving great results. So this drug was truly our miracle drug. And it was a blessing to our family, and I will forever fight for poziotinib and for those patients that can benefit from it. So for us, poziotinib has been a lifesaver. It gave us a quality of life for my husband and allowed him to do many things, and it allowed him to see 3 grandchildren be born. So as I mentioned, we will forever be grateful to poziotinib. Thank you.
Thank you, Mrs. Johnson.
Absolutely.
We'll now move with the speaker number 2. Speaker number 2, please begin by stating your name and any organization you are representing for the record.
Good morning. My name is James Filipiak, and I appreciate the opportunity to speak with you today about my experience with poziotinib. I would like to state for the record that I have no financial interest whatsoever with Spectrum Pharmaceuticals or any other, and am not being compensated for my time today. I feel that I have a unique perspective as a caregiver for many years of my wife, Bobby, who had exon 20 mutated lung cancer and was enrolled in the poziotinib trial for 18 months. Additionally, I've been the co-sponsor of the leading social media site for poziotinib patients at the HER2 exon 20 group in the Bobby Johnson Flight Club, which has helped navigate HER2 exon 20 patients from around the world for the last 5 years with side effects, etc. Having been at my wife's side 24/7 throughout her poziotinib treatment, I can conclude that not only are the side effects manageable, but a high quality of life can and was maintained throughout her 18 months on the drug. In concert with our local oncologist, local pharmacists, and dermatologists, we found that reasonably simple solutions work to counteract the side effects. For example, diarrhea was mostly remedied by over-the-counter medicines. Paronychia and skin rash was helped with tea tree oil, Aquaphor and Saran wrap for a short period, and other moisturizers. Magic mouthwash with lidocaine for mouth irritations worked well, and the occasional few days break from the drug let things calm down when necessary, and it seemed to work quite well. We've since shared these remedies with other patients with similar success. Additionally, some patients found a short course of steroids helped as well, although we did not require that. Lastly, a dose reduction from the initial 16 milligrams helped greatly and did not alter the effectiveness of the drug for us. My wife and others, after some adaptations, were able to work and have an excellent quality of life. What I'd like to leave you with mostly today is, over the past 5 years, I personally know of no HER2 exon 20 patient who left the poziotinib trial because of the side effects. And I feel that with these guidelines, it's manageable, and so many have extended their lives, some up to 2 years and possibly beyond. Sadly, after a 7-year flight, my wife did pass away, but I cannot express enough how grateful our young daughter, my wife, and I were to have that time that poziotinib gave us. We were proud to be a part of the trial and the solutions that have helped so many. Thank you.
Thank you. Will speaker number 3 begin by stating your name and any organization you're representing for the record?
Thank you. My name is Maria Urbano. I live in Tampa, Florida. And first of all, I wanted to thank you for allowing me to speak today and share of my personal perspective as a sister who has lost her brother to HER2 exon 20 non-small cell lung cancer. I apologize for my accent since I was born in Colombia, so I ask for a little bit of patience. Before I begin, I would like to let you know that I have no financial relationship to any drug company or its competitors. I'm here only to share my family experience, hoping that it can save at least one cancer patient. Today is really hard to speak to me. Today, 2 months ago exactly, my brother passed away. He was diagnosed with stage 4 cancer back in March. At the beginning, he had very few symptoms, actually only one, pain in his back. So it was a really complete surprise for everybody. We were shocked to know that he had metastases everywhere. Brain, bones, lungs, of course. One case, my brother, was a very difficult one. His disease moved through his body extremely fast. It was made worse by the fact that he lived in Colombia, where he only had access to one treatment, and that was chemotherapy. And chemotherapy failed him. He had a long hospitalization with a lot of complications and passed away really quick, just months after diagnosis. It's impossible to know if this medication, poziotinib, would have helped him. But here is what I do know. All he had access in Colombia was a therapy, a chemotherapy. That therapy that was not targeted to his specific genetic HER2 exon 20 mutation. Chemotherapy was the best and only thing he had access to, and that one therapy didn't help him. Had one lived, I had hopes to take him to the Moffitt Cancer Center here in Tampa to get access to poziotinib as a trial. We like the idea of an oral therapy, a target therapy, a therapy for only HER2 exon 20, a target therapy that can be used as a first line of treatment, especially for patients in stage 4. Very sure that have made a difference. I appreciate the chance to speak here today about the needs of HER2 patients like my brother, hoping that it can save one life and that can help somebody else in the future. Thank you.
Thank you. We'll move on. Now speaker number 4. Please begin by stating your name and any organization you're representing for the record.
Hi, my name is Bill Brand, and I live in Redondo Beach, California. I have no financial interest in your decision today of any kind. I was diagnosed with HER2 exon 20 stage 4 lung cancer 3.5 years ago. It was discovered when, out of the blue, I suffered a seizure during a flight on my way to go surfing in Mexico. I had felt fine up to that point, so it was quite shocking to wake up from what I thought was a short nap, only to have a nurse sitting next to me with an oxygen tank and a mask telling me I had suffered a seizure. Since then, I have survived because of numerous radiation treatments and infusion of various chemicals. My best success was with Enhertu, which gave me 18 months of a normal life. Enhertu is not FDA approved for my condition, but it is for breast cancer. There are very few treatments for HER2. All the various drugs I've been on are crapshoots. One never knows if they're going to work on you, how long if they do, or will they produce side effects that make it impossible to continue. Enhertu, for me, proved to be a huge success. But for some HER2 patients, it only works for a short time or not at all. One never knows if something is going to work until they try it, so options like poziotinib are all we have. There is no cure for us, so we jump from treatment to treatment, knowing if it does work, it will likely stop working and another one will be required. Once we are out of options, well, that is the end for us, which it is for about 200,000 people per year in the U.S. My next drug will likely be poziotinib. Please do whatever you can to make this readily available. While it has side effects, it is one of the most successful drugs treating HER2 exon 20, of which we have very few options. Please, 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 on behalf of patients like myself, looking for the next treatment for a rare condition, and those who have passed, who benefited from poziotinib, which you heard about just now, but who are not here today to testify about the extra months or even years of life this drug gave them. Please advocate for FDA approval. Thank you to James, Susan, and Mary Lee for calling in. And anyone who wants to contact me can email me at the number 1, billbrand@gmail.com. That's the number 1, BILLBRAND@gmail.com. Thank you.
Thank you. Will speaker number 5 please begin by stating your name and any organization you are representing for the record?
Hello, my name is Michael Bockhart. By way of disclosure, I work in cancer drug discovery, but would like to make clear I have no financial relationship to Spectrum Pharmaceuticals. I'm here today speaking on behalf of my mom, who was diagnosed in November 2018 with non-small cell lung cancer HER2 exon 20 insertion. Her first line of therapy was carboplatin pemetrexed with pembrolizumab. Overall, this treatment allowed for good quality of life. However, the chemotherapy dropped 9 months later due to increasing anemia and weakness. Then, a couple of months later, she started second-line therapy consisting of docetaxel combined with ramucirumab. This treatment was not well tolerated and resulted in hospitalization after the first cycle. During this time, she could not live a normal life. Further, the treatment did not control her brain metastasis. Luckily, we were already in the process of applying for the early access program of poziotinib as her third-line therapy, which she started 2 months later. She remained on poziotinib for the next 18 months. Her treatment with poziotinib did require dose reduction from 16 milligram to 12 milligram daily. In the last 6 months, my mom had to reduce the dose further to 8 milligram daily and occasionally 10 milligram daily. However, during this period of time, my mom had a good quality of life. Yes, there were some side effects like broken skin and diarrhea, but in contrast to the previous lines of therapy, she was not bound to clinic-based infusions and could live relatively normal life. Further, the duration of stable disease for her was much longer than on any other of her previous lines of therapy. I strongly believe that the access to poziotinib allowed her to live long enough to gain access to her current fourth line of therapy, which is trastuzumab combined with durvalumab, which is currently on 13 months total so far. I hope with accelerated approval of poziotinib that other HER2 exon 20 non-small cell lung cancer patients get the same opportunity for a longer life. Thank you for considering my statement.
Thank you. Will speaker number 6 please begin by stating your name and any organization you're representing for the record?
I have a slideshow with my presentation, if you could please start that now. Good morning, my name is Kristen Lennes, and I'd first like to make it clear I have no conflict or financial interest in today's proceedings. All of those featured in the slideshow you see before you today have given their expressed consent to allow the use of their images as well. Next slide. My mom, Stella Martinson, has been fighting non-small cell lung cancer since 2011, and we were devastated when she received a stage 4 diagnosis in 2018. At that time, we learned of her rare exon 20 mutation and also that there were no approved drugs available. Next slide. Consequently, when we learned she qualified for the poziotinib trial, we were incredibly grateful. We did learn how to effectively manage the side effects she experienced working with her clinical team. After nearly a year on this regimen, poziotinib reduced her cancer significantly and was a critical bridge to future treatments. Next slide. As a volunteer patient advocate with the exon 20 group, I also have firsthand experience with hundreds of exon 20 patients being treated with poziotinib and other drugs, both at major research hospitals and local community cancer centers across the country. I've witnessed the challenges and the triumphs from many participating in this trial and walk with them through the day-to-day experience of what it's like to be on this regimen. Next slide. I've learned through that shared experience how this diverse range of healthcare teams are handling side effect management and patient care. This has given me a unique opportunity to not only see the progression with dosing strategies, but also how patients have been able to effectively manage the side effects of this drug with the help of their clinicians. Next slide. As with many cancer drugs, the toxicities can be challenging, but they are manageable. Overall, with the patients I've worked with, the side effects were similar to other TKIs in this class, and many patients have benefited greatly from this drug. Next slide. Taken since she participated in the poziotinib trial, the photos you see before you today are all gifts of time that poziotinib has given our family. Poziotinib has given my mother the ability to see her grandchildren graduate, spend countless birthdays with loved ones, celebrate holidays together, and create priceless memories we will treasure always. In short, poziotinib gave my mother an opportunity to make memories instead of just becoming one. Because of poziotinib, Mom also has a fighting chance to meet her very first great-grandchild, Charlotte, who's due in early December. Next slide. In conclusion, based on my unique experience with my mom and many other exon 20 patients, I've seen that poziotinib is a safe, effective, and manageable drug. The meaningful benefits absolutely outweigh the manageable risks. I respectfully ask for your consideration to approve this critical drug so that other families like mine will have a chance at a better future. Thank you for your time.
Thank you. Will speaker number 7 please begin by stating your name and any organization you're representing for the record?
Thank you for the opportunity to speak today. My name is Mary Modica. My only relationship to this meeting is that you were talking about a drug that I take as I have no affiliation with any company or organization. After a lung x-ray, my primary care doctor sent me for a consultation with a thoracic surgeon. This was July 2019. I was told I had small cell non-smokers HER2 exon 20 lung cancer that was inoperable and incurable. Needless to say, I was devastated and dumbfounded. From this appointment, I met with an oncologist to discuss treatment. I began with radiation and infusion, which is short of a nightmare for me. From there, I had chemotherapy and chemo maintenance. I also had a drain in my lung. Needing something more, I began to search for a trial program for something more than chemo. My biggest concern at that time was that I would not qualify for the poziotinib program. However, more than 2.5 years later, I am still on the drug and doing well. Without it, I don't think I would be here today. In the beginning, I was always afraid I would be told I needed to come off the drug. The side effects are varied, last different amounts of times, and sometimes difficult. They can be a running nose, a bloody nose, eyelashes that stick together, burning and cuts in my hands, loss of hair, and others. The two that have persisted are diarrhea and dry skin. I have learned how to manage to live with these side effects and am determined to continue with the treatment as long as possible. My days are busy and productive. I get up and out and amaze my family and friends with my positive attitude. I am president of an organization with over 380 members. This weekend, I spent a day working at a tent at a street fair and attended a fundraiser. Yesterday, I volunteered at a charity golf outing. This is in addition to a number of doctor appointments and meetings. I am speaking from my heart that I need this drug to continue living. I truly hope the FDA will approve it so that I and others can lead a full and productive life. Again, thank you for this opportunity.
Thank you. Will speaker number 8 please begin by stating your name and any organization you're representing for the record?
So hi, good afternoon. I'm Joshua Sabari, thoracic medical oncologist at NYU Langone Health in New York. My clinical and research focus is on developing targeted therapies for patients with solid tumors, and I've been involved in the Zenith 20 study since 2018 as a co-investigator and have treated around 20 patients with poziotinib, including patients with HER2, exon 20 insertion mutant non-small cell lung cancer. It's important to note that I have no personal financial relationship to Spectrum. However, my institution, NYU Langone Health, receives funding for participation in the clinical trial. I'm speaking on behalf of myself and not my institution. So in my clinical experience, the first-line treatment options for patients with HER2 exon 20 insertion mutations remain platinum doublet chemotherapy, with response rates of about 30% and median progression-free survival of about 6 months. With regards to the addition of immunotherapy, response rates and durability of response to immunotherapy has largely been ineffective in this patient population, and of concern to me and to patients is the potential immune-related adverse events, including transaminitis, colitis, and pneumonitis, particularly in patients who go on to receive HER2-directed therapies post-immunotherapy. As you all heard, trastuzumab deruxtecan recently received an accelerated approval, and we saw an updated data set at ESMO at the 5.4 milligrams per kilogram dose, which excluded patients with any pulmonary risk factor, and the rate of ILD, or interstitial lung disease pneumonitis, was about 12%. This is now a standard of care in my practice in the second-line setting. However, ongoing confirmatory phase 3 studies are underway. There are patients who are not eligible for trastuzumab deruxtecan given cardiac or pulmonary comorbidities, particularly prior immune-mediated pneumonitis, as well as patients who may have poor marrow function, right? So trastuzumab deruxtecan, the payload, is a topoisomerase 1 inhibitor, a potent and conventional chemotherapy. For those who are eligible, there is still a significant unmet need for patients who progress on trastuzumab deruxtecan, and the current standard of care includes docetaxel, which we talked about has a response rate of sub 10%, and docetaxel and ramucirumab about 23%. I do not use single-agent checkpoint inhibitors in this population until all HER2 targeted therapies have been exhausted. Poziotinib has toxicity, including rash and diarrhea due to EGFR and HER2 wild-type inhibition, but these toxicities are predictable and have been manageable in my clinical practice with routine follow-up. Diarrhea can be well controlled with the use of loperamide, 4 milligrams twice a day, and rash has been manageable with topical emollients, topical steroids, oral antibiotics, and close dermatologic follow-up. Dose reduction and dose holds have been common, and I've treated multiple patients with ongoing radiographic response and clinical benefit past 1 year, including patients with durable CNS or central nervous system control with patients with known brain metastases. In those patients who do not tolerate treatment, the drug is typically discontinued early, hence the lower duration of response seen on the study, and the side effects, including rash, diarrhea, in my experience, are immediately reversible without long-term harm to our patients. There are other FDA-approved agents in the non-small cell lung cancer space, such as second-generation EGFR tyrosine kinase inhibitors like afatinib, which are commonly dose-reduced in clinical practice and continue to maintain efficacy. I agree, further confirmatory dosing studies are needed. However, our patients need treatment options today and are not able to wait for 2 to 3 years for this data to be generated. Thank you for your time.
Thank you. The committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments. We will proceed with the questions to the committee and panel discussions. I would like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. So question number 1 is for the committee to discuss, relates to NDA 215643, poziotinib, and the applicant is Spectrum Pharmaceuticals Inc. And the question is, discuss the overall risk-benefit ratio of poziotinib 16 milligram once daily, given its limited response rate with poor durability, high rate of toxicity, inadequate dosing optimization, and delayed confirmatory trial. If there are no questions or comments concerning the wording of the question, we will now open the question to discussion. So let me just start with our voting committee members. it is clear that we had a pretty active and robust discussion during the clarifying questions to the FDA and also the applicant. And I'd like to actually prioritize our thoughts, as a group in 4 sections. One really is for us as a group to review and really discuss the efficacy and whether or not we as a committee feel that the efficacy that we saw today and was presented today by both groups is better or no better in contrast with existing agents. And it is clear that for us, clinicians, we have to consider the HER2 antibody in that context as an existing clinical agent for us. The second topic that I want to review is obviously talking about safety, the concerns that exist with regards to dose reduction, drug interruptions, and lastly, obviously, one of the biggest challenges that we clearly express as a group, which is the lack of drug optimization and the challenges that we see, some of us see, moving from phase 1 to phase 2 and changing drastically the dose to the confirmatory phase 3 trial. And also, to finalize our discussion, I want to actually ask as a group to address how do we feel and think about the timing of the enrollment of that confirmatory trial that may actually take several years based upon what Dr. Pazdur has discussed and based upon what the sponsor has presented today. Please raise your hand. Right. So Mr. Mitchell, we start with you.
Yes, Doctor. Thank you very much. First, I would like to say that I'm a little bit confused, and I would like to direct this question kind of comment to the FDA to help me. The FDA appears to want us to look at poziotinib in light of real-world alternatives that are available even if they are not yet granted full approval, which is apparently the standard for comparator treatments in order for the FDA to grant accelerated approval. Yet in sponsor slide 73, which the patient representative raised, the FDA wants us not to consider the real-world potential clinical use of poziotinib in sequence because it reflects a different indication for use than that sought by the sponsor for the accelerated approval. It seems like the FDA wants it both ways, and I'm wondering if someone from the FDA can address that contradiction first, and then I have one other question.
Okay, this is Dr. Harpreet Singh from the FDA. I will try to address it. I addressed earlier. We believe that it is our regulatory purview at the FDA as regulators to deal with the regulatory confines of what is defined technically from a regulatory standpoint as available therapy. In the same vein, and so taking that separately, we are asking the committee, who is comprised of clinicians in clinic, as many of us are, but today we're wearing our regulator hats. We're asking you to put on your clinical hats, which is why we seek your advice, and think about what is available in the clinic to patients. There are layers of uncertainty, to use the term that Dr. Dresner used, whether or not HER2 is considered an available therapy from a regulatory standpoint is just a finer point. Really, if you want to invoke this, you can invoke it in two ways. First, yes, we are asserting that poziotinib does not represent an advantage over even the regular approved drugs, so the drugs under traditional approval. However, we are also saying that we acknowledge and recognize, as everybody has, and you, the committee, have told us on several occasions that you are not considering what we consider
as available therapy from a regulatory standpoint. You consider what you are confronted with in clinic, and therefore, yes, ENHERTU is available to you. And yes, it likely will have a great impact on whether or not this confirmatory trial can be enrolled. So I hope that it's not confusing. I see how it can be. We're not asking you to look, to kind of play both sides of the coin here. We're asking that you let us, kind of make the regulatory decisions. We're asking you, in the face of this available therapy with a 16% response rate that is clinically available, how does that play in to all these layers of uncertainty? And for this point, the uncertainty of whether or not the confirmatory trial can be reliably conducted. Dr. Pazder or Dr. Beaver, did you want to accentuate any of those points? Okay.
Well, there's too much confusion about it. Let's go ahead. Dr. Beaver.
Go ahead, Rick.
I think to make this simpler, let's take the monoclonal antibody off the table here, okay? But give us your opinion both ways, basically, with it and without it, so to speak. But from making the regulatory decision, we would not consider this available therapy. So if that simplifies your discussion of this, consider it that way, because that's the way we are going to make it. The other point I want to make very clearly in the voting question, we are not asking you whether this drug should be approved or not. We are asking you for your consideration of the risk-benefit, and there's many things that come into play here, so to speak. So if it makes everybody's life simpler here, we will, when we make this regulatory decision, be looking at what is available, and the availability of the ENHERTU drug is not going to be considered here in making that issue of comparing it to ENHERTU. But here again, there are many other issues here. Is that helpful to you, Mr. Mitchell?
Yes, Dr. Pazder, let me just take it one step further. So we want to look at the overall risk-benefit based on what we know today. I understand the difficulty in enrolling the confirmatory trial given this other treatment, but it may take longer. On the other hand, if I'm looking at slide 73, maybe people would get the other drug first and then enroll in the confirmatory trial. And by the way, I'm a big believer in timely, quick, not delayed confirmatory trials, and the fact that not one patient is enrolled is a problem. But if we're looking at the overall risk-benefit based on what we know today, that's all we have. We don't have a confirmatory trial. So what it feels like to me is I need to look at when I do whatever I will do in this discussion, I have to look at, based on what we know today, the delayed confirmatory trial is a problem, but it isn't going to help with overall risk-benefit for me.
Okay. Thank you both.
Hi, this is Julia Beaver. Could I just add very quickly another related point is that we are asking the committee for a risk-benefit discussion for the studied indication with the data that we have, not as a potential sequencing agent, not as an agent to have available. We're asking you really to discuss the studied indication and the data that we have today.
Only the studied indication.
Thank you.
But only the studied indication. Period.
Correct, Mr. Mitchell. Dr. Krause?
Yes, hi, Albert Krause, industry representative. I think that those clarifications discussion that Dr. Mitchell drove are very helpful. I'd like to add an additional point, and this often is a point of industry concern broadly. It's around assumptions within a question. There are a number of adjectives here that I think ODAC members should judge a bit for themselves, because there's alternate interpretation from FDA versus the industry presentation you saw. the words like poor durability as opposed to what the durability is. It's a relative concept. High rate of toxicity, again, a relative concept. Inadequate dose optimization, it's a relative concept. And delayed confirmatory trial. I'll leave confirmatory trial off the table because I think Dr. Mitchell emphasized it's a problem, but it's a little separate than today's benefit risk. But it is an issue. But I would say for ODAC discussion, I think it's important that ODAC members themselves assess their sense of that data when they're coming to risk-benefit rather than the kind of a bit directional leading of the question for discussion. But I think the discussion topics are excellent. Thank you.
Thank you, Dr. Krause. Dr. Madan?
Yeah, Robbie Madden, NCI. So I think one point that's worth kind of highlighting here is a little bit, in my opinion, the disconnect between the response rate and the durability of response. Generally, it looks like poziotinib maybe has a similar response to other agents, but a much shorter clinical benefit or duration of response period. And often we see that in the clinic because of toxicity, because the patients can't tolerate the drug after initial clinical benefit, and it either has to be reduced to a dose where it loses efficacy or isn't as effective. So I think that's an interesting point to make when you're looking at this question, and I think that factors into my opinion quite a bit.
Thank you, Dr. Madan. I just probably have a question, a comment also related to that. I think that the notion that an applicant or a company develops an agent and leaves the agent, if indeed the agent gets to the market, and it leaves the management of that agent to the discretion of a clinician. Although I do understand the ability that I have as a clinical person to adjust up or down the dosing, I really think that today's, at least for me, the drug development of this agent clearly stresses the importance and the need for us, when we're thinking of drug development, of a major redo of how we develop agents from the traditional phase 1, phase 2, and phase 3. Because it is clear that when you look at the data and you look at actually optimization of therapy, drug discontinuation, time to dose reduction within 29 days, the time that it takes patients to get back on therapy at a lower dose of around 9 days or so, or 8.5 days, the half-life of the agent, it is pretty predictable that that data would imply that a significant proportion of our patients getting this agent, which I'm not disputing that this agent appears to have some efficacy. There's no doubt, in my opinion, based upon the data that I have reviewed, but my concerns are how the agent got developed, how the dose got picked, and more important than that, it is predictable based upon the data that we saw that a significant proportion of those patients do not get adequate dose, meaning that they get therapy, but maybe they're not getting the appropriate dose for their disease. So it's something that I think we all have to also think as we think to voting today. Dr. Halmos, do you have a question or a comment?
Hi, Balash Thomas. I have more of a comment than a question just around the discussion. I mean, clearly, if you think about how to sort of position poziotinib here, there's a number of concerns in terms of limited efficacy, limited durability, high toxicity, certainly a very, very complex development process and somewhat poorly defined dosing. But it's hard not to see that it's clearly beneficial, for some patients, and in fact for some patients long term. It clearly seems to be desired by patients, and I want to highlight the comments from a major patient advocacy group, the Exon 20 group, that represents truly hundreds of patients with this disease. Also, it has a very well anticipated and well-known AE profile. So indeed, we've had 20 years of experience with the class of agents as to how to optimize dosing for the individual, not for the cohort. And in terms of efficacy, the profile is not too dissimilar from recently approved drugs as highlighted with reasonable dose adjustment options. So if I put my clinical hat on and I don't have another one in a way, yes, indeed, I think I would feel comfortable offering this to a patient if approved, and I think some or many of our patients could potentially benefit from it. So I do feel that the overall risk benefit profile based on that is acceptable.
Thank you. Dr. Liu?
Yeah, I'm going to tag along. This is Chris Liu from University of Colorado. I'm going to tag along to Dr. Halmos's point. And a question to the FDA, because the way this question is framed in terms of the vote, I need to know if the FDA wants us to consider this in a complete vacuum. And so when we think about the current benefits and the risks for a patient population that really has limited treatment options, I agree with Dr. Halmos in terms of, there aren't a ton of options here, and there is a response rate, even if the durability of response is quite short. And in regards to toxicity and risks, I think as oncologists, we have a pretty good experience modifying doses of TKIs, and certainly have a lot of experience with this as well. But my issue with the question is really, how much can we look at this question in a complete vacuum in the setting of, other agents that are available? And I think that actually influences the vote. If we just take it at face value, I think to me, the answer is fairly clear. But if we look at the bigger picture, which is what I would like to do, then I think that influences the vote significantly. And so I guess that is a real question to the FDA of, how much of a straight, in a vacuum, do you want this answer, answered by the committee? Because I think it's hard to ignore the landscape, the current treatment landscape.
Okay, this is Dr. Singh, Harpreet Singh from FDA. We do not live in a vacuum. We live in the real world. And when we bring things to ODAC, to you, our esteemed committee, we do ask you to consider these questions we're asking you in the context of the world you live in. And in that world you live in, you have the available FDA-approved therapy, in your tool bag, and you also have the therapies under accelerated approval. I know this is a point of confusion for the committee, but yes, you must consider what is out there in the real world, particularly when you're talking about the uncertainty and being able to confirm or refute benefit. It's a total package here. We're not asking you if poziotinib is efficacious and if you can manage the toxicities. We know it's a TKI, but I think we've shown you it is not comparable to other therapies. In fact, Dr. Malino accurately stated, and this is based on data, if approved, this would be the lowest, least efficacious, most toxic TKI on the market. That all the numbers stand up to that. That is a numerical, factual piece of information for you. Yes, you must consider the entire landscape, given all the several questions that we're asking. I hope that answers your question, but I'll open it up to my team for a few moments if they wanted to add. Okay, I'm hearing nothing from my team, so we'll thank you for the question.
Yep, I appreciate it, and I have no further questions or comments.
Thanks, Dr. Liu and Dr. Singh. Dr. Roscoe?
Hi there, Ashley Roscoe. So, one of the concerns I have about the dosing schedule is that it didn't seem like it served the majority of patients. And one of the misses from the applicant, they provided health-related quality of life data for patients that was largely incomplete. So really to get a better sense of what the patient experience was. One thing that was that, I think oncologists are very familiar with managing rash and diarrhea. And an oral option is appealing for patients, whether that increases access or not. I do think patients at times would prefer that. But what wasn't clear to me is this something where there's an oral agent that patients have to come in weekly for fluid resuscitation and patients are coming in with these grade 3 toxicities? That wasn't abundantly clear because if you're going to offer something and have kind of typical TKI management, or alternatively, you're bringing patients in, much more regularly and impairing their health-related quality of life, that wasn't clear to me, and I'm not sure if the applicant has that data regarding increase the frequency to the clinic, or how, what the management was beyond kind of, typical management for diarrhea related to TKIs. So that's just something that was weighing in my mind as we're discussing this. I also, do want to kind of land on that point that they did enroll patients with CNS mets, which I think is also an important part of the discussion to make sure that, an agent is available to be able to address that as well. So more comments than anything else, those are things that were weighing on my decision.
Thank you, Dr. Roscoe. I think that when you think of that as well, and for us as a committee members, there is no doubt that patient convenience is critical when we're thinking of patient care, right? And oral agents may actually have the perception of easy administration, as you described in Dr. Roscoe, compared to IV agents. But at the end of the day, I think most of us, from the patient perspective and or from the physician perspective, will likely pick an agent that has the most activity and the least side effect profile over the route of administration. And I think that's something that I think is important to stress as well. Dr. Madan, you have an additional comment?
Yeah, I just want to follow up what you said and what Dr. Roscoe said. It's a good point. The benefits of an oral agent are you're not in the doctor's office, but if the toxicities require dose reduction and within a month, you're back in the doctor's office pretty quick. But so that I think is a great point. The only other thing I'll say is, from a committee standpoint, this whole should we consider the other option as an accelerated approval as an acceptable alternative or not? From my perspective, it's hard for me to say, well, the other agent has accelerated approval, so I can't guarantee my patient will get that. But if this gets accelerated approval, it'll be good to have that option. So I think they're both kind of in the same category, best case scenario. So, from just that keeps coming up amongst the committee, I think that, they'll be equivalently available. I don't think we would say one would be more available than the other in the circumstance that this agent got accelerated approval as well. At least that's my perspective. I don't know if that helps the committee or not.
Thank you, Rob. Dr. Thomas?
Hi, my name is Thomas NCI. So this comment is in line with an earlier comment that Dr. Halmos brought up. So there's a lot of discussion in terms of dose optimization. But I would have liked to see maybe, I know it's a difficult to enroll population, very rare and so forth, but are there any patient variables that predict benefit? I mean, this is a unique population, not too many of those patients. But from this drug's experience in the EGFR setting, it seems that the sensitivity to poziotinib is highly dependent on where the mutation is located. There are some mutations where the drug is highly active, so up to 45%, versus no activity, even the same, HER2 exon, EGFR exon 20 insertion, a different mutation results in very low activity. So again, you're sort of dissecting an already small population, but that might really influence the risk benefit. And I know that earlier there was a question raised. I didn't hear any directed responses to that question, but again, not relevant to this discussion, but I think it would be really helpful if early in the drug development process we focus on, in addition, or probably more so than dose optimization, what are the patient variables that might influence the efficacy?
And I'm just perhaps curious, thank you, Dr. Thomas, I'm just perhaps curious to hear other committee members' comments on how do we all feel about the potential delay of that phase 3 trial, meaning having an agent out there for several years without having a real clear understanding as to what is the optimal dose for our patients, and maybe actually, as we reviewed earlier, the potential of having also an antibody drug conjugate that may actually also pan out to be effective for the same patient population. Obviously recognizing that, the confirmatory trial that the applicant has put forward may allow patients to get the TKI after HER2 therapy. Any thoughts or any comments on that? Dr. Thomas? But Dr. Sela, do you have any comments?
Yes, I was going to say, I do think that that's a concern. I mean, ideally, we would have already had patients well enrolled in the confirmatory study, and I know we've been having a lot of discussion about the side effect profile of the medication and the overall response rate and duration of response. We're potentially exposing patients to potential risk and toxicity for a long period of time before we have any of these confirmatory trials. And being that the confirmatory trial is going to be at a different dose than what has been studied in the majority of patients previously, I think there's just a lot of questions about what this means for our patients, and we probably won't have those answers for quite some time.
Thank you. Dr. Harrington, do you have a comment or a question?
I do, thank you. And it is about the confirmatory trial. So, the ODAC has been asked to make a decision at this point in time about the accelerated approval, and so we can only make a prediction about the rate at which the confirmatory trial will enroll. But if we lift that barrier for a second, that we have this point in time, it should be known relatively soon how well that confirmatory trial will enroll. And in fact, if it goes reasonably well, it would be one route of access for the agent to patients who have this particular mutation. So I think I would urge the sponsor and the FDA, as I said before, to focus on that confirmatory trial, to get that done as quickly as possible, and the FDA and the sponsor will soon learn whether that is a feasible trial or not. But I don't think it'll take 6 years to find that out. Thanks, that's all I have on that.
Thank you. Dr. Kearns?
Hi, everyone. I just wanted to also comment on the question that was just posed around the timing of the confirmatory trial. I also agree and am concerned about not knowing the appropriate dosage and about delays in enrolling patients. I agree that it may perhaps enroll more quickly than the 6-year time frame, but I think given that we don't have that information or have a trial that's currently enrolling, that certainly weighs into my decision, and I have some concerns about that.
Thank you. Dr. Wallman?
Yeah, I'm going to sort of pile on here a little bit. I think the importance of the confirmatory trial is amplified by the FDA statements that this agent, if it gets approved, is going to be one of the most toxic of the TKIs for the indication that gets approved. I think that really sort of showcases how important the confirmatory trial is. And that confirmatory trial is going to be at least 2 years out from, if I understand correctly, from the first evaluation. It'll take 2 years to get to a first evaluation from the start of enrollment. That's a ways off to put patients at risk. The other thing that I'll come back to that we heard just a minute ago was, and it's not clear what the right dose is, and they're using a dose in this confirmatory trial that is not the dose that's being asked to be approved, and that's problematic. It communicates a lack of confidence in the dose that's up for approval. That's a problem. I'll finish here.
Thank you, Dr. Wallman. Are there any other comments? Any member? Dr. Krause?
Yes, thank you, Albert Krause, industry representative. Just a commentary as someone who has, some 30-plus years experience in drug discovery and development. Dose selection, dose optimization is a very tricky business and very difficult. And it's true that we can do better and we should do better, particularly in oncology. where a lot of the doses in many drugs I've worked through discovery, development, and approval had subsequent dose adjustments for a variety of reasons, being misled by pharmacodynamic markers because, you know, you did a bunch of work, but then you found more things out later, et cetera, et cetera. And so certainly we need to do robust dose schedule finding, and I think obviously FDA is emphasizing that, and I think it's the right thing. That said, I just want to give a commentary that in my estimation, you can always declare dose schedule work as being inadequate in a sense because it's a totality argument with all sorts of data pieces. You never have the perfect clinical data. You know, often a dose is focused on maybe more than schedule. So it is a tricky business and difficult business. And determination of adequacy is tough. It's certainly true that if there's a lot of toxicity, you know, it becomes even more imperative. And it's also true that many drugs are well used, delivering great patient benefits with very significant dose treatment holiday and dose reduction schema. So I just wanted to kind of, from a drug development perspective, lay that out, which many of you know, but some may not. But given the point of inadequate dose optimization on the table, and emphasize, remember, this is a rare orphan disease. It's not just orphan, but you know, there aren't a lot of patients to do a lot of work on. You know, you can always look back and say we could have done different things. The old hindsight's 20/20. But that's you know, this company has done a fair bit of work. Let's just say, you know. So, anyways, I'll stop there. Thank you.
Thank you, Dr. Krause. Mr. Mitchell, do you have a comment?
Yes, Doctor. First, I want to clarify, I have to do this periodically during these meetings. I'm not a doctor. I'm the consumer rep, and I'm a multiple myeloma patient. I want to go back to this issue of whether oral is superior to infused for a minute and just state that I think that's really a subjective kind of issue for the patient. It turns out that of the 4 drugs I'm on, the 2 oral drugs take a much greater toll on me right now than the infused drugs, which I tolerate just fine. I think that someone said earlier on, what a patient is looking for is the drug that works best with the least toxicities. And right now, that would be my infused drugs. The oral drugs are tough. Second, if you believe in financial toxicity in cancer treatments, oral drugs can frequently be more burdensome, as you all know, because I can buy a supplement policy that covers Part B completely, but I'm on Medicare. I can't buy a supplement to absorb the cost of my oral Part D drugs. So I'm just saying that when someone puts forward kind of blithely, isn't oral better than infused, I don't think that that's always the case. It's subjective and it's specific to given patients. I would offer that as a consumer and a patient.
Thank you, Mr. Mitchell, for that thoughtful comment. So perhaps if I can summarize some of our points that we have discussed and comment on. I think when you look at the activity, I think that most of us do feel that this agent does have efficacy, at least for some patients. And certainly based upon the treatment sequences that we have right now from front to second to third line therapy, then an agent like poziotinib would have, in fact, a role through the natural history of non-small cell lung cancer, specifically HER2 mutations. It doesn't appear that the committee is too worried about side effects. It seems that we all feel quite comfortable for many years now treating toxicities from AEs, and it does appear that the group feels comfortable with dose titration, if you allow me to express it like that. Perhaps because it's similar to many of the agents that we have used over the last 2 decades or so. We agree that efficacy and side effects probably would be far more important than the route of administration for the vast majority of our patients. We also talk about dose optimization, and it is clear and consistent that we all think that the dosing and the dose escalation schema of how this agent got developed is spotty at best, and we still don't know which is the ideal dose or the optimal dose with regards to efficacy and also side effects. Looking at the confirmatory trial, I think we all, at least most of us, agree that it is critical for the sustainability of an agent such as this. There are concerns about the timing of the trial, and certainly significant concerns as to how one go from 60 milligram QD dosing to a confirmatory trial with an absolute different dose of 8 milligrams BID without having clear and consistent data from the phase 1 and 2, suggesting perhaps, as someone described, a lack of confidence in the dose selection, and perhaps obviously implying the potential risk that patients may embark on if we have to wait 3 to 5 years for that trial. Are there any questions or any final thoughts before we move on to question number 2? If there is no further discussion on this question, we'll now begin next question. We will now move on to question number 2, which is a voting question. Dr. Shixia Chen will provide the instruction for the voting. I will read the question. Or perhaps, Dr. Chen, if you can just proceed with the instructions before.
Yes, thank you, Dr. Garcia. Question 2 is a voting question. Voting members will use the Adobe Connect platform to submit their votes for this meeting. After the chairperson has read the voting question into the record, and all questions and discussion regarding the wording of the vote question are complete, the chairperson will announce that voting will begin. If you are a voting member, you will be moved to a breakout room. A new display will appear where you can submit your vote. There will be no discussion in the breakout room. You should select the radio button that is the round circular button in the window. That corresponds to your vote. Yes, no, or abstain. You should now leave the no vote choice selected. Please note that you do not need to submit or send your vote. Again, you need only to select the radio button that corresponds to your vote. You will have the opportunity to change your vote until the vote is announced as closed. Once all voting members have selected their vote, I will announce that the vote is closed. Next, the vote results will be displayed on the screen. I will read the vote results from the screen into the record. Next, the chairperson will go down the roster, and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. Are there any questions about the voting process before we begin?
I will now read the question for us to vote, and the question reads, do the current benefits of poziotinib outweigh its risk for the treatment of patients with non-small cell lung cancer with HER2 exon 20 insertion mutations? Are there any questions about the wording of this question? If there are no questions or comments concerning the wording of the question, we will now begin the voting on question number 2.
We will now move voting members to the voting breakout room to vote only. There will be no discussion in the voting breakout room. Voting has closed and is now complete. Once the vote results display, I will read the vote results into the record. Voting has closed and is now complete. The vote results are displayed. I will read the vote totals into the record. 4 yeses, 9 noes, and 0 abstention. The chairperson will go down the list and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. Thank you.
Thank you, Dr. Chen. We will now go down the list and have everyone who voted state their name and vote into the record. You may also provide justification for your vote if you wish to. Will start with Dr. Thomas?
Hi, Anish Thomas, NCI. So there's definitely an unmet need for a subgroup that was probably first identified probably more than a decade ago now. The drug poziotinib seems to be active, and there are patients who definitely benefit from this agent. But for the overall population of HER2 exon 20 insertion patients, and in the specific setting where this accelerated approval is sought, there are several open questions. So it's hard to make a case, although as an oncologist I would love to see more options for these patients. The big advantage seems to be that it is an oral administration over available therapies, but it is somewhat offset by toxicities as well as uncertainties around the dose itself. While I recognize that this is a rare patient population and that the dose and schedule is not always straightforward, I feel like it needs to be looked at, and it has not been so far in its extended development course. And I think that would, benefit the patients in the long run. And I also feel like, given the low therapeutic index, the better predictors of response should be sought. Thank you.
Thank you. Dr. Sung?
Anthony Sung, Duke University. I voted no for many of the same reasons as Dr. Thomas just outlined. I mean, I think part of it comes down to me if I have a patient before me and I could give them trastuzumab or I could give them poziotinib, I would probably give them trastuzumab. There may be a role for poziotinib in patients who fail trastuzumab, but the data is not there yet. And there are a number of concerns around the drug that have already been highlighted, such as around the dosing and the confirmatory trial. And I think just currently, it's not there yet.
Thank you. Dr. Roscoe?
This is Ashley Roscoe, Ohio State. I found the patient and the caregiver statements incredibly persuasive. I believe that the dosing schedule may not serve many of the patients, but it does serve a population of the patient population. I understand that there are dose reductions for the patient population. The oncologists are familiar with TKI management of toxicities. That being said, I recognize that this patient population requires additional therapy, and there's much to be explored in this area.
Thank you. Dr. Halmos?
I'm Blas Ramos. I voted yes. I believe as much as poziotinib has a very narrow therapeutic index, it could fill a niche in the treatment continuum for patients with ERBB2 mutated non-small cell lung cancer, and with the expertise we've gained over the last 2 decades could be supported appropriately despite its adverse event profile.
Thank you. Dr. Liu?
This is Chris Lee, University of Colorado. I voted no. It took a look at the fact that accelerated approval requires meaningful benefit over existing therapies. And while I have no doubt that poziotinib has efficacy as well as toxicity that can be managed, I think the issue here is twofold. One is, is the agent potentially more efficacious than trastuzumab deruxtecan? We don't have evidence to suggest that. Two, could poziotinib be sequenced in a way to salvage patients that have progressed on trastuzumab deruxtecan? And we unfortunately don't know that answer. Of course, my hope is that the answer is yes, that the current phase 3 study will answer that question and increase options for this patient population that really needs more treatment options. That concludes my statement.
Thank you. Harrington.
Thank you. David Harrington, Dana-Farber Cancer Institute. I voted no. I agree with the reasons that others have stated for a no vote. I also think that statistically, there is just not sufficiently strong signal to justify giving this accelerated approval and exposing patients to this while the definitive phase 3 trial at a different dose will be in the field. So I think it would be premature to give this one accelerated approval.
Thank you. Mr. Mitchell?
I voted yes, and I'll start by saying that it was very clear that we're not supposed to vote on whether to give accelerated approval, but whether we deem the benefits to outweigh the risks, which I do. The drug clearly has efficacy for some patients who are in real need of additional options. The toxicities, I think, from all the clinicians we heard from are manageable. Dose reductions are so common based on what I think I know. That doesn't worry me. Overall, this drug belongs in the armamentarium of those clinicians who are trying to treat these patients that lack sufficient numbers of options. The challenges of the confirmatory trial are important, but it didn't overcome my overall sense that, yes, the benefits outweigh the risks.
Thank you, Mr. Mitchell. Mr. Pantelis?
I voted, I'm Jim Pantelis, patient representative. I voted yes. I think that the drug has meaningful benefits and that most of the risks are manageable, and it's a population much at need of options. Thank you.
Thank you. Jorge Garcia, I voted no. I think that I echoed sentiments from everybody else who voted no. I think that there is no doubt based upon the data that we have that this agent has or may have a role in the natural history of this disease. I just fundamentally believe that the way that this agent got developed and the dose finding status and the drug optimization, which is not ideal, and I'm just afraid that that we don't have the ideal dose for that ideal orphan patient population. So I voted against it, and certainly I believe the confirmatory trial needs to be probably maybe not redesigned, but certainly, it needs to be looked at better. We don't want to have an ex-US patient population that may complicate the interpretation of that data if, in fact, the bulk of the patients in the United States are going to be getting HER2-based therapies. Dr. Silla?
Hi, Catherine Schilla. I voted no. I think my reasons are similar to what others have already mentioned. I definitely feel that there is an unmet need for this patient population, and we have heard about the efficacy and the potential ways to improve tolerability. But I think for me, the main issues are I'm not sure that this represents a meaningful therapeutic benefit over other agents that we have, as we've heard about, and I have concerns about the dosage and whether that's the most appropriate dose, and the fact that we're probably not going to have answers about a confirmatory trial for many years. Thank you.
Thank you. Dr. Coons?
Thank you. This is Dr. Pamela Coons, and I voted no. I agree with comments made. The main reasons for my vote were that I had concerns about efficacy over existing treatments, and I also have concerns about dose finding and finding the appropriate dose. Thank you.
Thank you. Dr. Madan?
Hi, Robbie Madden, NCI. I voted no. And this is in the context of an existing therapy with trastuzumab that is available for this population. I view the poziotinib data as very compelling preliminary evidence that I look to find confirmation in future trials. I think we've discussed a lot of interesting hypotheses today that this agent may be effective in patients who are not able to tolerate or not able to get the other HER2-directed agent, or it may have better CNS penetrance. I, again, look for confirmation of that, those hypotheses in the upcoming trials. And, you know, while we should always respect the ability of the oncologist in the clinic to manage toxicities, you know, I think we have to set the patients, or the providers, but more importantly the patients, up for success as best we can by getting them the best data on what doses are best tolerated. Thank you.
Thank you. Dr. Waldman?
Scott Waldman, Thomas Jefferson University. I voted no for many of the same reasons that everybody else has articulated. Clearly, the drug there's a clinical unmet need for the drug. The drug has activity, but I don't know that it has a meaningful improvement over other drugs that in the real world are available to patients right now. It has a high level of toxicities associated with it. Yes, they can be managed, but there's still a high level of toxicities associated with that. That's complicated by the fact, as many have pointed out, that the dose of the drug has not been adequately optimized, and that uncertainty is going to be carried through now into the potential confirmatory trial, which has an endpoint that is far away, given that these patients are at risk for toxicity, and again, isn't studying the same dose that's seeking approval right now for the agent. So for all of those reasons and others, I voted no.
Thank you, Dr. Waldman. So just to summarize to some extent the voting, clearly it's evident that it wasn't an easy decision for most of us. And for those who voted yes, it does appear that relates to the efficacy of this agent in a patient population that is who is in need of active therapies instead of orphan disease. Those who voted yes were not too enthusiastic, or rather were not too worried or concerned about the tolerability of this agent just based upon prior experience and years of practice. And clearly were not concerned as to confirmatory trials and the need for those trials to be here ongoing at least. For all of us who voted no, I think that the theme is the quote-unquote non-meaningful difference compared with existing agents. Clearly, we all were concerned about not ideal dosing and the dose finding or the dose optimization issues that have been noted. And certainly the lack of an ongoing confirmatory trial that may take a long while before we know the true dose and the true efficacy of an agent. Certainly believe that all of us were quite sympathetic to the statements by the public, by all the presenters from the FDA and also from the applicant perspective. And clearly, I believe we did have a robust discussion and a pretty active session of clarifying questions. And I think that that probably is reflected on the vote as we see it. So before we adjourn this topic, are there any last comments from the FDA?
This is Harpreet Singh. No, we at the FDA would simply like to thank you, Dr. Garcia, for your moderating of this panel, and certainly the members of the committee. We thank you. We appreciate the robust discussion. We take account more than just your yes or no vote, but also all
comments, very thoughtful and meaningful comments. Thanks to patients for spending their time with us today. So we appreciate the committee's time, and thank you again.
Thank you. We will now adjourn the first topic and break for lunch. We will reconvene at 1:50 p.m. Eastern Standard Time. Panel members, please remember that they should not be chatting or discussions of the meeting topics with other panel members. Additionally, those panel member participants in the second topic should plan to rejoin the call at 1:20 p.m. Eastern Standard Time, just to ensure that we are connected before we reconvene at 1:50. Thank you again, and I appreciate all of you. Good afternoon and welcome. I would first like to remind everyone to please mute your line when you're not speaking. For media and press, the FDA press contact is April Grant. Her email and phone number are currently displayed. My name is Jorge Garcia, and I will be chairing today's meeting. I will now call the next session of the September 22, 2022 meeting of the Oncology Drug Advisory Committee to order. Dr. Shixia Shen is the designated federal officer for this meeting, and she will begin with introductions.
Good afternoon. My name is Shixia Chen, and I'm the designated federal officer for this meeting. When I call your name, please introduce yourself by stating your name and affiliation. We'll first start with ODAC members. Dr. Garcia.
Jorge Garcia, GU Medical Oncologist and the current chair of the Solid Tumor Oncology Program at University Hospital's Sidman Cancer Center, Case Western Reserve University in Cleveland, Ohio.
Dr. Kunz.
Hi, my name is Pamela Kunz. I'm a GI medical oncologist and director of the GI Cancer Program at Yale Cancer Center in New Haven, Connecticut.
Dr. Liu.
Hi, everybody. My name is Chris Liu. I'm a GI medical oncologist and associate director for clinical research at the University of Colorado Cancer Center.
Dr. Madan.
Good afternoon. My name is Riley Mann. I'm a medical oncologist at the National Cancer Institute with a focus on prostate cancer.
Mr. Mitchell.
Good afternoon. I'm David Mitchell. I am the consumer representative to the ODAC. I'm the founder of Patients for Affordable Drugs, and I'm a multiple myeloma patient.
Dr. Nieva.
Hello, I'm George Nieva. I'm the section head of solid tumors at the University of Southern California, Norris Comprehensive Cancer Center, and the Keck School of Medicine of USC.
And Dr. Song.
Anthony Sung. I'm an associate professor of medicine in the Division of Hematologic Malignancies and Cellular Therapy, Duke University. Thank you.
Now we'll move on to temporary voting members. Dr. Chen.
Hi, I'm Andy Chen. I'm at Oregon Health Science University, where I focus on lymphoma.
Dr. Crawford.
Good afternoon. My name is Stephanie Crawford. I'm professor in the Department of Pharmacy Systems, Outcomes, and Policy at the University of Illinois, Chicago, and Executive Associate Dean for Faculty Affairs and Strategic Initiatives for the College of Pharmacy. My expertise is drug safety and health equity in the medication use process.
Dr. DeFleece.
Good afternoon. I'm a gastroenterologist, a patient advocate with multiple myeloma.
Dr. Freeland.
Good afternoon. I'm Boris Friedland. I'm a chief of biostatistics branch in the Division of Cancer Treatment and Diagnosis, National Cancer Institute.
Dr. Harrington.
Good afternoon. David Harrington, biostatistician, Dana-Farber Cancer Institute, Harvard School of Public Health.
Dr. Kwok.
Hi, my name is Mary Kwok. I'm a clinical associate professor in the Division of Hematology at the University of Washington. I'm a clinician in the myeloma service at the Fred Hutchinson Cancer Center.
Dr. Nowakowski.
Good afternoon. I'm Greg Nowakowski. I'm a malignant hematologist at Mayo Clinic Rochester, where I also serve as a deputy director of Mayo Clinic Cancer Center for Clinical Research.
Dr. Sekaris.
Good afternoon, everyone. I'm Mikhail Sekaris, professor of medicine and chief of the Division of Hematology at the Sylvester Cancer Center, University of Miami, also former standing member and chair of ODAC.
And Dr. Woolman.
Good afternoon, everybody. My name is Scott Waldman. I am the chair of the Department of Pharmacology, Physiology, and Cancer Biology at Thomas Jefferson University. I'm an internist. Subspecialty boards are in clinical pharmacology, and I study, my area of research is GI oncology.
Thank you. Next is acting industry representative to the committee. Dr. Kraus?
Yes, hi everyone. Albert Kraus. I'm work for Pfizer, and I've been involved with many companies over the last few decades doing drug discovery and development work. I'm particularly focused in oncology and a lot of different developments in various tumor areas. So look forward to today's discussion.
Lastly, we'll introduce FDA participants. Dr. Pazzer?
Hi, Richard Pazzer, and I'm the Director of the Oncology Center of Excellence at the FDA.
Dr. Fiore?
Yeah, hi, my name is Mark Fiore, and I'm Deputy Center Director of the Oncology Center of Excellence.
Dr. Gormley?
Hi, I'm Dr. Nicole Gormley. I'm the Director of the Division of Hematologic Malignancies 2 here at the FDA. Thank you.
Dr. Kanapuru?
Hi, I'm Bindu Kanapuru. I'm a hematologist oncologist physician and the team lead in the Division of Hematologic Malignancies 2 at the FDA.
And Dr. Schwarzing.
Hi, I'm Alexandria Schwarzen, a clinical reviewer in the Division of Hematologic Malignancies 2 at the FDA.
For topics such as those being discussed at this meeting, there are often a variety of opinions, some of which are quite strongly held. Our goal is that this meeting will be a fair and open forum for discussion of these issues, and that individuals can express their views without interruption. That's a gentle reminder. Individuals will be allowed to speak into the record only recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the Government in the Sunshine Act, we ask that the Advisory Committee members take care that their conversations about the topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with the FDA about these proceedings. However, FDA will refrain from discussing the details of this meeting with the media until its conclusion. Also, the committee is reminded to please refrain from discussing the meeting topic during the break. Thank you. Dr. Shixia Chen will now read the conflict of interest statement for the meeting. Dr. Chen.
Thank you, Dr. Garcia. The Food and Drug Administration, FDA, is convening today's meeting of the Oncologic Drug Advisory Committee under the authority of the Federal Advisory Committee Act, FACA, of 1972. With the exception of the industry representatives, all members and temporary voting members of the committee are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on status of this committee's compliance with federal ethics and conflict of interest laws, covered by but not limited to those found at 18 U.S.C. Section 208, is being provided to participants in today's meeting and to the public. FDA has determined that members and temporary voting members of this committee are in compliance with federal ethics and conflict of interest law. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflict when it is determined that the agency's need for a special government employee's services outweighs his or her potential financial conflict of interest, or when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Related to the discussions of today's meeting, members and temporary voting members of this committee have been screened for potential financial conflicts of interest of their own, as well as those imputed to them, including those of their spouses or minor children, and for purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, CARDACs, teaching, speaking, writing, patents, and royalties, and primary employment. Today's agenda involves receiving updates on new drug application NDA 2143A3 for PEPAXTO, melphalan flufenamide for injection, submitted by Oncopeptides A.B. This product was approved under 21 CFR 314.500 subpart H accelerated approval regulations for use in combination with dexamethasone for the treatment of adult patients with relapsed or refractory multiple myeloma who have received at least 4 prior lines of therapy and whose disease is refractory to at least 1 proteasome inhibitor, 1 immunomodulatory agent, and 1 CD38-directed monoclonal antibody. The confirmatory trial demonstrated a worse overall survival and failed to verify clinical benefit. Confirmatory studies are post-marketing studies to verify and describe the clinical benefit of a drug after it receives accelerated approval. Based on the updates provided, the committee will have a general discussion focused on next steps for the product. This is a particular matters meeting during which specific matters related to the Oncopeptides A.B.'s NDA, approved under 21 CFR 314.500 subpart H, accelerated approval regulations will be discussed. Based on the agenda for today's meeting and all financial interest reported by the committee members and temporary voting members, conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208 B3 to Drs. Mary Kwok and Greg Nowakowski. Dr. Kwok's waiver involves her employer's research contracts for 4 studies funded by competing firms. 1 study is funded by Harpoon Therapeutics, and Dr. Kwok's employer receives between $250,000 and $300,000 per year. The second study is funded by Celgene, and Dr. Kwok's employer receives between $200,000 and $250,000 per year. The third study is funded by Nectar Therapeutics, and Dr. Kwok's employer receives between $300,000 and $350,000 per year. The fourth study is funded by Jensen, and Dr. Kwok's employer receives between $250,000 and $300,000 per year. Dr. Nowakowski's waiver involves his employer's research contracts for 4 studies funded by competing firms. 1 study is funded by Amgen, and Dr. Nowakowski's employer receives between $250,000 and $300,000 per year. The third study is funded by Novartis, and Dr. Nowakowski's employer receives between $0 and $25,000 per year. The third study is funded by a competing firm, and Dr. Nowakowski is not aware of the funding amount being provided to his institution for the study. The fourth study is funded by Celgene, and Dr. Nowakowski's employer receives between $0 and $25,000 per year. The waivers allow these individuals to participate fully in today's celebration. FDA's reasons for issuing the waivers are described in the waiver documents, which are posted on FDA's website at www.fda.gov/advisory-committees/human-drug-advisory-committees. Copies of the waivers may also be obtained by submitting a written request to the agency's Freedom of Information Division, 5630 Fisher Lane, room 1035, Rockville, Maryland 20857. Or requests may be sent via fax to 301-827-9267. To ensure transparency, we encourage all standing members and temporary voting members to disclose any public statements that they have made concerning the product issue. With respect to FDA's invited industry representative, we'd like to disclose that Dr. Albert Krause is participating in this meeting as a non-voting industry representative. Acting on behalf of regulated industry, Dr. Krauss' role at this meeting is to represent industry in general and not any particular company. Dr. Krauss is employed by Pfizer. We would like to remind members and temporary voting members that if the discussions involve any other products or firms not already on the agenda, which an FDA participant has personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all other participants to advise the committee of any financial relationships that they may have with the firm at issue. Thank you.
Thank you, Dr. Chen. We will now proceed with the FDA introductory comments from Dr. Nicole Gormley. Dr. Gormley?
Great, thank you. Good afternoon. I'm Nicole Gormley, a hematologist and the director of the FDA's Division of Hematologic Malignancies II. I will provide a brief introduction to the issues presented by the melphalan flufenamide application, which I will hereafter refer to as melflufen. I'd like to briefly review the evidentiary criteria for approval. It is important to note that drugs granted accelerated approval or traditional approval must meet the same statutory requirements for safety and effectiveness. For safety, there must be sufficient information to determine that the drug is safe for use under the conditions prescribed, recommended, or suggested in the proposed labeling. For effectiveness, there must be substantial evidence of effectiveness based on adequate and well-controlled investigations that allow for the conclusion that the drug will have the effect it is represented to have in the proposed labeling. There are two approval pathways available in the U.S., regular approval and accelerated approval. Accelerated approval is available for drugs or biologics that are intended to treat a serious or life-threatening illness. The product should provide a meaningful therapeutic benefit over available therapy, and approval is based on an endpoint reasonably likely to predict clinical benefit or an intermediate endpoint. For products granted accelerated approval, there is a requirement to conduct post-approval trials to verify the anticipated clinical benefit. The sponsor has highlighted in the briefing document the regulatory decisions of the EMA. The regulatory actions of other agencies are not relevant to the discussions at today's ODAC or any FDA regulatory decisions. The FDA must make regulatory decisions that adhere to U.S. laws and regulations. The information discussed at the ODAC should be viewed independently to inform decisions regarding the benefit-risk of melflufen for the indicated U.S. patient population. Also of note, the U.S. FDA is the only regulatory agency that reviews the primary source data, conducting our own analyses to inform the benefit-risk profile of a given product. This table shows the drug and biologic regimens approved for the treatment of relapsed refractory multiple myeloma, with the approval specifically for patients who have received at least 4 prior lines shown in red. Of note, many of the other therapies not in red can be used to treat patients who have received 4 prior lines of therapy or those with refractory disease, but those in red are specifically approved for these later line populations. Unlike solid tumors, in some cases, patients with multiple myeloma may be retreated with the same agent or the same agent in combination with different combination partners. Melflufen was granted accelerated approval in February 2021, but the confirmatory trial failed to confirm the clinical benefit. Specifically, the overall survival result was worse than that observed in the comparator arm, pomalidomide dexamethasone, and there was not a demonstration of PFS superiority. With regards to the regulatory history, as mentioned, melflufen was granted accelerated approval in 2021 based on the results of the single-arm trial Horizon. Melflufen is an alkylating drug indicated in combination with dexamethasone for the treatment of adult patients with relapsed or refractory multiple myeloma who have received at least 4 prior lines of therapy and whose disease is refractory to at least 1 proteasome inhibitor, 1 immunomodulatory agent, and 1 CD38-directed monoclonal antibody, typically referred to as triple-class refractory disease. The top-line results shared with the FDA showed that the OCEAN trial failed to demonstrate PFS superiority and suggested worse survival results. After the FDA expressed concerns with the results, the sponsor submitted a reanalysis of PFS based on reassessment of 29 patients, which indicated nominal superiority. The FDA concerns were not allayed with this reassessment, and the IND was placed on clinical hold and a CDER safety alert was issued. The agency planned to present this information at an ODAC scheduled for October 28, 2021. After receipt of the FDA briefing document, an Oncopeptides board member requested a meeting with the FDA, which was held on October 19th, in which the FDA expressed concerns with the application and presented options and recommendations to the board member and sponsor. A second meeting was held with the company on October 20th, in which the sponsor stated that they planned to voluntarily withdraw the NDA and requested the FDA to cancel the ODAC meeting and requested that the FDA briefing document not be made public. On October 22nd, the NDA withdrawal request was received and the ODAC was canceled. The FDA initiated the administrative processes to withdraw the NDA. During this time, there were no additional analyses conducted by the FDA, and there was no communication from the sponsor regarding marketing of the product until the receipt of the notice from the sponsor to rescind the withdrawal request in January 2022. The sponsor has not submitted new clinical data with melphalan, but rather post-hoc exploratory subgroup analyses from existing trials and analyses of IMID trials external to the OCEAN trial. The FDA is reconvening an ODAC now to discuss the benefit-risk profile of melphalan. Most recently, on October 12, 2022, the sponsor proposed postponing this ODAC to allow for consideration of results with a separate external trial that does not include melphalan, but includes an IMID in the control arm. With regards to the top-line results, the original IRC-assessed progression-free survival results submitted to the agency showed a PFS hazard ratio of 0.8 and failed to meet statistical significance for superiority. The sponsor conducted a readjudication of 29 patients and subsequently claimed that statistical significance was met. The FDA does not agree that PFS statistical significance has been met. The PFS results and subsequent readjudications will be discussed later in the FDA presentation. While the PFS results are important, the FDA's primary concerns lie with the overall survival results. Shown here are the original overall survival results. Notably, the OS analysis showed a hazard ratio of 1.1, with a median OS of 19.7 months in the melphalan arm compared with 25 months in the pomalidomide arm. While this is not a statistically significant result, and this is a trial against an active comparator, the available data suggests that patients who receive melphalan dex have unfavorable outcomes compared to those receiving pomalidomide dex. Previously, the sponsor proposed that the detriment observed in OS may be due to prior transplant and subsequent therapies received by these populations. However, it should be noted that there were multiple subgroups that had a worse overall survival, including those aged less than 65, those with 3 or 4 prior lines of therapy, those with better creatinine clearance, and those with extramedullary disease, among others. Most recently, the sponsor has proposed that the potential detriment observed in OS is due to those that received a transplant previously and had a time to progression after transplant less than 36 months, and that melphalan is safe in patients who have never been transplanted and those who have been transplanted but had a time to progression of 36 months or more. However, when looking at the forest plot, we see that while patients with a time to progression of 36 months or more had a point estimate of 0.79, the confidence interval crosses 1, and this subgroup is small, with only a total of 43 patients, so we cannot say that this group is without harm. The confidence interval for those who have never received a transplant also crosses 1. The FDA analysis does not support that the overall survival results are due to worse outcomes only in those that received a transplant and had a time to progression after transplant less than 36 months. Furthermore, this is a post-hoc exploratory subgroup analysis, and there are multiple challenges with post-hoc subgroup analyses that limit their utility beyond serving as the basis for hypothesis generation for subsequent study. One of the fundamental underpinnings of clinical trial research is control of type 1 error probability. Type 1 error probability is the chance of finding a difference when there is none. Conventionally, the type 1 error is set at 5% or less. Stated another way, if you assume no difference between the arms, if you perform the test 100 times, 5 times you will observe a difference as large as the one observed, but it would be a false positive. We accept this level of risk, but this is the significance level for one test. There are statistical methods to control the type 1 error when there are multiple analyses, but these must be pre-specified. So when thinking about subgroup analyses, there is often interest in comparing treatments among subsets of patients using recognized prognostic factors, such as age, gender, stage, histology, among others. If there were only 3 binary factors, 8 subsets could be formed. If you compare the treatments among these 8 subsets, there would be a 33% probability to observe a statistically significant treatment effect, even if there were no true difference between the treatments. To illustrate this point a little further, I would like to share with you an example from the field of cardiology. The ISIS-2 trial randomized more than 17,000 patients who were suspected of having an MI to either streptokinase, aspirin, both, or neither in a placebo-controlled fashion. Streptokinase alone, aspirin alone, and the combination significantly reduced the rate of 5-week vascular mortality compared with placebo alone. In reporting the results in The Lancet, the editors urged the authors to include nearly 40 subgroup analyses. The authors agreed on the condition that they also include an analysis based on astrological signs. The result? Geminis and Libras had an adverse effect from aspirin, a 9% increase in mortality, compared to patients in other astrological signs who had a 28% reduction, with the p-value shown there. The authors mentioned that even in a trial as large as ISIS-2 with over 17,000 patients, subgroup analyses are unreliable and potentially misleading. Instead, more weight should be given to the overall result than to data from subgroups of interest. Given these concerns, the FDA and other regulatory bodies through the International Council for Harmonization, or ICH, provided regulatory guidance on the use of subgroup analyses in the ICH E9 guidance document. Specifically, the ICH E9 states that any conclusions of treatment efficacy or safety based solely on exploratory subgroup analyses is unlikely to be accepted. Additionally, only results from analyses envisaged in the protocol can be regarded as confirmatory. This table shows the post hoc analyses submitted to the FDA by the sponsor to explain the observed OS results. While prior transplant, yes or no, was included as an exploratory analysis in the statistical analysis plan, it was without type 1 error control. The analyses shown in the table were not included in the original statistical analysis plan and did not have a type 1 error plan established. We also do not know what other post hoc analyses the sponsor conducted but did not submit to the FDA. Additionally, the sponsor also asserts that there is an age interaction with overall survival in the IMiDs, as observed in the OCEAN trial and other trials of IMiDs, and that these interactions significantly confound the overall survival results. There are several flaws in the sponsor's argument, which will be discussed in more detail later in the FDA presentation, but suffice it to say that the sponsor's analyses do not support this assertion. Additionally, FDA conducted their own analysis of data with IMiDs and did not find evidence of an age-OS interaction with the IMiDs. But even if there were an interaction, the preponderance of evidence from the pre-specified analysis on the ITT population demonstrates a hazard ratio greater than 1 in the melphalan arm compared to the pomalidomide arm, and the data from the OCEAN trial does not provide substantial evidence of the safety and effectiveness of melphalan. One factor which may be contributing to the overall survival finding is the dose. The 40 milligram fixed dose is poorly tolerated, and there are multiple safety events associated with higher exposure. As we will hear later in the FDA presentation, there were high rates of dose modification in the OCEAN trial. 78% of patients experienced at least one adverse event leading to dose modification. 47% of patients experienced at least one adverse event leading to dose reduction, and 26% of patients experienced at least one adverse event leading to drug discontinuation. These rates were significantly higher in the melphalan arm as compared to the pomalidomide arm. PK exposure response analyses suggest that weight or body size-based dosing may be more appropriate than the currently approved flat dose, and analyses suggest that the current 40 milligram dose may not be the correct exposure target. A lower dose may be more tolerable and may have similar efficacy. So in conclusion, there are several issues presented by the melphalan application, but there are three central issues. First, in the confirmatory trial OCEAN, melphalan dex demonstrated a potential detriment in overall survival compared to pomalidomide dex. Several subgroups have been identified that performed worse in the trial, but as previously stated, these post-hoc exploratory analyses should only be used to inform future trial design and cannot be relied upon to provide substantial evidence of safety and effectiveness. Second, the trial failed to meet the primary endpoint of a statistically significant improvement in PFS. The PFS reassessment by the sponsor and concerns regarding the censoring rules used will be discussed later in the FDA presentation. Third, there remains significant concern regarding the dose of melphalan. So from a regulatory perspective, we are in a situation where the randomized controlled trial has shown a potentially worse overall survival compared to an active comparator. We cannot adequately assess overall survival from single-arm trials, so we cannot rely on the initial single-arm trial horizon to assess the overall survival. With the available data, we are unable to assess if melphalan is causing harm in the currently indicated patient population. The toxicity, dose modifications, and subgroup analyses suggest the potential for harm. Lastly, accelerated approval requires that the drug provide a meaningful advantage over available therapies. Given what is currently known, we would not have granted accelerated approval to melphalan, as the concerns and issues outlined above would preclude a conclusion that melphalan provides a meaningful advantage over available therapies. Given these major concerns, we would like for the committee to discuss the benefit-risk profile of melphalan for the currently indicated patient population, considering the results of the confirmatory OCEAN trial. The voting question is, given the potential detriment in overall survival, failure to demonstrate a progression-free survival benefit, and lack of an appropriate dose, is the benefit-risk profile of melphalan favorable for the currently indicated patient population? Please note that we are asking for the committee members to use your clinical and scientific expertise to assess the benefit-risk profile of the product based on data and discussions presented at this meeting. I'd like to make a comment in closing. You will hear in the sponsor's presentation and the open public hearing about the need for additional therapies. Please note that at the FDA, we strongly agree with that sentiment. Many of us have had family members affected by cancer or cancer ourselves. So when applications are submitted for new therapies that are effective, we do all that we can to expedite the regulatory decision for these therapies. But the need for new therapies must be balanced with the fact that we must first do no harm. The sponsor has proposed a new indication based on a subgroup and a different dosing strategy for select patients. Any indication granted must meet the same clinical and statistical rigor as would be expected for a new application. You can't carve out a new population without studying it. The product should be studied prospectively and demonstrate in a rigorous manner that it is safe and effective in this new population at the proposed dose. Please bear this in mind during the remaining presentations and discussions today. Thank you very much.
Thank you, Dr. Gormley. Both the Food and Drug Administration, FDA, and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages all applicants, including the Oncopeptides A.B.'s non-employee presenters, to advise the committee of any financial relationships that they may have with the sponsor, such as consulting fees, travel expenses, honorarium, and interest in the sponsor, including equity interest and those based upon the outcome of the meeting. Likewise, FDA encourages you at the beginning of your presentation to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your presentation, it will not preclude you from speaking. We will now proceed with presentations from Oncopeptides A.B.
Good afternoon. My name is Jacob Lindberg with Oncopeptides. We are here today to give a different perspective to the FDA's voting question. Rather than waiting to hear your independent ODAC assessment, they have asked the leading voting question that has already determined that there is a survival detriment when compared to well-established active therapy. that the primary PFS endpoint based on blinded IRC review was not met, and that the dosing for this cytotoxic agent is inappropriate. We are here because we strongly believe that patients need to understand the implications from the newly identified interactions that affect the interpretation of OCEAN. One for PEPAXTO that can lessen potential risk, and an unexpected independent age interaction for immunomodulators. And this is important. The median patient on IMiD therapy in the US right now is 74 years old. This information is too important for patients to already predetermine the interpretation. In the upcoming months, as independent experts assess these data, I personally believe that OCEAN will be remembered as the canary in the coal mine regarding this key interaction between IMiDs and age. And I hope it will help guide the assessment of heterogeneous studies with an active comparator. We only ask you to keep an open mind. We assert that OCEAN met its primary endpoint of superior PFS based on pre-specified independent review committee evaluation using IMWG response criteria and the pre-specified statistical analysis plan. In the docket, you can see a letter from the chairman of the IRC stating that the IRC followed IMWG guidelines for disease progression and was completely blinded throughout the process to assess the primary endpoint. We agree that OCEAN demonstrated an overall survival hazard ratio of 1.14 in the full analysis population as of the latest update. However, the hazard ratio in comparison to an active comparator doesn't tell the full story, given the significant survival heterogeneity for both study drugs. Because the primary endpoint was confirmed in accordance with the pre-specified analysis plan, this allows for assessment for homogeneity of treatment effect across pre-specified subgroups. And it is here that OCEAN has a lot to teach us. OCEAN identified significant PFS and OS differences for PEPAXTO based on patients' prior exposure to an autologous stem cell transplant. These data merit the limitations of use that I will review shortly. For pomalidomide, OCEAN identified unexpected significant survival differences based on patient age that was not reflected in the surrogate endpoint of PFS and which has not been properly discussed in the literature. This is the major confounder for the OS interpretation in OCEAN. Given these data, in the recommended patient population, the confirmatory study OCEAN fulfilled accelerated approval obligations to confirm benefit risk in a population that desperately need access to products with novel mechanisms of action. Let us further review the heterogeneity that confounds the OS interpretation in OCEAN. Here you see that PFS for pomalidomide treatment across pre-specified age subgroups is identical. However, OS differs significantly. The hazard ratio between old and young patients is 2 to 3x. In relapsed refractory myeloma, FDA's own analysis on age in 2019 shows that the survival should be very similar across these age groups at this stage of the disease. In OCEAN, there was a clear deterioration in survival benefit with increasing age for pomalidomide. This phenomenon is replicated across studies that isolate pomalidomide or other immunomodulatory drugs that allow for detailed survival analysis with consistency. Further details regarding this data can be found in the appendix of your briefing book as well as in the public docket. The forthcoming DREAM3 study will corroborate this phenomenon. To the left, you see the heterogeneity for both PFS and OS for PEPAXTO based on prior ACT. For PEPAXTO, this reveals an identified subgroup risk which should be addressed in labeling. As per the ICH guidelines for clinical studies that meet the primary endpoint, a review of pre-specified subgroups should be conducted to assess homogeneity of treatment effect, since ITT may not adequately characterize treatment effects across patient subgroups. This assessment is intended to identify patients that may be at increased risk. treatment effect differences need to be considered prognostic or predictive. This requires the finding to be in line with the mode of action and biological rationale, along with precedent in other trials, and the finding needs to be supported by other endpoints in the trial. Prior ACT was a pre-specified subgroup in OCEAN and is regularly used for treatment decisions in myeloma. An ACT interaction for PEPAXTO aligns with biology and mechanism of action. Patients receive conditioning therapy in conjunction with ACT that typically consists of high-dose melphalan treatment. This likely induces partial or complete drug resistance to further alkylator-based therapy. On the pre-specified subgroup level, prior ACT is the patient group that would be excluded from treatment with PEPAXTO. However, albeit the post-hoc analysis, the patient group with real increased risk with PEPAXTO treatment are those patients with a less successful ACT in line with treatment guidelines. They describe that tumors relapsing early after ACT are more resistant to further alkylator-based therapy. Considering the OCEAN data, we are recommending a limitation of use that aligns with clinical treatment guidelines and published recommendations regarding ACT. PEPAXTO should not be used in patients with post-ACT progression less than 3 years after transplant. We propose to include this in the prescribing information and adequately inform physicians of this update. Let me summarize the important considerations that address the 3 FDA concerns. First, PEPAXTO met its primary endpoint with statistically significant superior PFS based on blinded IRC review. The chair of the IRC has submitted written verification of this outcome. Therefore, it's appropriate to look further into subgroups to identify potential heterogeneity of treatment effect. We found that the OS risk with PEPAXTO in comparison with pomalidomide is driven by patients with prior ACT. Removal of this subgroup with risk improves both efficacy and safety for PEPAXTO. And this is a biologically plausible risk, which is why we propose to update the label to include the limitations of use. Additionally, PEPAXTO is an alkylating cytotoxic drug being used in a severely ill patient population with very aggressive disease. As such, it is appropriate to administer at the maximum tolerated dose to establish tumor control. And when looking at the recommended population, there were less dose modifications, higher response rate, and patients were able to remain on treatment longer. With this information in mind, here is the agenda for the remainder of the presentation. We also have additional experts to answer your questions. Thank you. I'll now turn the presentation to Dr. Richardson.
Thank you very much, Dr. Lindberg. Good afternoon, ladies and gentlemen. It is my privilege to provide a discussion on the unmet medical needs in relapsed and refractory multiple myeloma. My name is Paul Richardson, and I serve as the R.J. Corman Professor of Medicine at Harvard Medical School, as well as Clinical Program Leader and Director of Clinical Research at our center. I was Principal Investigator on the Horizon study, and our site was one of the largest enrollers. Moreover, subsequent to its accelerated approval last year, we at our center prescribed PEPAXTO according to the label prior to the suspension of that last year. It's important to add that I've been treating patients with relapsed and refractory myeloma for more than 20 years and currently have a busy clinical practice with up to 4 clinics a week where we see patients for both participation in clinical trials as well as for receiving treatment as standard of care. And please note, I am not being compensated for my time in today's meeting. Now, the treatment of multiple myeloma is truly a marathon and not a sprint, and strategic and practical considerations are absolutely key, particularly in the relapsed and refractory setting. Very importantly, this can improve long-term outcome, and in our practice, we run out of options, unfortunately, every week as myeloma progresses rapidly once the disease becomes increasingly resistant and refractory. At this stage of the disease course, the goal of treatment is to stop further progression and maintain disease control, preserve quality of life, and recognize that these patients are unfortunately past cure. From my clinical experience with PEPAXTO, this has been a generally well-tolerated drug that is easy to administer in the outpatient setting and meaningfully adds, in our experience, to the treatment paradigm. Not having PEPAXTO readily available over the past 9 months has been a real loss in my view for both our patients and for us as providers. Now let's turn to the evolving role of autologous stem cell transplant in the management of multiple myeloma. It's important to outline that outside of triplet or quadruplet treatment combinations we now typically use for newly diagnosed disease, we also have autologous stem cell transplant as a first-line treatment in younger, fit, eligible patients. And it's important to also note that outcomes had previously improved with the introduction of autologous transplant over 25 years ago, which at that time improved median survival between 2 to 5 years. However, currently only about half of patients with multiple myeloma are eligible for ASCT, typically due to advanced age and frailty, and of those eligible, only about a third currently undergo transplant. For those not eligible or after initial treatment fails, they can quickly cycle through other current available options and need newer and more abundant options urgently. Now, very importantly, novel therapies have been critical in improving long-term outcomes, regardless of whether a patient has had a transplant or not. In fact, we've seen this effect, especially in younger patients, as shown in recent randomized practice-changing studies of early use of transplant versus delayed or deferred ASCT. Now, in terms of the current treatment landscape for relapsed refractory myeloma, we heard very nicely from this from a moment ago from Dr. Gormley. There are 3 primary classes of drugs, and these consist of proteasome inhibitors, immunomodulatory agents, and monoclonal antibodies. And generally, when one progresses on a particular regimen, we try to switch classes of agents and or utilize a next-generation drug. Yet as disease continues to progress and become refractory, these options rapidly become exhausted. Now, if one of our 3 go-to novel agents targeting BCMA and other newer targets is not improving outcomes, and especially in the elderly, the need for additional therapies with novel mechanisms of action are absolutely vital for continued successful salvage. Now, while specifically anti-BCMA therapies have clearly improved outcomes for some patients, they do, however, come with their own challenges regarding both ease of administration and tolerability. These issues are not mutually exclusive. We need all of these therapies and more, in particular for our older patients. And this, in my view, is where PEPAXTO fits nicely, adding meaningfully to the treatment landscape where its novel mechanism of action as a peptide drug conjugate and its use importantly as an outpatient can add benefit. Now, the challenges with the efficacy of current treatments in relapsed refractory myeloma are several, but to understand patient need and treatment selection in the current era, and especially as these evolve, we also need to address the challenges with the efficacy of current treatments. And this is why the OCEAN results have caused experts to re-examine the data and reassess decision-making in this context. As one of the 3 cornerstones of our combination treatments, IMiDs are extensively used to treat myeloma, and it is estimated that about 80% of our patients who receive IMiD therapy are indeed over the age of 65. And as you heard, a very important and plausible age-related interaction has been identified in OCEAN and now in other studies. Whilst it's not a specific question in today's discussion, this IMiD interaction is a concerning finding that in my view needs further evaluation and also needs to be communicated. We are leading the development of a forthcoming paper with colleagues to share the data and hopefully prompt additional investigation as well as careful consideration of the implications in our treatment paradigm. In sum, you can see this interaction that was unexpected since IMiDs have been used widely for over 15 years. However, a retrospective evaluation of the data in the literature has found little information regarding efficacy by age, especially in our older patients. So I share with you on this slide new data derived from myeloma studies that show that overall survival hazard ratios worsened with older age for IMiDs consistently across the studies we examined, with younger patients doing better on IMiDs than their older counterparts. You can see the studies included on the right and the hazard ratio separated by age within those studies, with each reflected by the colored dots. This negative interaction with advancing age should be considered when making treatment decisions, in my view. Yet importantly, the current pomalidomide label doesn't provide this subgroup information. Patients and, of course, providers should have full transparency of data, in my opinion, so we can adequately communicate the benefit-risk of therapies and determine the most appropriate therapeutic option for our patients during their treatment course. Now, it's also important to emphasize that other recently approved therapies for refractory disease have challenges. As mentioned, we essentially have 3 viable choices: XPO1 inhibition, the use of antibody drug conjugates, and cellular therapies. Selinexor has considerable GI toxicity and anemia, which can be especially challenging with upwards of 40 patients discontinuing their prescribed dosing during clinical trials to date. Belantamab mafodotin provides similar efficacy as PEPAXTO, but comes with significant ocular toxicity, which can be poorly tolerated and leads to discontinuation, particularly in older patients. And although manageable with dose reduction and schedule change, it occurs in over 60% of patients treated. It also requires sophisticated input with expert ophthalmic care, as well as frequent monitoring. And finally, CAR T therapy is very effective when attainable. However, it lacks ready accessibility, and many patients have had to wait almost 6 months in real-world experiences to receive treatment, according to a recent ASCO abstract. That's not at all practical for patients with relapsed refractory disease, who are of course in immediate need of therapy. Toxicities can be substantial, with almost all patients experiencing cytokine release syndrome and a significant number encountering complex CNS issues, some of which can be very serious. Hospitalizations are of course a routine and required part of management, which further stretches resources, especially in the COVID era. Now, as we think about overall survival in relapsed refractory multiple myeloma, it's clear that given the short survival in triple-class refractory patients that we heard defined so nicely earlier by Dr. Gormley, it's key to understand in that group that multiple options are absolutely needed. I also want to be clear, as we start our conversation today, that the OS seen with PEPAXTO is promising and meaningful in my view compared to current historical norms. For example, for triple-class refractory patients, as an example, median overall survival has been reported to be at best 9.2 months, and in penta-refractory patients just 5.6 months, with double refractory patient outcomes estimated at a median of 11.2 months. For PEPAXTO, consistent improvements in overall survival compared to these contemporary historical controls are seen, reflecting on the one hand, in my view, its novel mechanism of action in the context of current therapies, and is replicated across phase 1, 2, and now phase 3 studies in these specific patient populations. Now, when we consider special considerations for patients with relapsed refractory disease, I've summarized them here. In addition to the clinical benefits, PEPAXTO offers many practical advantages for patients with relapsed disease that shouldn't be dismissed. As noted, multiple myeloma is predominantly a disease of the elderly and highly heterogeneous. We need to be particularly mindful that not all patients can tolerate current options, and yet drugs with new mechanisms of action are essential to improving outcome, and especially in the relapse setting. In this context, data from our Horizon studies support that PEPAXTO can be used in patients with extramedullary disease, a key and very ominous feature of relapsed myeloma, especially after the failure of monoclonal antibody treatments. In fact, Horizon is one of the few studies in which we included a significant number of relapsed refractory patients with extramedullary involvement, where they made up more than 40% of the population. We saw remarkable single-agent activity in this advanced setting, with approximately 25% of patients responding, further supporting a role for PEPAXTO for this important subgroup of unmet medical need. And of course, no conversations about myeloma patient care in 2022 is complete without discussing the implications of COVID, from which our patients remain uniquely vulnerable. We now more than ever must consider the frequency of visits needed for patients to be treated and the rates of COVID infection, morbidity, and mortality seen. Patients and providers alike are turning to easier to administer outpatient-based treatments whenever possible, with the lowest rates of COVID mortality seen. PEPAXTO has this attribute and is given monthly, which is a real advantage, both reducing the risk of infection and providing additional convenience for our patients, as well as a mechanism of action which doesn't increase the risk of COVID mortality in this exquisitely vulnerable population. So to conclude, PEPAXTO in its current indication is, in my opinion, meeting an important unmet medical need with patients with triple-class refractory myeloma, which remains incurable and should be accessible to patients to meaningfully improve outcome. Patients with triple-class refractory myeloma urgently need additional treatment options that provide efficacy, above all in an outpatient setting, and that are also meaningfully different to the current BCMA-targeted treatment paradigm. The practical advantages of PEPAXTO cannot be overlooked, as these patients are often older and frailer and cannot readily tolerate currently available treatment options. The presentation of data you will hear will demonstrate clinically meaningful efficacy and a manageable safety profile, with importantly minimal non-hematologic side effects in our experience that critically reflect our own real-world experience in this setting. and the unique features specific to older patients regarding its efficacy and value. I thank you very much for your kind attention, and I'll now turn the presentation to Dr. Bakker.
Thank you, Dr. Richardson. I am Klaas Bakker, and I'm the Chief Medical Officer at Oncopeptides. I will review the results from our OCEAN study and share our learnings related to PEPAXTO's benefit risk. This study provides important data for both the efficacy and safety of PEPAXTO. I will first review the study design of OCEAN and share the efficacy results in the overall population. Then, I will share the data demonstrating a clear interaction for PEPAXTO based on prior autologous stem cell transplant. The identified heterogeneity of response in this pre-specified subgroup is biologically plausible and compelling and will guide future use and development of PEPAXTO. I will not elaborate on the age interaction with IMiDs. This material can be found in your briefing documents and the public docket. Lastly, I will review the safety data supporting PEPAXTO. OCEAN was a phase 3 randomized active control study comparing PEPAXTO plus dexamethasone to pomalidomide plus dexamethasone. Treatment was continued until disease progression or unacceptable toxicity. This study enrolled patients who had received 2 to 4 prior lines of therapy other than pomalidomide and were refractory to lenalidomide as well as their last line of therapy. Patients were also required to have an equal performance status of less than or equal to 2. In comparison, patients in Horizon, the study that supported the accelerated approval, had an average of 5 lines of prior treatment and were mostly triple-class refractory. The primary endpoint was progression-free survival as assessed by an independent review committee. Key secondary endpoints included overall response rate and overall survival. As per the statistical analysis plan, because this was a head-to-head study with an active comparator, the pre-specified anticipated difference to result in superiority was a median PFS advantage of 1.54 months. The pre-specified censoring rules, also included in the SAP, follow IMWG guidelines. Progressive disease required confirmation of a secondary measurement with 2 exceptions. If the first assessment was progression of EMD or showed a progression with rapid subsequent treatment initiation, prohibiting a second measurement, this was also considered progressive disease. Clinically, this makes sense, as patients are often put on a subsequent treatment before there is time for a confirmatory assessment. Now let's look at demographics. Baseline demographics and characteristics were well balanced between both groups. Of note, the median age was 68 years, and about half of the patients in both arms had a prior ASCT. If we look at the patient disposition, a total of 495 patients were randomized. 18 patients randomized to PEPAXTO were not treated, compared to 3 in the pomalidomide arm. With that, 228 patients received at least 1 dose of PEPAXTO, and 246 received pomalidomide. A similar percentage of patients who were dosed discontinued the study, mostly for progressive disease. Of those treated with study drug, a similar number went on to receive subsequent therapy post-progression. This means that treatment with PEPAXTO did not impact the probability of receiving subsequent therapies. If we look to the OCEAN efficacy results, the OCEAN study met its primary endpoint as defined per the statistical analysis plan, demonstrating superiority compared to pomalidomide with a median PFS of 6.8 versus 4.9 months and a hazard ratio of 0.79 and a p-value of 0.03. Here you see PEPAXTO in blue and pomalidomide in gray. Again, I would like to emphasize that the IRC agree with these findings as described in their letter, which was submitted to the public docket. Additionally, in that letter, in contrast to what the FDA states, the IRC confirms that no re- or post-hoc analysis was performed with only one final analysis, and that they were blinded throughout the full review process. The 2-month difference in median PFS is meaningful and translates to more than 40% improvement above the active comparator, which is in itself already a very efficacious drug. This is an important improvement in a study comparing 2 different mechanisms of actions. The OCEAN trial demonstrated a meaningful overall response rate of 33% for the PEPAXTO arm, and the clinical benefit rate was 50%, both numerically higher than the pomalidomide arm. Additionally, responses were equally durable in both treatment arms. Here are the Kaplan-Meier curves for overall survival. As noted earlier, the updated overall survival hazard ratio from the OCEAN trial was 1.14 in the intent to treat population. While this was not statistically significant, it suggests a potential worsening of survival, which of course deserves careful analysis. From a safety perspective, the survival curve is confounded by 18 patients assigned to PEPAXTO and 3 assigned to pomalidomide who were never treated. When looking at patients who actually received study drug, the early separation through the first year, as seen in the intent to treat population, is eliminated. This means that all the events impacting the numerically worse overall survival occurred after completing the assigned study treatment. Here are the overall survival results by pre-specified subgroups to explore the homogeneity of the overall survival hazard ratio. As you can see, as mentioned also by Dr. Gormley, there are clear, large differences in various subgroups. Careful and systematic evaluation led us to identify ASCT as the interaction driving the potential detriment for PEPAXTO. It is important to note that other subgroups like age, prior lines of therapy, and creatinine clearance are variables strongly associated with ASCT. Now let me review the impact of this biologically plausible interaction within the pre-specified subgroup of patients with and without ASCT. Here is the PFS in patients with no ASCT. You can see early and sustained separation between the curves and an improved PFS for PEPAXTO, while the PFS for pomalidomide remains the same as the overall population. As such, removal of the subgroup with identified risk further enhances the PFS benefit, now providing 4.7 additional months over pomalidomide. Other endpoints, including ORR, CBR, and median duration of response, also improve in patients with no prior ASCT. When looking at overall survival results in the group of patients with no ASCT, the median overall survival improves to 22.2 months compared to 17.5 months with pomalidomide. Now, because of the interaction with ASCT, the sponsor further investigated the finding. IMWG criteria state that an ASCT with a time to progression of less than 24 to 36 months should be considered a failed transplant. Based on the guidelines, the sponsor investigated the ASCT group by time to progression, which show a clear linear relation with efficacy results. This is in line with the biology, as an ASCT is invariably accompanied with induction therapy with high-dose melphalan. For this reason, we established 36 months as the threshold for the limitations of use. The response to high-dose melphalan can be predicted based on the time to progression following ASCT. We investigated the optimal time since transplant to better isolate the subgroup at risk. We found that the risk appears to be removed for patients that do not have progression until more than 36 months post-ASCT, as shown on this table. We detect a clear risk for potential harm for progression less than 24 months from progression. Here we show the PFS when we implement the proposed limitation of use. We see a statistically significant almost doubling of PFS with 9.3 months on PEPAXTO compared to 4.6 months on pomalidomide. The curves separate early and are sustained throughout the study. Here we show the median survival in patients with either no transplant or who progressed more than 36 months after ASCT. These are the patients that should be treated with PEPAXTO, providing improved survival over pomalidomide. In comparison, patients with early post-transplant progression had lower survival. For this reason, we aim to communicate the potential risk as a limitation of use, and patients with a time to progression of less than 36 months after an ASCT should be treated with an alternative drug. When looking at the overall survival curve for the recommended population, we see separation from pomalidomide at 3 months that continues through the duration of the study. And here is the forest plot looking at the recommended patient population based on the ASCT interaction. You see the improved point estimates favor PEPAXTO with no increased risk for potential detrimental survival anymore. Here we give context to the limitation of use, showing why we recommend to be more specific about time to progression since transplant rather than by patients with no ASCT. This change, shown at the bottom, now clearly separates the risk with separate, non-overlapping confidence intervals. Let me now share the safety data, which demonstrate a consistent and manageable safety profile of PEPAXTO. Here's an overview of the adverse event profile across OCEAN. Grade 3-4 adverse events were primarily of hematologic nature and occurred at higher rates compared to pomalidomide. These events are well known and managed with dose modifications in clinical practice. Serious adverse events were comparable between groups. As noted, given the number of hematologic adverse events, there were more dose modifications for PEPAXTO compared to pomalidomide, but with comparable adverse events leading to discontinuation. Deaths were also comparable between arms. Now here we are showing grade 3 and 4 adverse events of special interest. Thrombocytopenia and neutropenia occurred most frequently and at higher rates in the PEPAXTO group compared to pomalidomide. It is important to point out that these events can be quickly identified and are effectively managed, seldom resulting in clinical sequelae, such as concomitant bleeding or infection. Now let me show how the limitation of use to avoid treatment in the at-risk population further enhances the safety profile and reduces fatal events. Data will now be shown based on the recommended limitations of use, thus showing the recommended patient population. The safety risk and improved adverse event profile from the restriction of use is most clearly communicated when looking at the event rate based on time to progression following ASCT. As we increase the time to progression, we see that patients at least 36 months out from ASCT have the best profile and the least fatal adverse events. This supports the identified safety risk, as we see that fatal adverse events are significantly reduced. The first thing to note is that for PEPAXTO, the recommended patient population stays on therapy more than twice as long as the group with a time to progression less than 36 months after ASCT. The median treatment exposure is 8.1 compared to 3.7 months. As a consequence, one would expect more dose modifications from longer treatment. Instead, we see less adverse events leading to discontinuation and less fatal events. And here we are showing the same table again, now in events per patient year, that even more clearly demonstrates the safety benefit in the recommended population across all parameters. This also applies to deaths, where we see fewer in the recommended population. Importantly, we also see a reduction in deaths more than 30 days after last dose. Now let's discuss dosing. As PEPAXTO is a cytotoxic agent, using the maximum tolerated dose is the appropriate strategy to define the dose, and dose modifications are used to manage hematologic adverse events. 40 milligram is the recommended dose for most patients. However, to lessen the risk of thrombocytopenia, we proposed a 30 milligram starting dose in patients with a body weight of less than or equal to 60 kilograms. This was based on PK data and the occurrence of cytopenia. In addition, we've revised the dose modification guidance with earlier dose reductions. To conclude, the data confirms a favorable benefit risk for PEPAXTO when a limitation of use is applied to exclude patients with a time to progression of less than 36 months after ASCT. In this population, tolerability is improved with more patients remaining on therapy and less need for dose modification. We intend to clearly communicate the risk associated with PEPAXTO in patients with progression from transplant less than 36 months. We will exclude these patients from future use and clinical trials. Thank you. It's now my pleasure to hand the presentation to Dr. Efebera to share her clinical perspective.
Thank you so very much. I am Yvonne Efebera, Professor and Medical Director of the Blood and Marrow Transplant Program at Ohio Health. It is truly a pleasure to be with you today to share my clinical perspective on the importance of PEPAXTO in the late-line setting. I have been treating patients with relapsed refractory myeloma for almost 20 years, and our site was part of the PEPAXTO study program. My research entails participation in clinical trials in newly diagnosed multiple myeloma, relapsed myeloma, autologous and allotransplant, cellular therapy, and amyloidosis. Like Dr. Richardson, I am not being compensated for my time in today's meeting. To recap from Dr. Richardson's presentation, relapsed refractory myeloma is incurable, and patients with triple-class refractory disease continue to need options despite even recent approvals. Once a disease becomes multi-refractory, survival quickly diminishes as there become few successful or tolerated options for our patients. PEPAXTO's mechanism of action acts on a different pathway, allowing for continued response with improved outcomes compared to the Mammoth study, which has been widely referenced. And my own clinical experience with PEPAXTO supports the clinical trial findings. As some of you may know, myeloma is a disease of the elderly, with more than 80% of patients being 65 years or older. These older patients with relapsed refractory myeloma are perhaps most in need of novel agents. Let me provide a narrative of a patient who was in the Horizon trial and who reflects a very good example of the type of patient who continues to need PEPAXTO as an option. The patient is a 74-year-old Caucasian woman who was first diagnosed in 2005. Her disease progressed through 4 lines of therapy, and due to comorbidities, she was not eligible for transplant. She entered into Horizon and fared very well on PEPAXTO. She stayed on PEPAXTO for 2 years with a very good partial response before progression. She tolerated PEPAXTO very well with no hospitalization and experienced only expected hematologic adverse events. Her dose was reduced 2 times for moderate leukopenia and thrombocytopenia. The first dose happened 6 months after she started treatment, from 40 milligrams to 30 milligrams. She continued on 30 milligrams for another 6 months before it was dose reduced to 20 milligrams, and she stayed on that dose for another 12 months. Dose reduction was an effective management tool as she never reported bleeding or neutropenic fever, only mild to moderate fatigue, and was able to stay on treatment until her disease inevitably progressed. This is one of many examples of patients who needed PEPAXTO as an option, and I continue to feel comfortable prescribing PEPAXTO to the right patient population. It is worth noting that in the CARTITUDE-1 trial with cilta-cel, for which approval was obtained, the oldest patient was 68-year-old, and patients were required to have an ECOG of 0 to 1, a stricter criteria for even patients undergoing autologous transplant, and would definitely have excluded this patient and many patients included in the Horizon study. Additionally important and informative from the Horizon data and my experience with PEPAXTO is used in patients with extramedullary disease, EMD. As you know, EMD is associated with very poor patient outcomes, and yet it is not well studied, and patients are typically excluded from clinical trials despite the high unmet need. Horizon represents the largest cohort of patients with EMD evaluated to date in a prospective clinical trial and showed positive outcomes with 24% of the EMD patients experiencing response. Importantly, the safety profile for this subgroup is consistent with the overall population. Turning to the data from the OCEAN, from my review, the OCEAN study is an important trial in relapsed refractory myeloma in earlier relapse and adds to our previous understanding from the HORIZON study. The data in patients without stem cell transplant or having progression after 36 months from transplant is compelling and should be considered a key learning from OCEAN. There's clear biological rationale supporting this subgroup, and these are the patients with high unmet need in the clinical setting. These patients are typically elderly and frail and cannot always tolerate all treatment options. Patients without prior transplant were able to stay on drug longer and reported less thrombocytopenia on a drug that actually demonstrates favorable activity in a difficult-to-treat group of patients. The early PEPAXTO data is particularly reassuring since we observed this level of efficacy with limited non-hematologic toxicity associated with many other agents. Certainly, from my clinical perspective, the absence of alopecia, cardiac toxicity, and neuropathy that we see with other agents, this agent with the lack of these, and as well as only minimal mucositis and low rates of infection, are particularly valuable aspects to its use. Patients tolerate drug well, and adverse events were effectively managed with methods such as dose modification and supportive medications. This is apparent when looking at the comparable discontinuation rate across the program. When my patients see the disease responding to treatment after being on 3 or 4 lines of prior therapy, they do feel good about the regimen and they do not want to stop treatment. Additionally, the ease and infrequency of administration cannot be underscored. A monthly infusion is particularly important for this elderly patient population who have difficulty coming to the infusion centers. I have personally heard from my patients that this is a much added benefit of PEPAXTO. I want to conclude by stating that patients with relapsed refractory myeloma should have PEPAXTO as a late-line option. Our patients with triple-class refractory myeloma are in urgent medical need of salvage therapy with different mechanisms of action. These patients do not have many remaining options. Many have been treated with combination therapy at onset, and many cannot tolerate other treatments that have significant toxicities. Multiple studies demonstrate PEPAXTO's benefit in this setting. Importantly, that benefit is observed in the context of a consistent and manageable safety profile where patients are able to remain on therapy. When patients with a time to progression of less than 36 months after transplant are excluded, the remaining recommended population experience an improved safety profile in addition to meaningful progression-free survival. Importantly, these are often elderly patients who continue to need alternative treatment options the most. Our patients urgently need therapies, and I hope to be able to continue to provide my patients this additional line of effective therapy. Thank you so very much for your kind attention. I will return to Dr. Barker now.
This concludes our presentation.
Thank you to Oncopeptides A.B. and the team. We will now proceed with the FDA presentation from Dr. Alexandria Charson. Dr. Charson.
Thank you. Good afternoon. I am Alexandria Schwarzen, a hematologist oncologist in the Division of Hematologic Malignancies II at the FDA. I will be presenting the FDA's discussion on melphalan flufenamide, referred to as melflufen in the presentation. The members of the FDA review team are listed here. My presentation represents their collective input. The central issues we would like to focus on today are 1. The potential detriment in overall survival seen in the melflufen dexamethasone arm of the Phase 3 confirmatory trial OCEAN as compared to the control arm of pomalidomide dexamethasone. 2. The failure to demonstrate a progression-free survival benefit. And 3. The lack of an appropriate dose. To highlight where melflufen resides in the current treatment landscape of multiple myeloma, I would like to begin with an overview of the current treatment landscape. The table presented on the slide represents the treatment options for patients with relapsed refractory multiple myeloma. The treatment landscape has changed dramatically over the last decade, including multiple novel therapies approved since 2015. Highlighted in red are the 4 currently approved regimens indicated for patients who have been treated with 4 or more prior lines of therapy, including a proteasome inhibitor, immunomodulatory agent, and a CD38 monoclonal antibody. While many of the other agents listed can also be used for this highly pretreated population and repeated, those in red are specifically only approved for later lines. Selinexor, in combination with dexamethasone, was granted accelerated approval in July 2019. The clinical benefit of selinexor was subsequently confirmed in a Phase 3 randomized controlled trial, Boston, and selinexor's accelerated approval was converted to regular approval in December 2020. Belantamab mafodotin received accelerated approval in August 2020, and the confirmatory trial is currently underway. 2 CAR T-cell therapies have received regular approval: idacaptogene Belucel in 2021 and ciltacabtagene autoleucel in 2022. Also, not on the slide, melphalan is an oral drug approved for use in patients with multiple myeloma. Now I'll turn to the presentation today, which will focus on melflufen. Melflufen is a peptide conjugated alkylator with the same alkylating moiety as melphalan. Melflufen is passively distributed into cells, then enzymatically hydrolyzed to melphalan. Like melphalan and other nitrogen mustard drugs, DNA cross-linking is involved in the anti-tumor activity of melflufen. As you have heard previously, melflufen was granted accelerated approval on February 26, 2021 in combination with dexamethasone for patients with relapsed refractory multiple myeloma who have received at least 4 prior lines of therapy and whose disease is refractory to at least 1 proteasome inhibitor, 1 immunomodulatory drug, and 1 CD38-directed monoclonal antibody. A 40 milligram flat dose administered intravenously via a central venous line was approved based on the Phase 2 study. However, at the time of accelerated approval, there were residual uncertainties about the dose, and 2 post-marketing requirements related to dosing were issued, which I will discuss further in the upcoming slides. Efficacy of melflufen in combination with dexamethasone was evaluated in the Horizon trial, or OP106, a single-arm, open-label, Phase 2 multi-center trial. The accelerated approval was based on 97 patients with relapsed or refractory multiple myeloma who were treated with 4 or more lines of therapy and whose disease was refractory to at least 1 proteasome inhibitor, 1 immunomodulatory drug, and 1 CD38-directed monoclonal antibody, which I'll refer to as triple-class refractory disease, indicated as TCR on the slide. 70% of patients had received a prior autologous stem cell transplant. The major efficacy outcome was overall response rate and duration of response assessed by the International Myeloma Working Group response criteria. The overall response rate in the 97 triple-class refractory patients was 23.7%, with a median duration of response of 4.2 months. Myelosuppression was the major safety concern for melphalan. Approximately 75% of patients had severe neutropenia and thrombocytopenia. Nearly half of subjects had severe anemia. The melphalan USPI includes warnings and precautions for thrombocytopenia, neutropenia, and anemia. In addition, a warning and precaution was included in the label to alert prescribers of an increased risk of mortality with melphalan at dosages higher than the recommended dosage. This was based on results from a non-clinical safety study in dogs which examined melphalan against equimolar doses of melphalan. There was increased mortality in dogs receiving melphalan at dosages exceeding the recommended dose. Also, subsequent clinical studies did not investigate doses higher than 55 milligrams. Therefore, a limitation of use was added to the label, stating that melphalan should not be used as a conditioning regimen. At the time of accelerated approval, several post-marketing requirements were issued. Since accelerated approval was based upon a single-arm trial with an intermediate endpoint of overall response rate, continued approval was contingent upon verification and description of clinical benefit in a Phase 3 randomized trial. OP-103, henceforth referred to as the OCEAN trial, was the proposed confirmatory trial we will discuss in the upcoming slides. Due to the concerns of dosing, 2 post-marketing requirements were issued to further evaluate the acceptability of the fixed 40 milligram dose. In the next slide, I will briefly review the OCEAN trial. This slide shows the trial design for the OCEAN trial. OCEAN was a randomized, controlled, open-label, Phase 3 study of melphalan flufenamide and dexamethasone, henceforth referred to as the melphalan arm, compared to pomalidomide and dexamethasone, referred to as the pomalidomide arm, in patients with relapsed refractory multiple myeloma. The patient population enrolled had received 2 to 4 prior lines of therapy and had to be refractory to lenalidomide. The trial evaluated the same flat 40 milligram dose of melphalan with dexamethasone as the HORIZON trial. The OCEAN trial was conducted under a special protocol assessment, and the agreed upon primary endpoint was progression-free survival superiority assessed by an independent review committee. Key secondary endpoints included overall survival and overall response rate. Over the next few slides, I'll highlight the results from the OCEAN trial and the FDA-identified issues. This slide shows the baseline demographics on the OCEAN trial. As shown, baseline patient demographics and disease characteristics, including patients who had previous autologous transplant, and characteristics traditionally associated with poor prognosis, such as high-risk cytogenetics by FISH and ISS score 3, were balanced between the 2 arms. The first issue we will highlight today is the potential detriment in overall survival in the melphalan arm compared to the pomalidomide arm, and the safety issues that indicate a potential for harm with melphalan. This slide shows the Kaplan-Meier curve and the median overall survival for the 2 arms on the OCEAN trial. In the table on the left, you can see there were more deaths in the overall population in the melphalan arm, 47.6%, compared to the pomalidomide arm, a rate of 43.4%. Additionally, the median overall survival was approximately 5 months shorter in the melphalan arm compared to the pomalidomide arm, raising significant concerns regarding the safety of melphalan. This slide shows the updated overall survival results with a median follow-up duration of nearly 3 years. The median overall survival remains shorter in the melphalan arm, 20.2 months, compared to 24 months in the pomalidomide arm, and the hazard ratio remains unfavorable at 1.14, indicating a 14% increased risk of death. the FDA's presentation will highlight the safety issues that could have contributed to the potential survival detriment seen and indicate a safety risk with the use of melphalan. The sponsor has made several contentions regarding the overall survival results. This includes that detriment in overall survival is not indicative of a specific toxicity signal, that overall survival is driven primarily by results in the transplant subgroup, specifically in patients with a time to progression within 36 months of transplant, and that for pomalidomide and immunomodulatory drugs, there is an overall survival effect modification based on age. In the following slides, we will address the applicant's first contention, that the potential detriment in overall survival is not indicative of a specific safety signal. In analyzing the deaths in the OCEAN trial, there is an increase in deaths in the melphalan arm compared to the pomalidomide arm in the safety population, similar to that observed in the overall population. It is notable that the deaths beyond 60 days is increased on the melphalan arm. Although the reasons for the increase in deaths beyond 60 days is not clearly identified based on the narrative, there is concern that the increase in deaths beyond 60 days indicate that treatment with melphalan may impact the ability to receive and tolerate subsequent lines of therapy. The safety results from the OCEAN trial demonstrate a high rate of grade 3 to 4 treatment-emergent adverse events, dose reductions, and dose interruptions. Even with early dose modification, grades 3 to 4 adverse events remained higher in the melphalan arm compared to the pomalidomide arm, indicating concerns of the overall safety of melphalan in the relapsed refractory multiple myeloma patient population. This slide shows the most common treatment-emergent adverse events. It is important to note that over 80% of patients had grade 3 to 4 thrombocytopenia in the melphalan arm compared to only 14% on the pomalidomide arm. While the rates of grade 3 to 4 hemorrhage were relatively low, there were 3 fatal hemorrhagic events. As mentioned previously, myelosuppression was the major safety concern for melphalan identified in the single-arm trial Horizon. The lack of a comparator arm limited the assessment of the risk of myelosuppression on the overall benefit risk. Given the potentially worse overall survival noted in the randomized phase 3 trial OCEAN population, it is possible that myelosuppression is leading to prolonged marrow toxicity, which could have a lasting negative impact on the patient. I would like to remind you again of the overall survival results from the OCEAN trial, indicating a consistent result with a follow-up of 3 years. Overall survival is not only an indicator of efficacy, but also provides for an evaluation of safety. Given the toxicity data from the OCEAN trial, the potential detriment in overall survival is indicative of a significant safety concern with melphalan. We will review the limitations of the sponsor's post hoc analyses and reasons for continued concern in the upcoming slides. The sponsor has conducted multiple exploratory analyses and has concluded the overall survival finding is primarily explained by those patients who had received prior autologous stem cell transplant, especially those patients with a time to progression of less than 36 months. However, the FDA does not agree with this conclusion. First, I would like to briefly highlight a few caveats about subgroup analysis. The subgroups proposed by the sponsor were not prospectively included in the statistical analysis plan with control of type 1 error. Subgroup analyses are important. They can be used to confirm a consistent treatment effect across subgroups, thus providing greater assurance that the treatment effect observed applies to the entire patient population studied. However, results from one subgroup cannot be used to confirm a subset of patients who benefit when the total patient population has shown a detrimental treatment effect. As previously presented by Dr. Gormley, there are concerns with post-hoc subgroup analyses. As another example, the FDA analyzed the OCEAN data to illustrate how post-hoc subgroup analysis can lead to false conclusions. When subgroup analyses of overall survival are conducted by month of randomization, which is independent of the treatment effect, the resulted Kaplan-Meier plots are in opposite directions for patients randomized in March and July. On the left shows comparison of overall survival curves for patients randomized in March, and on the right is for patients randomized in July. We cannot conclude that the overall survival effect is different for patients randomized in March and July based on this post-hoc subgroup analysis. This has no scientific basis, but illustrates how false conclusions can be obtained by looking for differences in treatment effect in a post-hoc manner. This apparent difference in treatment effect is a random artifact of the data. It is not clear that other observed differences the sponsor proposes may be due to random chance alone. Prospectively defined hypotheses should be evaluated in prospectively designed studies to support a conclusion. However, acknowledging the limitations of subgroup analysis, we reviewed the applicant's analysis of time to progression from previous transplant. Before looking into the results of this subgroup further, I would like to point out the limitations of the definition. While the International Myeloma Working Group guidelines state that duration of remission after the first autologous stem cell transplant procedure is an important prognostic factor, this is in reference to the outcome for progression-free survival after a salvage transplantation. The IMWG guidelines also state the 3-year cutoff is arbitrary. Acknowledging the limitations of the definition, if we review the results of the subgroup of time to progression from previous transplant on the forest plot, we note that the confidence interval for patients without a previous transplant and patients with a time to progression greater than 36 months crosses 1, indicating a lack of difference between treatment groups. We cannot conclude a detriment is not occurring in these subgroups. While on the other hand, those patients with a time to progression less than 36 months from a previous transplant clearly fared worse with melphalan. Overall, the results from this subgroup are consistent with the results seen in the ITT population. Here, I would like to point out that the sponsor had previously proposed a different analysis for the transplant subgroup utilizing a different definition, time since transplant. The two definitions are shown on the slide. Time to progression, the current definition used by the sponsor, is defined as the time from the transplant date to progression date after the transplant. This is irrespective of treatment initiation with melphalan following progression. The previous definition used by the sponsor is time since transplant. The time since transplant is evaluated from transplant to randomization on the OCEAN trial and considers patients who receive melphalan at relapse. To illustrate the limitations with post-hoc analysis, FDA conducted an analysis based on the sponsor's previous definition, time since transplant. Utilizing the time since transplant definition, the previous definition proposed by the sponsor, and the cutoff of 36 months, the new time frame proposed by the sponsor, the results show that regardless of the time frame, less than or greater than 36 months, the hazard ratios are greater than 1, indicating potential harm. The upper limit of the confidence interval of the hazard ratio for the no transplant group is also greater than 1, indicating that potential harm cannot be ruled out. I would like to underscore that FDA does not endorse any conclusions based on post hoc analyses. The FDA conducted this subgroup analysis to highlight the limitations of post hoc analyses and making conclusions based on these analyses. By simply varying definitions or the cutoffs, different results are obtained. To further illustrate this point, this slide depicts the two subgroup analyses side by side. As stated earlier, the sample size and results differ based on the definition of the variable. In the subgroup analysis of time since transplant, previously proposed by the sponsor on the right, the hazard ratios, regardless of the less than 36 months or greater than 36 months, are greater than 1, indicating potential detriment and harm. Whereas in the analysis based on the current sponsor proposed definition of time to progression on the left, the group with a time to progression greater than 36 months has a hazard ratio less than 1. This illustrates the concerns regarding exploratory post hoc analyses. Additionally, even if we consider the results of the post hoc subgroup analysis, the detriment in overall survival is not limited to the time to progression from previous transplant less than 36 months alone. In the next slide, I will review the results from subgroups identified by the sponsor for further exploratory analysis. This slide shows the forest plot analysis for subgroups identified by the sponsor for further exploratory analysis. We want to highlight that the potential overall survival detriment seen in the ITT population was seen across multiple subgroups. Multiple subgroups showed a hazard ratio favoring the pomalidomide arm, and the majority of others include a confidence interval which crosses 1. Therefore, the overwhelming result is one that suggests a decreased overall survival with melphalan. Although the sponsor has highlighted the subgroup of patients who have not had previous autologous transplant as one which derives benefit from melphalan, we do not agree with this conclusion. It is important to note that these subgroups were not powered prospectively for individual analysis. They were only identified as exploratory analyses to be conducted and therefore can only be hypothesis generating. The sponsor also contends that for pomalidomide and immunomodulatory drugs, or IMiDs, there is an overall survival effect modification based on age, and the overall survival results from the OCEAN trial should be viewed in the context of this heterogeneity. The sponsor has concluded that there is an overall survival effect modification of age based on within treatment comparison of age groups in the OCEAN trial. The FDA does not agree. The OCEAN trial was not designed to compare or evaluate the effect of pomalidomide treatment in the various age subgroups evaluated by the sponsor. The subgroup analysis within the single treatment arm is not a valid approach to explore the modification of overall survival effect because it was not a randomized comparison and is unlikely to be balanced with respect to prognostic factors. The estimates provided from such comparisons would be influenced by many factors for which the investigator did not control. Additionally, analyses within treatment arm do not provide information on the treatment effect of the study drug. Even if this post hoc evaluation of age was valid, it does not rule out the potential detriment in overall survival observed in the ITT population and multiple subgroups in the OCEAN trial. The FDA reviewed the sponsor's exploratory post hoc model that was used to derive age and transplant as relevant factors. Although FDA does not endorse post hoc model building, FDA evaluated different post hoc models to further interrogate the variability in overall survival in the OCEAN study data. FDA only conducted this in order to evaluate if additional factors could explain the variability in overall survival. The results of the FDA analyses suggest that multiple factors, other than those suggested by the sponsor, can explain the variability in overall survival. However, the post-hoc models are unstable and the results rely heavily on the model being used. These exploratory analyses indicate that different model approaches yield different results. However, as all models under discussion, the FDA's and the sponsors were based on a post-hoc data-driven approach, the results may only be considered hypothesis generating and are not suitable for making conclusions. The sponsor also conducted additional post-hoc analyses to investigate the modification of overall survival effect by age in immunomodulatory drug trials and concluded that age interaction is also noted in other immunomodulatory drug trials. However, once again, there are several limitations. The age cutoff used for the sponsor's analysis is arbitrary and post-hoc. FDA conducted its own analysis of age interaction with treatment in immunomodulatory drug trials. This analysis was based on trial data submitted to the agency that isolated the treatment effect of an immunomodulatory drug. The FDA's exploratory analysis did not indicate that there was an interaction between age and immunomodulatory drug treatment. Even if this post-hoc evaluation of overall survival effect modification based on age is valid, these are exploratory analyses and does not negate the detriment in overall survival noted in the ITT population. In summary, in evaluating the overall survival effect modification, the findings of this exploratory analysis of heterogeneity in overall survival should be evaluated in a prospectively designed trial. The sponsor's claim based on exploratory post-hoc analyses do not address the finding of potential overall survival detriment in the ITT population. The available evidence from the OCEAN study does not provide evidence that melphalan is safe and effective. In summary, patients treated with melphalan had a potential detriment in overall survival indicating a potential for harm. There are safety concerns with an increase in the number of deaths and toxicity on the melphalan arm suggesting a potential for harm. The FDA does not agree with the sponsor's conclusion that the overall survival detriment is limited to those patients with a time to progression less than 36 months, as the detriment in overall survival was seen across multiple subgroups. Post-hoc subgroup analyses, while hypothesis generating, should not be used as confirmatory evidence to conclude a benefit of a treatment effect or lack of harm. The second major issue to highlight today is the failure to demonstrate a progression-free survival benefit. We do not agree with the sponsor's conclusion on the progression-free survival benefit from the OCEAN trial. The primary progression-free survival results from the OCEAN trial did not meet the pre-specified statistical superiority, and the potential detriment in overall survival negates any observed progression-free survival improvement. This slide shows the original progression-free survival results submitted to the FDA following database lock. The applicant's original primary analysis of progression-free survival results submitted on June 9, 2021 showed that the OCEAN trial failed the primary endpoint with a p-value of 0.0644. While the median progression-free survival in the melphalan arm was 2 months longer than the pomalidomide arm, the results were not statistically significant. On July 6, 2021, the applicant submitted revised progression-free survival results. The applicant noted that these revisions were due to discrepancies in 29 patients identified by an independent audit. The independent audit was initiated by the applicant following the database lock and the top-line data readout on May 25, 2021. This slide shows the original progression-free survival Kaplan-Meier curve on the left compared to the revised progression-free survival results on the right. The progression-free survival from this revised data demonstrated nominally significant superiority with a p-value of 0.0322. FDA conducted their own analysis on the revised progression-free survival results. As the original primary analysis of progression-free survival results were not significant, all reported p-values, except for the original primary analysis result, are considered nominal and not suitable for inferring statistical significance. FDA's assessment of the revised 29 patients confirmed the nominally significant p-value. However, the FDA obtained a p-value that was different from the applicant's due to a difference in FDA adjudication of 4 patients. When using FDA censoring rules, which censors unconfirmed progressive disease, progression-free survival analysis resulted in a p-value of 0.0837, indicating that there was no difference between the treatment arms. Regardless of the method used for progression-free survival analysis and the significance of the p-value, the difference in median progression-free survival between the arms remained approximately 2 months or less. Additionally, the variability of the results indicate a lack of robust treatment effect of progression-free survival. Importantly, given the detriment in overall survival observed, any difference in progression-free survival, whether statistically significant, clinically significant, or not, would not support a determination of clinical benefit. Finally, this table presents the overall response rate and duration of response from the OCEAN trial. While there was a 5.6% median difference in overall response rate favoring the melphalan arm, the 95% confidence interval crosses 0, and thus no meaningful difference is apparent. Additionally, there was no difference in duration of response. In conclusion, the sponsor concluded that progression-free survival results are significant. However, FDA does not agree. Regardless of the p-value, there is only a 2-month difference in progression-free survival. As a reminder, there is no difference in overall response rate and duration of response. Importantly, a potential detriment in overall survival is noted. Overall survival is the ultimate clinical benefit endpoint. Any marginal improvement in progression-free survival is negated by the potential detriment in overall survival seen in the OCEAN trial. The results from the OCEAN trial indicate a lack of a confirmed benefit for melphalan and a potential for harm. In the next few slides, we will show that this lack of benefit and increase in toxicity is likely due to lack of a dose that has been optimized for a favorable benefit-risk profile. Another major issue is the lack of an appropriate dose. The applicant's proposed 40 mg melphalan dose is poorly tolerated, and there was limited dose exploration of lower doses in the clinical program of melphalan. There are significant safety concerns with high melphalan exposure, which are not resolved with the applicant's melphalan dosing proposals. The 40 mg dose was identified to be the maximum tolerated dose in the phase 1-2 dose escalation study and was the only dose used in subsequent clinical studies. As shown in the table, the lower doses were not fully explored as only 4 patients treated with 15 mg and 7 patients treated with 25 milligrams had safety and efficacy data. Pharmacokinetic data in the dose escalation study was derived only from 12 patients, and 8 of these patients received the 40 milligram dose. Further, no pharmacokinetic data was collected in the pivotal phase 2 study. Consequently, no population pharmacokinetic or exposure response analyses were conducted to aid in dose selection to support the proposed 40 milligram flat dose before the phase 3 OCEAN study. A post-marketing requirement was issued for an exposure response analysis to aid dose selection, but this analysis was limited because all of the exposure response data from the OCEAN study was derived from patients treated with 40 milligrams. An optimal dose remains unestablished. Exposure response analyses indicate that there are rates of several safety events increased with higher exposure following melphalan administration. Higher exposure was associated with increased risks of grade 3+ anemia, any grade 3+ treatment emergent adverse event, grade 3+ leukopenia, and treatment emergent adverse events leading to melphalan discontinuation, dose interruption, and dose reduction. Furthermore, no relationship between melphalan exposure and overall survival or progression-free survival have been identified. The extensive dose modifications in OCEAN suggest that the flat 40 milligram dose is poorly tolerated. This figure shows the percentage of patients who received melphalan dose per cycle. All patients initially started with 40 milligrams in cycle 1. However, by cycle 7, more than half of the remaining patients needed dose reductions of melphalan. With successive cycles, progressively more patients required 1 or more dose reductions. By cycle 12, the 20 milligram dose was the most commonly administered dose. The large proportion of patients needing doses lower than 40 milligrams illustrates that the 40 milligram dose was poorly tolerated. This slide depicts 2 issues with the melphalan 40 milligram dosing strategy contributing to poor tolerability. As you can see in the figure, patients with lower body weight had higher exposures. A similar relationship between body surface area and exposure was observed. As previously shown in the exposure-response safety relationships, higher exposure is associated with increased risk of various safety events. Because the body size metrics of body weight and body surface area were significantly associated with melphalan exposure, the flat dosing with melphalan exacerbates the variability of the exposure, even though there is still high pharmacokinetic variability at all body sizes. Therefore, dosing based on body size would decrease some variability in exposure and may reduce the overall risk of safety events. These figures depict another representation of the higher exposure at lower body weights just described on the previous slide. The upper panel displays exposure following 40 milligram flat dosing in all patients, with exposure in patients weighing 60 kilograms or less in the pink icon. The lower panel shows the predicted exposure with the sponsor's proposed dosing. The sponsor is proposing a reduced starting dose of 30 milligrams in patients who weigh 60 kilograms or less in order to match the exposure in patients weighing 60 to 90 kilograms in green. As you can see, the exposure in patients 60 kilograms or less in the pink icon is reduced to more closely match exposure in patients weighing 60 to 90 kilograms. However, the 40 milligram dose was poorly tolerated in the overall population and at all body weights in OCEAN, not just patients weighing 60 kilograms or less. So the exposure matching proposal is not adequate because it matches an exposure associated with significant safety concerns. This conclusion is further underscored by the observation that patients across all body weights required doses lower than 40 milligrams in the phase 3 study, as seen in the doses administered over time according to weight category. The top right figure shows poor tolerance of 40 milligrams in patients weighing 60 kilograms or less. Similarly, the bottom figure shows that 40 milligrams was also poorly tolerated in the average weight subgroup, and so the sponsors proposed exposure matching strategy in patients weighing 60 kilograms or less would not resolve the tolerability issues in those patients. Further, the sponsor is still proposing a 40 milligram starting dose in patients greater than 60 kilograms, and so the FDA's safety concerns with the overall population have not been addressed. In review, the limited data from the phase 1 and 2 studies raised concerns regarding the optimal dosing of melphalan and prompted post-marketing requirements for optimal dosing. In the phase 3 OCEAN study, there were high rates of treatment-emergent adverse events leading to dose reductions and dose interruptions and other safety events which signal that the 40 milligram dose is poorly tolerated for the general patient population. Multiple safety events were associated with higher exposure, and higher exposure is not associated with better efficacy. The proposed 30 milligram dose for patients with less than 60 kilograms is inadequate because it matches the exposure of the 40 milligrams, which is considered too toxic. The FDA analysis also indicates that dosing by body size or weight may reduce variability and may be more appropriate for melphalan. Additional exploration of the dose and body size-based dosing for melphalan is warranted. Over the previous slides, we have reviewed the major issues which suggest an unfavorable benefit-risk profile in light of the potential overall survival detriment, lack of progression-free survival benefit, and lack of an appropriate dose. Next, we will highlight some additional uncertainties in the clinical benefit of melphalan that arise from interpreting the results to the currently indicated patient population and inadequate representation of the U.S. multiple myeloma population. Interpreting the results of the OCEAN trial to the currently indicated patient population indicates that the potential for harm exists for the currently indicated population as well. A total number of 30 patients, or 6% of patients from OCEAN, are consistent with the current indication of having both 4 prior lines of treatment and having triple-class refractory disease. This slide focuses on the overall survival forest plot results for the patients that fall under the currently indicated patient population. While we cannot make definitive conclusions from this subgroup analysis, it is concerning that the overall survival hazard ratio favors treatment with pomalidomide in patients who are triple-class refractory, have had 4 prior lines, and those patients with both, consistent with results seen in the overall ITT population. This raises serious concerns about the safety of melphalan in the currently indicated population. Adding to the uncertainty is the applicability of the study results to the U.S. patient population. The OCEAN study had a low representation of the typical U.S. multiple myeloma population. Only 15% of patients on the OCEAN trial were 75 years of age or older, compared to 32% of patients 75 years of age or older diagnosed with multiple myeloma in the U.S. The OCEAN trial also enrolled a very low percentage of U.S. racial and ethnic minorities, and most patients were enrolled outside of the U.S. Finally, only 18% of patients had previous treatment with an anti-CD38 monoclonal antibody. In the U.S. today,
most patients would have received an anti-CD38 monoclonal antibody in the first 2 treatment regimens. Now, I would like to summarize FDA's conclusion on the overall benefit-risk assessment of melphalan. The available evidence suggests an unfavorable benefit-risk of melphalan in the currently indicated patient population. Specifically, the overall survival results from the randomized confirmatory trial OCEAN not only show a lack of efficacy, but also indicate a potential safety concern. The progression-free survival results indicate lack of a confirmed clinical benefit. Additionally, the flat 40 milligram dose is poorly tolerated in the general patient population. Further studies are needed to identify an adequate dose. FDA's analysis indicates body weight dosing may be needed. Given what is currently known, we would not have granted accelerated approval, as we cannot conclude melphalan provides a meaningful benefit over available therapies. In today's treatment landscape for myeloma, where multiple therapies exist, and the overall survival benefit for patients comes from their ability to receive and tolerate a sequence of therapies, the risk of melphalan appears to outweigh any potential benefit. Further studies are required to establish the benefit-risk of melphalan flufenamide. We'd like for the committee to discuss the benefit-risk profile for melphalan for the currently indicated patient population, considering the results of the confirmatory OCEAN trial. The voting question to the advisory committee is, given the potential detriment in overall survival, failure to demonstrate a progression-free survival benefit, and lack of an appropriate dose, is the benefit-risk profile of melphalan flufenamide favorable for the currently indicated patient population? This concludes my presentation. Thank you for your attention.
Thank you, Dr. Charlson. We will now take clarifying questions for the presenters, Oncopeptides A.B. and the FDA. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and end of your follow-up question with that is all for my questions, so we can move on to the next panel member. Perhaps I can go ahead and start. Clearly to me, and thank you for both the FDA and Oncopeptides for great presentations. I mean, clearly there's a big difference in what I have heard today. Clearly one is, no PFS improvement, and the applicant states that there is a PFS improvement. And I want to get a bit more understanding from the applicant related to their review PFS that was resubmitted to the FDA after the initial PFS submission. And specifically, if the applicant can expand as to how they censor their patients, and equally important, what exactly happened to those 29 patients which appear to be in question, and the reason why your PFS appears to be different compared to the initial PFS report. Thank you.
Absolutely. Klaus Brocker here for Oncopeptides. So the data cutoff of the study was the 3rd of February 2021, and there was data cleaning ongoing until the 7th of May. The final IRC meeting took place on the 19th of April. That was before the end of the data cleaning. That, of course, should not have happened, because the IRC should have the final data after all the data has been cleaned. This is an operational oversight that should not have happened. However, the CRO noticed the 29 patients where changes had been made during the data cleaning process and actually then asked the IRC to not reanalyze these patients, but to look at the final data for these patients. The IRC has been blinded throughout the full process. The sponsor had no involvement with the response assessment, and the 29 patients were provided by the IRC. So the sponsor can only assert that there was a statistically significant primary endpoint here with a superior PFS, and more importantly, the IRC also agreed with this assessment.
Thank you. Dr. Waldman, do you have a question or a comment?
Yes, I do.
Before, can the FDA respond to that comment, please?
Sure, go ahead.
Yeah, great. Hi, this is Nicole Gormley. So, I just, want to clarify from our perspective, the FDA received the top-line results, which did not demonstrate statistical significance. We shared our concerns with the agent, with the sponsor, and then, the sponsor said that provided additional information, as they described just previously, but submitted new information then stating that it met statistical significance. I think there are several concerns that we have with the progression-free survival, in addition to, the reassessment, which is highly unusual. I'll just leave it at that. But also concerns with the censoring rules that were used, and then also, our own readjudication, we had discrepancy with 4 patients. Regardless, though, of the methods used or the timing or the original analysis or the reassessment, from the FDA perspective, this still represents a very, very small, relatively difference in PFS of only 2 months. So I just wanted to add those comments. Thank you.
Can the sponsor give a reaction to that comment?
Please go ahead. If you can be succinct and precise, that would be great. Thank you.
Absolutely. Regarding the censoring rules, the sponsor used the censoring rules as was stated in the pre-specified statistical analysis plan, which meant that for a progressive disease event, the second assessment was necessary unless there was a subsequent initiation of treatment because the patient progressed too fast, prohibiting a second confirmation, or when there was progression of extramedullary disease, which also precludes a secondary measurement. This is according to IMW guidelines, and what you see here is the letter from the chair from the IRC stating these points very clearly from their perspective, and this is how it states in the pre-specified statistical analysis plan. So from the sponsor's perspective, there can be no misunderstanding whether the primary endpoint was met or not.
Thank you for that. Dr. Waldman?
Yeah, so Scott Waldman, Thomas Jefferson University. I'm just going to perpetuate this discussion. So listening to the discussion and reading all of the materials, there seems to be, and again, I'm going to amplify what Dr. Garcia said. There seems to be an incompatibility with the data interpretation from the sponsor and the data interpretation from the agency. That is, progression-free survival, at least by the agency standards, doesn't look like it was met by pre-specified standards, number one, and number two, in terms of overall survival, post hoc analysis is not sufficient to establish the hypothesis. So I guess where I'm going with this is I need to hear discussion about how we either bring these things to compatibility or are we entrenched in incompatibility. I'm sorry, it's confusing. I'll take any response.
This is Nicole Gormley. I wasn't sure. Was that a question specifically to the FDA, or were you asking the sponsor?
I'm actually asking both, and I apologize for maybe a little bit of nebulous question, but There's clearly incompatibility in your positions. And so, yes, I'm asking the FDA, and I'm asking the sponsor to respond.
Okay, so this is Nicole Gormley again, FDA, and I'll start and then allow others to chime in, and then we can turn it over to the sponsor. So please, just to be clear, the FDA's position, we don't agree that a PFS statistical significance was demonstrated. But please note, even if we did, we would have significant concerns and would not be able to, this trial would not provide demonstration of safety and effectiveness because of the overall survival results. So we've had multiple instances throughout oncology and unfortunately particularly in multiple myeloma where we've seen discordance in between progression-free survival and overall survival. Overall survival is the paramount endpoint that is needed for determination of clinical benefit. When we have a primary endpoint of progression-free survival, we still require data from overall survival to ensure that it is favorable and that there's not a potential for harm. Unlike some other endpoints, overall survival in particular is really both a safety and an efficacy endpoint in that it incorporates information about toxicity and allows us really a better understanding of the overall clinical benefit. So we can have discussions about whether or not the PFS endpoint met statistical significance or not, but please note that that's not the most germane issue for us. The most germane issue is that the data suggest potential for worse overall survival.
This is Dr. Pazdur. Could I just jump in on this? May I?
Sure, go ahead, Dr. Pazdur.
I would just like to emphasize it is the sponsor's requirement to demonstrate a safety and efficacy, and that efficacy should be demonstrated by substantial evidence, okay, not by post hoc analyses. It is not the responsibility of the FDA to demonstrate that the drug doesn't work or is unsafe. It is incumbent upon the sponsor to provide substantial evidence here. And here again, when you have people resubmitting data and changing analysis, this could bring up issues of study conduct and integrity of the study also, which needs to be addressed. So there's many factors here, but I just want to emphasize they have to demonstrate with substantial evidence, okay, efficacy. Okay, it is not our responsibility to disprove something, and this is the central question here, is have they demonstrated with substantial evidence the safety and efficacy of this drug? It's their responsibility. Thank you, Dr. Pazdur. Perhaps just to wrap up Dr. Waldman's question, maybe the sponsor of the applicant can also have a few minutes to address Dr. Waldman's question.
Absolutely. Thank you. Klaus Barker here from the sponsor. First, I would like to state about saying about multiple analyses that have been provided to the FDA. The transplant has always been, straight from the beginning, the only interaction that the sponsor submitted to the FDA as the interaction that mattered for PEPAXTO. The interaction was based on a biological rationale and overall endpoints that moved with it. It's very important to state that there were no reanalysis with regarding to that interaction. However, what is very important, and this comes back to the IMiD interaction that the FDA says doesn't play a role. What we see here is OCEAN on the left with an ITT PFS hazard ratio of 0.79. An ITT overall survival hazard ratio of 1.14. And one sees the clear split by age. Earlier this year, albeit a phase 2 study, we saw a well-known proteasome inhibitor against pomalidomide with the same trend. The request from the sponsor, where Dr. Gormley alluded to potential postponing of this ODAC, was not to postpone in it itself. It is because we know that DREAMM-3 is imminently reading out. And we are just very curious what the ITT overall survival hazard ratio will be from that study. And given that that analysis is coming imminently, we think that the panel would have benefited from seeing also that data. That was the reason for the sponsor to request the FDA to at least wait until this important data became available. Then one final comment. The FDA mentioned that their own exploratory analysis basically refuted the age-IMiD interaction. But I would only like to mention that from the 5 studies that were mentioned, 3 studies were actually, 2 out of 3 were crossover studies. There cannot be any pomalidomide isolation, and 1 study compared to a high-dose dex. When it comes to the conduct of the study, because I hear that was also in question, there have been audits all the time. During the study, there has been no findings, and this is very important. Additionally, and I think this is important to conclude with, the sponsor is willing to corroborate the findings in a new study. The sponsor is willing to look into a new study, excluding patients at risk, because we only used subgroups to identify a patient at risk, not to identify patients with benefit. And it is very important to state that we are willing to do a new prospective study in the recommended population. With that, I would like to give it back to the chair.
Thank you for that. Let's move on to Dr. Fredlin. Do you have any questions or comments?
Yes, Boris Friedlin. I have a question to the sponsor. So you presented analysis that argues for heterogeneity of OS treatment effect, and indeed the analysis showed that some subgroups have detriment in overall survival, for example, in younger patients, in women, in patients with more than 2 lines of therapy. And then it is proposed that by excluding patients who progressed less than 3 years after transplant, the indicated population will avoid OS detriment. How can we be sure that these rules reliably excludes population with potential harm? For example, consider a hypothetical 64-year-old patient who relapsed 40 months after transplant. According to your slide 44, OS hazard ratio for this patient could be as high as 2, indicating potential harm. How do we know treatment is safe for a patient like this? Thank you.
Thank you, Klaus Bakker here from the sponsor. The slide that is currently up shows the subgroups after the exclusion of the group at risk. That is, the patients who progressed less than 36 months after their transplant. And what you basically see is that all the point estimates move to the left, excluding any real potential risk. There is still an age relation visible, but that is due to the pomalidomide interaction with age. If we would, however, look at medians, the median for PEPAXTO in the patients less than 65 years of age is 35 months. For pomalidomide, it's 31.5 months. So this gives the sponsor the reassurance that with all the point estimates moving to the left, basically taking away all the other FDA-identified heterogeneous subgroups, gives us confidence that this population at risk and excluding it is sufficient to conduct further studies with this drug.
This is Nicole Gormley at FDA. Could we leave that slide up? I'd like to respond to that comment, if that's okay.
Please go ahead, Dr. Gormley.
Great. So, as you noticed here on the slide, and I think, Dr. Friedland, you bring up a great, comment. Most of these confidence intervals cross 1. And so, there is not confidence that, that just limiting the population to what they've pre-specified would not cause harm. I just also, like to point out, from a regulatory perspective, we do not use subgroup analyses to carve out indications. And we would wholeheartedly endorse the sponsor's proposal to conduct a prospective randomized trial in the population that they deem, would not experience harm, but that should be done before an indication is granted. Further information is needed to have confidence that we would not be causing harm to patients. Thank you.
Thank you.
Thank you for the answer.
Dr. Nowakowski?
Hi, Greg Nowakowski. A question to the sponsor. So with all the uncertainty regarding PFS and overall survival, which could indicate potential harm for melphalan, I'm trying to look at other time-dependent endpoints here. And your slide CO31 shows the median duration of response is essentially identical in both arms. How do you reconcile this median duration of response? That's number one question. And then related to it, do you have any other time-dependent endpoints like time to next therapy per arm?
Yes, I will ask in the meantime to bring up the time to subsequent therapy. The time to subsequent therapy was marginally longer for the pomalidomide arm, so patients on PEPAXTO were somewhat earlier able to start their new treatment. With regards to the secondary endpoints, as shown here, we see a consistent benefit of melphalan or PEPAXTO over pomalidomide. I would just like to respond to Dr. Gormley, who I thank for the outreach to conduct a new clinical study in the recommended population. And I would like to state that it has never has been the sponsor's intent to carve out a subgroup of benefit, as the FDA suggests. We have been used guidelines to identify a patient group at risk. And that is consistent with when you meet the primary endpoint of PFS, that you have the obligation to look at subgroups. And of course, there are subgroups where the confidence interval go above 1. I think there is no single study in oncology where you have all the subgroups with the full confidence intervals below 1. So I just think that's the important point.
Thank you. Dr. Crawford?
Because I just wanted to summarize response because there was additional comment from the sponsor, which I just want to make sure that I'm clear. So the median duration of response was no different as you showed, and then you mentioned that the time to next therapy was actually favoring the standard arm in the study, pomalidomide arm.
Mm-hmm.
That's correct?
Yes, sorry for the confusion. So it was favorable for PEPAXTO, the time to subsequent therapy. I just want to be clear there. That was shorter than for pomalidomide.
Oh, sure. Okay. And then, in terms of the overall survival, which we had a discussion here, one of the potentials for harm is then delayed toxicity, which then results in inability of patients to receive effective additional lines of therapy, primarily due to cytopenias or other residual delayed toxicity. Do you have any data about what treatments patients received after progression, and how many were able to receive the treatment? And maybe you were able to capture PFS too with those treatments subsequently?
Yes, we do have those data, and I hope you can see the slide that I see in front of me. So a similar percentage of patients was able to receive subsequent therapy. In fact, a somewhat higher percentage of patients on the PEPAXTO arm. And there are two main differences between both drugs here. So what is very clear here is that more patients on the pomalidomide arm were able to receive daratumumab following progression as a next line of therapy, whereas for PEPAXTO patients that progressed, a significant number of patients received pomalidomide. Now, we know from a study by Dr. Richardson, the Tourmaline trial, that the earlier daratumumab is used, the more favorable the outcomes are. And of course, this is hypothesis generating, but it is clear that potentially this is impacting the pomalidomide arm favorably because of the earlier use of daratumumab.
Okay. And any data on PFS too?
Sorry, no, we did not capture PFS2 as a formal endpoint.
Okay, thank you.
Thank you. Dr. Crawford?
Thank you, Mr. Chair. This is Stephanie Crawford. Certainly I appreciate the presentations both from the sponsor and the agency. We have heard that exploratory post-hoc analyses are not used in regulatory decisions, notwithstanding because so much in both sets of presentations was on post-hoc analysis. I ask a clarifying question to the sponsor. I invite the sponsor's response to issues raised by FDA regarding inflated type 1 error without adequate statistical adjustments to control for the multiple comparisons in the post-hoc analyses performed.
Thank you for the question. So if I can ask to pull up slide CO5, please. So regardless of whether we pre-specify multiplicity for studies, it's important that a study meets its primary endpoint. And when, according to the statistical analysis plan, the primary endpoint is met, and that was it according to the statistical analysis plan, we are obliged to look at subgroups, regardless of multiplicity adjustments. But because of the risk of a random finding, as illustrated by the FDA by the patients randomized in March versus July, the subgroup should be substantial, should be supported by a biological rationale, precedent, and other supportive endpoints. And this is actually what holds true for the transplant group. So while not formally pre-specified in the statistical analysis plan, I need to say that it was a pre-specified subgroup.
Thank you. Thank you.
Thank you.
Just to clarify, were statistical adjustments made in the post-hoc analyses, and if so, what were they?
I will ask our statistician, Mark Stuarson, to answer that question.
Hi, this is Marcus Stuarson, a statistician at Oncopeptides. Well, while we are not making any formal claim on the subgroups in terms of statistical significance, we believe that we had a statistical significance on the overall PFS results, and the evaluation of subgroups revealed that we cannot really say that there is a homogeneity across subgroups. So it's more, as we discussed before, a way to sort out a subgroup of risk rather than identifying a subgroup of where we have efficacy. So there's no need for adjustment for multiplicity.
Thank you for the response.
So this is Dr. Gormley at the FDA. Could we just comment again? So, I just would like to underscore, and perhaps, we could pull up FDA slide 80. So, just to, and I think, I think the committee understands, what I've mentioned earlier is that, we do not grant indications based on, subgroup post-hoc exploratory analyses. And while the sponsor's claiming that, they're not carving out the population by adding a limitation of use, that's essentially what they're doing, and that's what I'm referring to. So, if we could have slide, FDA slide 80, just to underscore, perhaps this is not the correct one. So just to, underscore, the statistical analysis plan outlined transplant, yes or no, as a outlined prior transplant, yes or no, as an exploratory analysis. There was no type 1 error control, thank you, for this analysis. So as you can see here, this is the statistical analysis plan from 2021. It was listed as an exploratory analysis, and it was only prior transplant, yes, no. There was no mention of time to progression or 36 months. So, from our standpoint, this is a really concerning analysis and one that we cannot use to confirm clinical benefit. So I'll pause there, and I don't know if any of other colleagues wanted to answer from the FDA.
I would like
this
just
is Dr. Wise, the deputy or the director of the Biometrics 9. Yeah, I'd just like to add that these subgroups are standard to look for heterogeneity in the treatment effect. They're not for testing or reducing the population or selecting the population.
Klaus Bakker for the sponsor here. If I may give a quick reaction, we of course agree that the only pre-specified subgroup was prior autologous stem cell transplant. But since there is such a clear biological rationale, it merits further investigation, such a finding. And I would like just to ask Dr. Richardson if he would be willing to comment on the 36-month window that was put into place.
Thank you very much, Dr. Bakker. No, I think the timeline is actually clinically very important to understand, because what we know is that in the past, when patients relapsed within 1 year of a transplant, the pathobiology of the disease was particularly poor. With the advent of maintenance, and the backbone of maintenance now, of course, is lenalidomide, that extended to 2 and then 3 years. So I think this 3-year or 1, 2, and then 3-year parameter that's been built has a strong clinical rationale, and I certainly would support the pathobiological argument behind that timeline.
This is Dr. Nicole Gormley. I'd like to just advance the slide. So as you see here, in a subsequent analysis, again, after we had top-line data, an adjustment to the SAP added additional time points that the sponsor planned to look at. Less than 2.5 years, 2.5 to 5, less than 5, more than 5, no transplant. And 36 months is perhaps inclusive of the second bullet, but from the FDA perspective, this really just underscores sort of data dredging, if I could say that. So thanks so much.
Thank you, Dr. Gormley. Move with Dr. Sakaris.
Thank you so much, Dr. Garcia. Reflecting back on some of the concerns about the differences in interpretation of progression-free survival, maybe I'm an outlier, but that actually doesn't bother me that much. I think when you're playing a little bit on one side of statistical significance or a little bit on another side of statistical significance, you're talking about a progression-free survival that probably isn't that great. And as has been shown previously, actually here at ODAC, progression-free survival can sometimes be in the eyes of the beholder or the eyes of the time of assessment. So that doesn't really bug me as much. I kind of anticipate there's going to be some debate about assessment of progression-free survival. What does bug me a bunch, however, is an approximate 4-month worsening of overall survival for patients who got the study treatment. And I was wondering if the sponsor could provide some information on the duration of serious grade 3 or 4 thrombocytopenia and neutropenia for patients who received the MELDEX.
So, well, I asked the team to look if we have a slide specifically looking at the time of the thrombocytopenias and neutropenias. There's one comment that the sponsor would like to make here, and that is if I can ask to pull up the slide that was previously shown by the FDA, and I believe it was their slide 44, if I was right. And in the meantime, we will create the slide, of course. Let me see if the slide is coming up. Right. So I'm coming to the duration of grade 3/4. This is the slide that the FDA showed about the flat 40 milligram dose that is poorly tolerated. What this basically shows, this graph, the sponsor could have also used to show actually that one needs a 40 milligram dose, because after one cycle, 85% of patients is still needing the 40 milligram dose. There's no need for dose reductions early. It is gradually decreasing the use of 40 milligram, which is standard if one uses a cytotoxic to get a disease under control. And then when the patient fares well, you can often go with a somewhat lower dose. So I think from a dosing perspective, I think to comment just on one of the other major disagreements that the FDA and the sponsor have, is that the 40 milligram flat dose actually is the right dose if we look at this graph. And I would just like Dr. Richardson maybe to comment on that.
Well, thank you, Dr. Bakker. I think Dr. Scaris, the critical question about your thrombocytopenia duration, I think is important to address, and it's coming. I think it's addressed also by the fact that this was primarily seen in the peri-transplant population in terms of tolerability issues. But going back to this 40 milligram dose, clearly dose optimization and so on is an ongoing process, but we established in the phase 1/2 study a clear dose response effect and also at the same time tolerability parameters that we felt comfortable with in terms of exposure and recognizing the novel mechanism of a peptide drug conjugate here and therefore the exposure that was required for efficacy. I think as we think about the dose reduction strategies here, it's important to share that in myeloma, obviously, we routinely dose decrease or dose adjust with pretty much all of our therapies according to tolerance. So I think that's an important other point to share. But in terms of the impact of tolerability and toxicity on that 4-month survival difference, I think, Klaus, that is very important to address. I think you can do that, can't you, from what you want to bring up next to help Dr. Sakaris understand that better.
Absolutely, and I now have a slide available where I can talk better to that. And we will come back with the timing also specifically. But what we do want to point out, by carving out the subgroup at risk, which is common practice, we also take away this concern. Because, of course, we understand the potential concern by putting a patient on another alkylator shortly after they have failed basically high-dose melphalan therapy is not the right thing to do. And basically what we see here is this time-dependent increase actually or decrease of adverse events when looking at the recommended patient population. The number of grade 3-4 adverse events goes down when we are past that 36 months threshold, but especially the need for dose modifications and also the discontinuations go down. So it's not only from an efficacy perspective, but also from a safety perspective that applying the 36 months threshold and taking out that population at risk, we really feel that we basically take away the risk from longer deprived bone marrow function. And I will come back after the pause with a specific slide on the duration of grade 4 thrombocytopenias to answer that question specifically.
Hi.
That's a very good question, Dr. Sakaris.
Could the FDA respond to that comment? Sorry.
Yeah, go ahead. Then I'd love to actually get an answer to the question I asked as opposed to the slide that the sponsor wanted to present.
You go first, and then we'll share some data. Please go ahead.
Can the sponsor, Dr. Sakaris, perhaps you can actually repeat your question so the sponsor can, you can clarify what you're really asking instead of the slide as you indicated.
Yeah, no, I know it's been a while since I asked the question, so I'm happy to repeat it if they've forgotten. So all I'm asking is what was the duration of grade 3 or 4 neutropenia and thrombocytopenia in your population? We're trying to get at cause of death here for the excess death rate that was seen on MELDEX. So let's start with duration of grade 3 or 4 neutropenia or thrombocytopenia in the entire population.
I'm just hearing that we have it in the TLF, so we'll try to get a slide for you together to show after the break, specifically answering your question.
Okay, so I can move on since you don't have that slide. Okay, can you discuss the cause of death, particularly among the excess patients who died on MELDEX?
Absolutely. So the majority is progressive disease. That is the only identified cause of death. And also all death narratives have been shared with the agency, and I think together with the agency we conclude that there was no specific toxicity signal there. And I will just pull this one up. That's within 60 days after first dose. We see the primary causes of death indeed being progressive disease. And within 60 days, also adverse event, 3% versus 1%. And now I need to look at the slide that I have in front of me. I'm going to put up a slide and try to answer your question succinctly. Here we see the fatal AEs by the various groups. On the left, we have the Horizon study that supported the accelerated approval. And of course, we see more adverse events there because of the late stage of the disease. But we see on the OCEAN study, we see that the number of patients with at least one fatal AE, that it doesn't seem to be that there is a difference there between PEPAXTO and pomalidomide with 12 versus 13%. And if we then look at the type of fatal events, we also don't see a difference there. Specifically, if we look at infections and infestations, it's basically balanced between the groups. So there's no real clear toxicity signal that we could identify in this study.
Okay, thank you. Thank you. I know there are a couple of questions from Dr. Nieva and Dr. Krause, and I apologize, Dr. Nieva and Dr. Krause. Perhaps if we have a little bit of time between the OPH and our discussion of the topic in question, we can actually have the two of you make your comments or questions before anybody else. But in the interest of time, I think it's time for us to take a break. So we will now take a 10-minute break. Panel members, please remember that there should not be chatting or discussion of the meeting topic with anyone during the break. We will resume at 4:45 p.m. Eastern Standard Time. Thank you all for a robust discussion. We will now begin the open public hearing session. Both the FDA and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationship that you may have with the sponsor, its product, and if known, its direct competitors. For example, this financial information may include the sponsor's payment of your travel, lodging, or other expenses in connection with your participation in the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. The FDA and this committee place great importance in the open public hearing process. The insights and comments provided can help the agency and this committee in their consideration of the issues before them. That said, in many instances and for many topics, there will be a variety of opinions. One of our goals for today is for this open public hearing to be conducted in a fair and open way where every participant is listened to carefully and treated with dignity, courtesy, and respect. Therefore, please speak only when recognized by the chairperson. Thank you for your cooperation. Will speaker number 1 begin by stating your name and any organization you are representing for the record?
Yes, thank you. I'm Dr. Diana Zuckerman, President of the National Center for Health Research. Next slide, please. We scrutinize the safety and effectiveness of medical products, and we don't accept funding from companies that make those products. Our largest program is focused on cancer treatments and prevention. My expertise is based on postdoctoral training in epidemiology and public health and previous positions at HHS and as a faculty member and researcher at Harvard and Yale. Next slide, please. All of us want more treatment options for refractory cancers, but we also want patients to be able to have confidence that FDA approval means that a product is proven safe and effective. The OCEAN study of 495 patients has important information that was not available when the drug received accelerated approval. And even if some patients taking the drug do well, it's only with a randomized controlled trial that we can determine if PEPAXTO is helpful or if the patients would do better without it. Our center's analyses support the FDA findings that the data do not confirm the indication. In the randomized trial, comparing PEPAXTO to another treatment option, the median survival was 5.3 months shorter, and the death rate was slightly higher. Next slide, please. The sponsor says that some patients do better, but we agree with FDA that, quote, results from subgroup analyses cannot be used to conclude benefit in a subset of patients when the overall patient population has shown a detrimental treatment effect. Next slide. We also agree with the FDA that progression-free survival is not improved and that an, quote, anti-cancer therapy that prolongs PFS is not considered safe and effective if the therapy results in a detrimental effect on overall survival, unquote. Next slide, please. Public trust in the FDA has been weakened in recent years, and FDA standards matter to all of us. Would you want your loved one to take this drug rather than a superior treatment option? Unfortunately, not all oncologists will be as knowledgeable about the data as those serving on this panel, and they won't be able to make the best decisions for themselves or their patients. It concerns us that the sponsor continues to ignore FDA concerns, rely on shortcuts instead of better research, and that the company withdrew the drug in October but then rescinded the withdrawal. Was this just a delaying tactic?
We agree with the FDA that the sponsor did not provide new data, and with Dr. Pazdur that FDA approval relies on solid information about appropriate dosage, and that's lacking here. Last slide, please. Maybe PEPAXTO would benefit some types of patients, and better research is needed to prove that. As FDA states, the preponderance of evidence from the pre-specified analysis and in all other subgroups suggests an increased risk of death in patients and a potential for harm. Thank you very much for the opportunity to speak today. I know that many patients feel that this drug could be helpful to them, but we have to look at the science to see if that's true. Thank you.
Thank you. Will speaker number 2 please begin by stating your name and any organization you are representing for the record?
My name is Jenny Alstrom, and I'm a multiple myeloma patient and founder of Health Tree Foundation for Multiple Myeloma, formerly known as Myeloma Crowd. I have been in myeloma advocacy for over 10 years, and last year helped over 1 million patients and caregivers through our programs. We have had programs sponsored by Oncopeptides and all other pharma companies in the myeloma space in the past, but not currently. The key question you're asking today is if there's an appropriate risk-benefit for melphalan's approval. As a myeloma patient, I weigh risks and benefits every day as I make treatment decisions and continue to relapse over and over again. For example, do I take a risk on CRS or ICANS with a CAR T? Will I have recurring infections on a bispecific antibody? Will my eyes on Blenrep be a big problem? Can I tolerate the GI issues on selinexor? Will Revlimid maintenance or a transplant give me a secondary cancer? We are adept at having risk-benefit conversations with our doctor because we have to be. So as a patient, am I willing to risk using melphalan to gain benefit? If I knew that IMiDs grew less effective over time and I was an older patient, I would risk it. If I were a younger patient with lots of options ahead of me, I may not risk it. If I had a long-lasting transplant years ago, but I didn't want to do a full salvage transplant again, I would risk it. If I needed bridging therapy to another option, I would risk it. If I'm one of 96,000 living myeloma patients having never received a transplant, I would risk it. If I were on my 5th line of therapy and relapsed after pomalidomide, I would risk it. Please give me the option to assess risk and benefit with my doctor. The worst thing would not be to have the option at all. We know that we have wildly different types of myelomas. We cannot all be treated in the same way. We need different tools in the toolbox. So how do we learn more quickly how to subset and personalize care? For selinexor and Blenrep, your early approval helped clinics across the country learn how to modify doses and better manage side effects. Not every patient will use these therapies, but this is a great success for patients to have the option, so thank you. Conversely, panobinostat was FDA approved but unused for the most part. Doctors and patients together assessed the risks and benefits and said, no thanks. We are smart like that. For melphalan, important information was learned as part of the Horizon and Ocean studies. EMA took the learning and approved around it. Don't use it for patients who didn't respond to transplant. It won't work well. Perhaps don't use it right after transplant. The impact on marrow recovery may look too similar. The over 75 patients had clear benefit on the melphalan dex arm, which is a plus for patients who typically can't have transplant. We've come a very long way in treating myeloma, but we need to go further and faster because 42% of my friends are still dying of myeloma within 5 years. Subsetted, more personalized care is where we are all headed. I want to have all options on the table when I talk to my doctor about what I'm going to do next. There is utility for this drug, and I request that you approve it and allow patients with their doctors to assess the risks and benefit of its use for their individual situation. More choices equal better outcomes for patients. Thank you.
Thank you. Will speaker number 3 please begin by stating your name and any organization you're representing for the record?
Hello, this is Dr. Jacob Laubach. I serve as the clinical director and the chief of the multiple myeloma division at the Dana-Farber Cancer Institute. I've been a longtime member of our program. I have a large clinical practice focused in the care of patients with multiple myeloma and other plasma cell disorders. I would add that I participated in the development of flufenamide as an investigational agent, primarily through participation in the phase 2 Horizon trial. I do not have any financial conflicts of interest associated with this drug or others. Following the FDA's accelerated approval of melphalan flufenamide in February 2021, and prior to the point that approval of the agent was withdrawn, my colleagues in the multiple myeloma program and I treated 12 patients with standard of care melphalan flufenamide. 8 of those 12 patients were from my practice. We have gathered, with the help of our pharmacy team, information related to these patients' clinical characteristics, treatment response, and reasons for treatment discontinuation from the electronic medical record, and I would like to share that with you now. The median age of these patients was approximately 74 years of age, ranging from 52 to 81, and patients had been diagnosed with multiple myeloma a median of 11 years prior to receiving the drug, with a range of 3 to 24 years. They had received a median of 5.5 lines of therapy, a range of 3 to 8. All patients had previously been treated with Revlimid, bortezomib, and daratumumab. Many of the patients, 75%, had been exposed to carfilzomib, 92% to pomalidomide, and 50% to cyclophosphamide, 25% to elotuzumab. 5 out of the 12 patients had undergone autologous stem cell transplantation. The median number of doses of melphalan flufenamide administered to these patients in the standard of care setting was 4. Response data was available to us in 11 of the 12 patients. The overall response rate was 55% using uniform criteria. A complete response was achieved in 3 patients, very good partial response in 1, and a partial response in 2 patients. Patients discontinued treatment due to progression in 58%, market withdrawal in 25%, adverse effects in 8%, and death in 8%. Mucositis, alopecia, and secondary bone marrow cancers were not noted. I would highlight the fact that treatment of patients who have what is considered pentarefractory disease is a very important area of unmet medical need in our field. I would highlight the fact that alkylating agents have played a very important role in the management of multiple myeloma for over 5 decades. I would also highlight the fact that in our experience in the standard of care setting at Dana-Farber, the median age of patients was greater than 70, and that they had received 5 prior lines of therapy as a median. I would also point out that less than half of these patients had undergone a prior stem cell transplantation with high-dose melphalan conditioning. In selecting patients within my practice who I consider to be good candidates to receive melphalan flufenamide, I favored those who had not previously undergone autologous stem cell transplantation with high-dose melphalan conditioning and were not in a position to do so in the future because of age, frailty, or the patient's own preferences regarding use of high-dose melphalan in their care. Thank you very much for the opportunity to share my insights gained from experience with the standard of care use of melphalan flufenamide for the treatment of relapsed myeloma.
Thank you. Will speaker number 4 please begin by stating your name and any organization you're representing for the record?
Hi, my name is Scott Johnson, and I have no connection, financial or otherwise, just treatments with any institution. I was diagnosed in April of 2012 with multiple myeloma and received a series of treatments since then, some at Hartford Hospital here in Connecticut, at Smilow Cancer Institute in New Haven, Connecticut, and at Dana-Farber in Boston. My experience with it began in April of 2012, coming downstairs here at home, and a severe pain bit me in the back. I told my wife afterwards it was like an alligator biting me in the back, and it was horrible pain, and she helped me to the couch, and eventually, with still in a lot of pain, got to the hospital, and they felt that I had some kind of a slipped disc or something, and that it was best to seek chiropractic help, and they gave me pain pills, which was a much relief, but still noticeable pain, and that the chiropractic work didn't get me anywhere. My physician gave me at that point more blood tests and discovered that I had to come into the office. I went in, and he explained I had no back problem that chiropractic work could help, but that I had a not very common cancer called multiple myeloma, but there were treatments, and he could expect to be around for 2 or 3 more years. I began those treatments, and within a year or so, I got a stem cell transplant at Smilo Cancer Institute, and then different treatments for a certain number of years. And finally, we ran out of FDA-approved treatments, and my doctor got me in with at Dana-Farber under the care of Dr. Richardson. The treatment I'm on now is one that Dr. Richardson has used for me, and the one prior to that was melphalan a little over a year ago, and I was on that for a little over a year. And for me, melphalan worked pretty well. I didn't have the nausea that some of my other treatments had caused, and I also didn't have as much tiredness. I did have bone pain, which even Tylenol would temper down a bit, and that only lasted for about a day after each infusion. And over the course of using it for around a year or so, I see it as a pretty good treatment. It wasn't like all of them a cure. They told me there's no cure for it, but it was sort of a life preserver that got me downstream a little further and made it so that, like melphalan, which wasn't even available when I started, another treatment was available, and that's the one I'm on right now. So for me, it was a positive treatment, and it worked. It got me through some time and provided me with into the next treatment. So I see it as having worked pretty well. Thank you very much for this time to relate my story. Thank you. Bye-bye.
Thank you. Will speaker number 5 begin by stating your name and any organization you're representing for the record?
The name is Ronald Ehlers, and I have no financial relationship to anyone. I reference this discussion. I was an 81-year-old, initially diagnosed in 2013 with myeloma. I'm very supportive of this treatment, and I want to just make a quick discussion, but I found out about this speaking engagement by accident. So here we go. My understanding, the best way to evaluate treatments and its effect on me is with a bone barrel. So I'll just give you my last bone barrel evaluation, which was done in May. Sorry, I'm cutting out here on this thing. I had a 1% plasma cell by morphology and less than 0.1% myeloma cells on the flow symmetry. Yeah, I've been on a treatment for just over a year, and it's done an awful lot for my health. As an example, I was using a four-wheeler to get around because of back problems, and was unable to stand very long without an aid prior to being on the treatment. After Dr. Blau put me on this treatment, I only use a cane if I'm going to try walking more than, let's say, 20 yards. The only real problem I have is my platelets tend to be low, so sometimes it's difficult to get them off the treatment. I have no problems with infections, nor any significant problems with fatigue. My treatment now includes immunoglobulin, which I understand will help with the platelets, and Evershield, which helps with the white blood cells. I've only recently started their use. We'll see how it works with those. But however, due to my improved health and immune system being on this treatment, I support it wholeheartedly, and it needs to be still an option for an 81-year-old as I'm running out of possibilities of different types of treatments that I can be exposed to. In closing, I just want to add that Thank you a lot for letting me speak. I'm having a little problem here with getting this out, but I hope you can continue to make this treatment available to especially older patients who have been through a multitude of different treatments. I need that option, and I don't feel that it should be taken away from me. So with that, I conclude. I'm sorry for the delay. Thank you. Damn.
Thank you. Will speaker number 6 please begin by stating your name and any organization you're representing for the record?
Hi, this is Dr. Taral Patel, and working in Columbus, Ohio in private practice for almost 25 years. I have no relationship with sponsor Oncopeptide. I had a patient, I submitted the case, who was 64-year-old male, has all the possible treatment in the FDA-approved, including stem cell transplant. Even stem cell transplant, unable to put this patient on a complete remission. All of the treatment, including CD38 antibodies, lenalidomide, Pomalyst, all the treatment was given, and unfortunately his condition was continued to deteriorate. He was about to go hospice, and we discussed the 2 treatment options, including CAR-T. Unfortunately, CAR-T was in a short supply because of that. So we decided to start him on melphalan flufenamide on a commercially available drug last year, around May 2021. And patient had tolerated fairly well, except mild thrombocytopenias, not mild neutropenia, and to avoid the complication as looking at the package insert, I did start him on a secondary prophylactic with the white cell growth factor. He is a plumber. He worked full-time. His quality of life is improved. We did the bone marrow on a May, I think sometime in March 2022. And that the bone marrow shows complete response. So this is a patient who unable to go in a complete response with even stem cell transplant with the high dose chemo and radiation. And I just saw him again yesterday. Unfortunately, he's still in a complete response, and he's tolerating treatment fairly well. I understand I saw some comment about the FDA and other about the toxicity. As a physician, it's our job to talk to the patient risk versus benefit. And patient most of the time make informed decision. So I really appreciate FDA panel and relook at this, not for everybody, but subset of the population, so this can benefit patient like me. And I appreciate everybody's time.
Thank you. Thank you. Will speaker number 7 please begin by stating your name and any organization you're representing for the record? Speaker number 7. Maybe they're having some difficulties, so let's just move on to the next speaker. So speaker number 8, please begin by stating your name and any organization you're representing for the record.
Yes, I am Dr. Sibel Blau. I'm a medical oncologist and also President and CEO of the national organization Quality Cancer Care Alliance Network and Exon Research. I have been working as an oncologist for more than 20 years, and I did training and worked at the Fred Hutchinson Cancer Research Center as a transplanter before going into independent oncology private practice. In 2015, my own husband, Dr. Tony Blau, was diagnosed with multiple myeloma, underwent double transplants, and has been doing great, but we were fortunate enough to be in that situation, always waiting a possible day that this disease might return, looking at all the options that are available. So I have not only been treating multiple myeloma very intensively, but also I have been a caregiver and a worried wife of a multiple myeloma patient. My husband was a professor at the University of Washington. Could you please push to the next slide? And he retired in 2019 to develop All4Cure, where it is a data sharing platform where patients can register, and after they agree to the terms of the privacy policy, the records are curated completely from all institutions and places the patients are treated. So the information is put on a dashboard where the physicians, patients, and scientists can collaborate in the decision making. We are fortunate to have many drugs available in multiple myeloma, but partly because of that, it has become very complex in how to treat these patients. Next slide. My patient is a 74-year-old man at the time of his diagnosis, and with multiple lines of therapy, the best response being a very good partial response at the time of diagnosis, with a very short duration, and more drug was added at that time. But after relapsing and going through several lines of therapy, melphalan was started in July 2021. Next slide, please. And the data in this slide shows that the patient, if you look at the lines, the blue shows the shots that he's having, and the yellow is the treatment. The patient has been in complete response ever since he was placed on this drug. He responded so well to the treatment for the first time in many years since his diagnosis that when FDA disapproval came, we had to apply for a single IND, and the patient has been on this drug on a compassionate basis ongoing. This is an older patient who has not had a transplant and had a great response to treatment. My hope is to be able to select my patients and continue treatment on the ones that can respond to treatment as well, and of course monitor the toxicities, knowing the overall survival data, and hope that we can use the real world data to treat these patients effectively. Thank you very much for this opportunity.
Thank you. The open public hearing portion of this meeting has now concluded, and we will no longer take comments from the audience. The committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments. We will proceed with the questions to the committee and panel discussions. I would like to remind the public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. I will now read our discussion question. The task is for us as a group to review and discuss the benefit-risk profile of melphalan flufenamide for the currently indicated patient population, considering the results of the confirmatory OCEAN trial. Are there any issues or questions about the wording of the question? If there are no questions or comments concerning the wording of the question, we will now open the question to discussion. Dr. Krause, I apologize again that we couldn't get you earlier, but now is your time. Please go ahead.
Thank you, Dr. Garcia. Yeah, I wanted to bring a point up that's relevant to the benefit risk, the discussion of survival decrement, as well as the interpretation of benefit. And it's around trial design and differences versus some trials that are designed differently here. Often in oncology, we have, let's say, a new therapy on top of existing best therapy versus a placebo plus existing best therapy. And there you're truly looking at the benefit or decrement of a new therapy. In this case, we have a comparison of this melphalan analog agent with dex versus pom-dex. And pom itself, if I'm recalling correctly from my memory in myeloma, has a very substantial progression and survival benefit over dexamethasone. So the interpretation of the data, to me, should encompass that thinking. Meaning, when we talk about survival decrement, it may not actually be a decrement if there is one. It may be less benefit than POM or similar benefit than POM. And you can look at PFS similarly. If indeed there was a benefit of PFS 2 months, it may be that's a benefit over quite an active therapy combination of this newer agent. And so I think that's important to keep in mind as we think about interpreting decrement data and benefit data, particularly in what I think people have aptly described benefit to patients. Many different agents and drugs approved that have been helping the survival of myeloma patients as the number of agents have multiplied over the years. I think there's good information on that. So I would just encourage, I won't ask the question of sponsor FDA, but I would encourage ODAC to consider that, because I think that's an important element. I was going to ask it and ask for both sponsor and FDA to comment. But to me, that is an important analysis element, given what the trial design is, that wasn't really brought out yet. And I just think we need to consider that, you need to consider that as ODAC when you're thinking about these various elements. Thank you.
Hi, this is Nicole Gormley. I'd like to just respond to that comment. And then I'll ask Dr. Kanapuru to provide some other additional, information. But, you're absolutely right. This is a trial against an active comparator. And that trial design is used in oncology and has been. And the larger point, though, is that, again, we're looking at a trial that, there's potential, detriment in overall survival. It's not statistically significant. This is an active comparator trial. But the onus is on the sponsor to prove with the clinical trial data that they have that the product is safe and effective. And so, that's the issue that we're stuck with, is that with this trial design, we don't see an improvement in overall survival. We don't see a robust progression-free survival result. And so all of these things are concerning. I'll turn it over maybe to Dr. Kanapuru to mention just about, some of the other the trial design aspects that you mentioned. Dr. Kanapuru?
Dr. Kanapuru, I think you're still in mute.
So perhaps she's having some technical...
Yeah, I'm here. Can you hear me?
Yes.
Yes, please go ahead.
Okay. Thank you. Thank you for that question. As Dr. Gormley just pointed out, this trial was designed to, demonstrate superiority and also to show that there was no detriment in overall survival. But we do have, that the trial designs of active comparators have been used in oncology and in multiple myeloma. If I could just bring up FDA slide number 72 onto the screen. So I would like to point out that we have had, prior approvals where we had active comparators. And on the slide that will just show up in a short while, you can see that, there were two trials, one with the confirmatory dex versus the bortezomib dex. Again, here these were actually two similar, mechanism of action, proteasome inhibitors. And in this trial, there was a significant PFS improvement as well as an overall survival benefit. It showed superiority for PFS as well as benefit in overall survival. And similarly, we had another trial called the first trial, but there was actually a doublet comparator compared to a standard of care triplet. And even in that trial, there was improvement in progression-free survival and overall survival. So these trial designs have been used, and the drugs have shown that they could demonstrate superiority as well as OS benefit. What we have here is a trial that was designed to show superiority for PFS. And at least, if not an improvement, it was also designed to show an improvement in overall survival, but there's not a demonstration of superiority for PFS, and what we have here is a concerning potential detriment in overall survival. Thank you.
Thank you. I just probably have a comment on that, just want to open the group for foot-provoking thoughts, perhaps. I mean, when I look at the data, I just want to make sure that everybody, I think that all of us would agree that I personally, just my own personal thoughts and comments, I come to these meetings with an open mind, and I come after reviewing the docket, the documents. I'm not a multiple myeloma expert. I'm a drug developer in GU oncology. But when I see the data, after I review the docket based upon the FDA presentation, based upon the applicant presentation, it's hard for me to understand, how we use subset analysis to try to tease out true benefits from a treatment. And I just don't, that goes against everything that I have actually been taught from the statistics, for clinical trial design and for drug development. And I think eloquently, many of you have actually reviewed why post hoc analyses are hypothesis generating and should not be actually used for us to make decisions as to who gets therapy, right? Forest plots are very, very imperfect. But even if you were to look at that, I'm also perplexed because if you look at every single possibility for survival, there is a detrimental potential because every single hazard ratio crosses the confidence interval of 1. So that is what I'm trying to wrestle here with, is yes, maybe there's a treatment that has some activity. Yes, there is no doubt that the natural history of multiple myeloma may allow some patients to have access to these agents, but every single point data that I see here will really leads me to believe that without confidence, even if the PFS is real, I cannot tell a patient I'm going to actually be able to put you in a therapy that may delay your progression-free survival, but it may have the chance of actually harming you and cause detrimental outcome by shortening your survival. And that's what I think many of us perhaps are struggling with. So with that, maybe I'll ask Dr. Nieva to express his thoughts.
Thank you, Dr. Garcia. And I think really the central question here is this PFS question, because if we say that in consultation with the FDA and with the FDA's understanding of what the planned statistical analysis was going to be, this was a positive trial for PFS, well then the company's done everything they've asked them to do and shown that, yeah, if we showed in the first trial that we had a response rate, and the second trial we showed a PFS benefit. So the issues to me are really, was the statistical analysis plan something that was brought to the FDA as a fait accompli, and the FDA would view it as being wrong, or was this developed in consultation with the FDA? Now, so that's a question that either the FDA or the sponsor can answer. But I think the other thing that this data is clearly telling us is that this drug really shouldn't be used in people who are melphalan resistant. And the data for looking at subgroups, I think, can tell us safety signals that certain people shouldn't get a drug, right? We know that, the FDA has sort of said, and I think that the community accepts, that EGFR and ALK mutated lung cancer patients, for example, shouldn't get checkpoint inhibitors. that even though they may have been included in some studies, they clearly don't show benefit. And I look at this in a very analogous way, that we've identified a clinical subgroup that shouldn't get the drug, but aside from that clinical subgroup, everything actually looks very good. So I'll see if anybody wants to talk about the statistics and whether there was actually collaboration in the statistical design, or whether or not the statistical design that was presented was not viewed by the FDA beforehand, and now they say, well, no, clearly it was wrong.
Yeah.
Thank you.
This sponsor, I have the data that was asked just before the break as well, if I may show that, and then come back to the question that was just asked about the statistical analysis plan. So I'll put up the slide on the duration of thrombocytopenias and neutropenias. I'm waiting for it to now appear on the screen. Here we have it. So the median time to resolution of grade 3 or 4 neutropenias was 8 days in both arms. For the thrombocytopenias, it was 15 versus 9 days. I think very important to mention for those who are concerned with MDS in the long term is that was only one patient in both treatment arms that developed MDS. To the question about the statistical analysis plan, this study was developed under a SPA, and with that comes the development of a statistical analysis plan together with the agency. So the SEP was initially submitted already in 2017, and there were modification agreements along the line until 2019 where no changes to the SAP were submitted. The final submission of the SEP was on February 2021, and there has been no further commonly agreed changes. So the censoring rules were based mutually under a SPA with the FDA. That's the sponsor's view.
Thank you for that. Let's just go ahead and ask Dr. Kwok for a comment. Or
Can the FDA respond, please? Actually, we need to stick over the question to both groups. Is it okay if we respond?
Go ahead, Dr. Gormley. Yeah.
Yeah. Nicole Gormley. So, there's several aspects here, and I'm going to allow, some of my statistical colleagues to comment as well. But, so first, the SPA process is one that we engage in with sponsors in order to try to, reach alignment on, the major aspects of the protocol. It is not by any means a uniform endorsement of every aspect of the protocol or the statistical analysis plan. So, I can have my statistical colleagues comment a little bit more about, some of the specific issues. But the censoring rules that we shared are the typical censoring rules that we use in multiple myeloma. But, even though we can discuss the SAP and the other analyses plan, that did not include for the, reassessment that occurred. That obviously was outside of the SAP. And then also, I want to just comment as well that when we make any assessment of, for a product, there's a requirement that there's demonstration of safety and effectiveness. So we don't look in an isolated fashion at just one endpoint. We look at the entire clinical picture of the data that's presented. And so as stated previously, we would never rely on a positive PFS value if there was evidence of detriment of overall survival. And when we've used subgroups in the past, the ITT result has been positive. We have not and do not use subgroup analyses to find a population that has a favorable benefit when the overall is negative. So I'll turn this over to our statistical colleagues. Dr. Rodriguez, do you want to comment?
Hello. Yes, so this is Lisa Rodriguez, Deputy Director for Division of Biometrics 9. So I agree with what Dr. Gormley just said. There are various PFS censoring rules that we looked into FDA's analysis censored unconfirmed PD, for example. I think it was what was stated earlier, though, however, regardless of how PFS was analyzed, the difference in the median PFS between the arms still remained approximately 2 months or less. And still the overall finding in the ITT population for OS is a concern. So we don't think limiting an indication based on a subgroup is appropriate. And
Thank
we also,
you.
Sorry, I wanted to add, we also did previously communicate censoring rules with the sponsor. Thank you.
Thank you, Dr. Rodriguez. Let's just get back to our group and try to actually address some of the comments that the committee members had. Dr. Nwap?
Hi, my name is Mary Kwok. I'm from the University of Washington Fred Hutchinson Cancer Center. I have a clarifying question first. So when we're just talking about the indicated patient population, now we're talking about patients that are greater than 36 months from transplant. Is that correct?
This is Dr. Nicole Gormley. No. That's not the indicated patient population. That's the new indication or new population that the sponsor is proposing. The currently indicated patient population is those that have received at least 4 prior lines and received refractory to at least 1 proteasome inhibitor, 1 IMiD, and 1 CD38. It does not.
So like the study was designed. Yeah. Thank you. That's helpful. I, treat patients with myeloma, and as I'm, looking at these slides and hearing the presentations, I completely appreciate the viewpoints that were shared by the clinical members and then, the public, because I agree that, there's a huge need for a medicine like melphalan. But at the same time, I think, how am I going to face the patient in front of me and describe how do I explain this trial to a patient, and I think that's where, Dr. Garcia, you mentioned that that's where the struggle lies, right? Even if the patient may potentially benefit from it, a conclusion from the study is that there's an increased risk, a detriment to overall survival. And so I think that's, that is something that's very, very difficult to do as a treating physician. I also think that the takeaway for me, at least from the study, is really probably that this would not be the desired treatment, at least in the first 2 to 4 lines of therapy. And then I also think of just Pom-Dex as a comparator. A lot of times when we treat patients, especially with early relapses, we're treating with triplets. And so it's, I don't know that it would be easy to make direct comparisons in actual clinical practice, especially within the first 2 to 4 lines of therapy. I guess I would just, and this is just an open-ended question, but I just want to know how to think about melphalan, especially when it's given as a single agent, and perhaps what was suggested, it finds its role in patients that are elderly, no transplant, relapsed and refractory, but I don't think that's the question that's being asked here. Thank you.
And
thank you.
Can I respond as a sponsor? Just one thing to clarify the sponsor's position is that we propose a limitation of use. So we very much agree that the findings from OCEAN should be applied to the current indicated population. So to the current label, we want to take the learnings from OCEAN into account. And I cannot emphasize this enough.
Yeah, if I may just add to that, I just wanted the efficacy in U.S. patients for Horizon, please, Ted, to be put up. It's a very important slide because Dr. Kwok raises an extremely good point. We see, obviously, the challenges of the heterogeneity in OCEAN, recognizing that we're dealing with a mechanism of action challenge between the 2 drug classes, and indeed one's a doublet, both are doublets rather. But I think as we think about the current label, to me anyway, the current label points to the Horizon population. I think it's very important to share with the audience this particular slide of efficacy in U.S. patients, because Horizon was a study in which we enrolled, the majority were actually U.S. patients, and we saw clinical benefit as reflected by these numbers reflecting response in PFS. And obviously, when you break them up according to the pre- and post-transplant populations, you can see the benefit here. So I hope this data is helpful in making some sense of in what setting one would consider PEPAXTO for patients.
Yeah, and I appreciate that. Thank you for bringing that. I think it's important for us to really actually get to our discussion point within the committee rather than trying to clarify questions that really, to some extent, have been addressed and depicted in the prior presentations. But thank you for that. Dr. Nowakowski?
Yes, thank you, Greg Nowakowski. I just wanted to comment on the comment by Dr. Nieva, who, for me, the PFS issue is not necessarily really a central issue here, because I think, like with any other trials, and others express this sentiment as well, we have to look at the totality of evidence. And here, the possible detriment and safety signal with the detriment of overall survival is very concerning. Regardless if this PFS difference, which is pretty modest, is real or not, and how do we interpret those? The other comment, just a short comment about the subset analysis, and I appreciate all the effort which sponsor really put into understanding different subsets and dissecting it in different ways, and even comparing with the image cohorts. And I also appreciate all the effort FDA took also to address those concerns and conduct a number of sensitivity analysis and subset analysis in those. And in a big picture, when one takes the bird's eye view on this, I was just thinking to myself what I would tell my residents or medical students about this study. And essentially what I would say, they're all hypothesis generating. So they're very interesting hypothesis, scientific hypothesis, which group may or may not benefit from the intervention. But it's not a definite proof. And I think that's what we are struggling here with.
Thank you, Dr. Nowakowski. Dr. Sakaris?
You, Dr. Garcia. Mikhail Sakaris from Sylvester Cancer Center, University of Miami. With accelerated approval, we have the opportunity to get drugs that work better than other drugs out there to desperate patient populations who have few other options. And that's predicated on a trial actually confirming the initial magnitude of benefit and safety, and ideally extending that to something that's more clinically meaningful, like overall survival. What we see here is a progression-free survival that, depending on how you interpret progression, toys around significance, but in the end is a median improvement of only 2 months. And we have a worsening of overall survival by 4 months. And we look at the totality of these data. We look at progression-free survival, overall survival. If it's available to us, we look at patient-reported outcomes. It's not available here. And the totality of data, for me, doesn't add up. And I keep circling back to what Dr. Kwok mentioned earlier, and that is, how the heck would I give informed consent to a patient to receive this drug and explain that progression-free survival, which itself is a difficult concept to explain to anyone, may be better by a couple of months, but that person will live 4 months shorter than if he or she didn't get this drug. And that's where I get stuck with this application.
Thank you, Dr. Sakaris. Dr. Madden?
Hi, Robbie Madden, NCI. So, we spent a lot of time talking about how we should censor and how the FDA did it versus the sponsor. But when you take a step back, you realize that the PFS benefit changes just by shifting 4 or 5 patients or less than that. And I think you have to wonder, is that really robust data? And then you default to the survival data, and whether or not the outcomes are worse, it's not better based on the data presented. And so I think that really factors into this part of the discussion significantly for me. Thank you.
Thank you. Dr. Harrington?
Thank you. My question has been answered by the previous 2 questioners. Thank you.
Thank you. Dr. Chan?
Hello. I was wondering if there's a precedent, I couldn't think of one, where someone is changing the label based on subgroup analysis and also changing the dosing without prospective studies to support that. Question for the FDA.
This is Nicole Gormley. No, this is an unprecedented scenario. Thank you.
Thank you. Thank you all. I think that if I can summarize some of the points that the committee has discussed, it does appear that for some PFS issues and the discrepancies between the FDA PFS and the sponsor PFS do not appear to be of a significant concern based upon how you censor, based upon how you measure PFS as described by Dr. Sakaris. It also is clear that all of us, for the most part, feel that the subset analysis and the post hoc analysis are really hypothesis generating and should not be interpreted as a dogma to deciding who gets therapy and who does not, including perhaps what the applicant is trying to address right now, which is to try to actually encase their agent into a very specific patient population based upon that subset analysis, for which it doesn't appear that the statistical design was actually built within the study. And clearly, the biggest issue is survival. The challenges of seeing every single hazard ratio, however you dissect the data, that crosses 1 and clearly appears to be with the potential detriment of around 4 months compared with POMDEX. So I think that that's sort of the theme of our discussion right now. And with that, I think the committee will now turn that attention to address the task at hand, the careful consideration of the data before the committee as well as the public comments. We'll proceed with, if there are no further questions, and I'm sorry, I was misreading my section here, so I apologize, but if there are no further questions, maybe we can actually move on to the next question, Dr. Chen. So this is voting question, and the question is, given the potential detriment in overall survival, failure to demonstrate a progression-free survival benefit, and lack of an appropriate dose, is the benefit-risk profile of melphalan flufenamide favorable in the currently indicated patient population?
Thank you, Dr. Garcia, and I will provide instructions for the voting. This is Shijia Chen, the DFO. Question 2 is a voting question. Voting members will use the Adobe Connect platform to submit their votes for this meeting. After the chairperson has read the voting question into the record, and all questions and discussion regarding the wording of the vote question are complete, the chairperson will announce that voting will begin. If you are a voting member, you will be moved to a breakout room. A new display will appear where you can submit your vote. There will be no discussion in the breakout room. You should select the radio button that is the round circle button in the window. That corresponds to your vote, yes, no, or abstain. You should now leave the no vote choice selected. Please note that you do not need to submit or send your vote. Again, you need only to select the radio button that corresponds to your vote. You will have the opportunity to change your vote until the vote is announced as closed. Once all voting members have selected their vote, I will announce that the vote is closed. Next, the vote results will be displayed on the screen. I will read the vote results from the screen into the record. Next, the chairperson will go down the roster and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. Are there any questions about the voting process before we begin?
Yes, to the chair, Dr. Garcia, may I ask a question about when would be the appropriate time to discuss the wording of the question? I'm sorry.
Maybe right now, since we're moving already to the voting.
Thank you. This is Stephanie Crawford. I do want to state in fairness to the sponsor, there was a comment made by the sponsor earlier that the voting question can be considered leading, and it can be the first 2.5 lines. Perhaps it would be fairer to just start the voting question, because some would say the first almost 2.5 lines would be a false premise to some. Maybe it would be fairer for the voting question to just start with the verb is. Is the benefit risk profile of melphalan flufenamide favorable for the currently indicated patient population without the part that was, quote, given? Thank you.
Thank you. Does anybody else have any question or comments concerning the wording of this question? If not, I'm sorry, please speak. Who's speaking?
So sorry, I had my hand up. I'm just agreeing with the comment the individual just said. There was obviously some disagreement about some of the data representation. So I'm agreeing with the prior comment, Albert Kraus, industry representative, in how to adjust the question a little bit.
Maybe we can ask the FDA for guidance as to how to retweak this if the committee...
That would be fine, okay. Is the benefit...
question, sorry about that. I'm so sorry. This is Shija. So the voting question is going to be stay as is. So the panelists will go ahead and vote when it starts, and then you can make comments once you finish the vote. Thank you.
All right, so I'll repeat and reread the question. Given the potential detriment in overall survival, failure to demonstrate a progression-free survival benefit, and lack of an appropriate dose, is the benefit-risk profile of melphalan flufenamide favorable for the currently indicated patient population?
Let's go.
So maybe Dr. Chen, you can move us to, if there are no questions or additional comments, we will now begin the voting on question number 2.
Thank you, Dr. Garcia. We will now move voting members to the voting breakout room to vote only. There will be no discussion in the voting breakout room. Voting has closed and is now complete. Once the vote results displayed, I will read the vote results into the record. Voting has closed and is now complete. The vote results are displayed. I will read the vote totals into the record. A total of 2 yeses, 14 noes, and 0 abstention. The chairperson will go down the list and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did, if you want to. Thank you.
Thank you, Dr. Chen. We will now go down the list and have everyone who voted state their name and vote into the record. You may also provide justification for your vote if you wish to. We'll start with Dr. Chen.
I voted no. I think there's marginal PFS benefit, and then in the setting of significant concerns about overall survival.
Thank you. Dr. Sung?
Anthony Sung, Duke University. I voted no. I do think it is possible that there is some benefit in specific subpopulations, but on the whole, I do not think that the benefit outweighs the risk.
Thank you. Dr. Fredley?
Boris Friedland, I voted no. I believe that given the potential for a detriment, the data provided does not allow reliable identification of the population in whom the benefit-risk profile is favorable.
Thank you. Dr. Liu?
This is Chris Liu. I voted no. We've discussed this. The post hoc analysis really should be used for hypothesis generation as opposed to labeling and indication for use. There's certainly a need for better drugs. We all feel that, but we shouldn't be using drugs that might actually be harming patients. And to me, the answer here is pretty simple. You have an analysis which may support the use in a specified patient population that could show a benefit, and a confirmatory study should be performed in this population, but the data do not support the use of this agent at this time.
Thank you. Dr. Harrington?
David Harrington, Dana-Farber Cancer Institute. I voted no. In the indicated population, the results of the trial are fragile, and because of the marginal PFS benefit and the possibility of a safety signal for survival. I think the proposal by the sponsor on how to trim the population will only lead to confusion if it's not confirmed in a subsequent trial.
Thank you. Mr. Mitchell?
Yes, thank you. I voted no, and I'd like to take a minute to explain why. This vote was very difficult and personal. I'm a candidate for this drug. I'm a multiple myeloma patient diagnosed 12 years ago and now with relapsed refractory myeloma, who's presently receiving a proteasome inhibitor immunomodulatory agent, a CD38-directed monoclonal antibody, and dex. I've not had an autologous stem cell transplant, so even based on the sponsor's post hoc analysis and suggestion for how to use the drug, I'm a prime candidate. My care is directed by oncologists at Dana-Farber, so the presence of doctors from Dana-Farber participating in this meeting weighs on me as well. Frankly, this could be a life or death decision for me and others like me, but after listening closely to both the sponsor and FDA presentations, I conclude that melphalan has demonstrated a lack of confirmed benefit, inferior overall survival, and a potential for actual harm. Post hoc analysis shouldn't be used to confirm a drug. If the history of ASCT and age are considered as hypotheses to explain and demonstrate effectiveness of this drug, then the sponsor should run a prospective randomized clinical trial and test those hypotheses. Then I and other patients will be able to take the drug with confidence based on substantial evidence that it's safe and effective. And thanks for letting me have that time to explain.
Thank you, Mr. Mitchell. Dr. Nowakowski?
Thank you, Greg Nowakowski, Mayo Clinic. I voted no due to reasons which already other mentioned that we discussed. So there was a questionable benefit in terms of the progression-free survival and potential detrimental impact on overall survival, raising safety concerns with this drug. I will also point out that in the representation of U.S. population in the trial was relatively limited, and it was not representative of U.S. population diversity as well, which is additional factor which limits applicability of this study to U.S. population.
Thank you. Dr. Please.
I appreciate what everyone has said, although I think there is a need for off-the-shelf treatment options for relapsed refractory multiple myeloma and the treatment of the extramedullary disease. Therefore, I voted yes.
Thank you. Jorge Garcia, I voted no. I remain concerned about the lack of PFS benefit based upon how I review the statistics of the study. And certainly the biggest issue, again, are the concerns for detrimental outcome regarding overall survival in the entire patient population. Dr. Nieva?
George Nieva, I voted yes. The OCEAN trial is a positive trial based on the PFS using a pre-specified analytic plan. And I think it's problematic to have the sponsor submit an SAP to the FDA, have the FDA see it, and have the FDA analyze the data using an alternative methodology. I think everything flows from the ITT analysis. Since when it's positive, you should look at subgroups, and when it's negative, you should not. It does appear that the OCEAN study confirms clinical response and benefits seen in Horizon. But to say it does not looks to me like an exercise in moving the goalposts. I do not think the numerical difference in OS is a safety signal, but it's a lack of efficacy signal. While it may not have been obvious at the time of study design that you shouldn't use melphalan in a melphalan-resistant patient population, it certainly seems obvious now. And the analysis provided show that patients may benefit as long as they're not melphalan resistant. The sponsor, however, is making highly problematic recommendations and setting that time frame at 36 months post-transplant. And the clear concern on the part of the FDA that this is retrospective is understandable. This minimum duration for avoiding flufel is post hoc, but it didn't have to be. The cutoff dates could have been transplant yes or no, or one of the pre-specified numbers, and it's a shame that we were not provided those. Thank you.
Thank you. Dr. Watt?
I also voted no, based on the inferior overall survival with melphalan. I also hope that melphalan has a place in future treatment of myeloma, but probably not in this patient population defined in OCEAN.
Thank you. Dr. Sakaris?
This is Mikhail Sakaris, and I voted no. Accelerated approval is designed to get drugs to patients who desperately need them quicker than they would get to those patients through the regular approval process. But a critical aspect of accelerated approval is that follow-up trials actually confirm the initial benefit that's seen. Unfortunately, in this case, the follow-up trial flopped. And not only did it not show the magnitude of benefit that we saw initially, but it potentially showed an increased risk of death in patients with significant toxicity. And for that reason, I voted no.
Thank you. Dr. Coons?
Hi, yes, this is Dr. Pamela Coons, Yale Cancer Center. I also voted no for many of the reasons stated, but mostly given the results of the confirmatory trial that showed the marginal PFS benefit and potential detriment with overall survival. I'm also not supportive of using a subgroup analysis for the indication, but like others have stated, I'm also open to seeing a prospective trial done to look at the subgroup. Thank you.
Thank you. Dr. Madan?
If I may, I just want to say that I appreciate Mr. Mitchell's ability to be objective in these circumstances and eloquently state his rationale. This is Robbie Madden of the NCI. I voted no. Essentially, I don't think the trial supported robust activity here as it was designed to do. I do appreciate the sponsor's willingness, as they stated, to conduct a prospective study based on the hypothesis generating data from this trial. Thank you.
Thank you. Dr. Wallman?
This is Scott Waldman, Thomas Jefferson University. I voted no for the same reasons that everybody else did. I think the confirmatory trial was not confirmatory. I don't think it was, it demonstrated clinical activity, or at least remarkable clinical activity. And I think that there is a risk, potential risk for overall survival in the patient population. So for those reasons, I voted no.
Thank you. Dr. Crawford?
This is Stephanie Crawford. I voted no for the reasons stated by others in consideration of the high bar of evidence needed for approval by FDA regarding safety and efficacy. I can only consider and evaluate the study results currently available and vetted, which is the reason for my vote. That stated, however, I think all of us would agree that multiple effective treatment modalities are greatly needed for relapsed or refractory multiple myeloma. Melphalan flufenamide may hold promise for some patients. I also note that the sponsor expressed willingness to corroborate findings in a new prospective trial in the recommended population. In my opinion, that would be of benefit, and I would hope that the results would be considered by the FDA at the appropriate time. Thank you.
Thank you, Dr. Crawford. Yeah, if I can summarize the voting, obviously the vast majority of the committee voted no. For our two colleagues who voted yes, it appears that was just the consideration that the study was met the primary endpoint, and perhaps the lack of the issues with survival related not to detrimental, but rather to lack of efficacy of the agent. For the vast majority of us who voted no, clearly related to the marginal PFS benefit, regardless of however one addressed that PFS, and certainly the concerns about detrimental outcome, survival specifically, with the hazard ratio in favor of, against, I should say, the combination of melphalan and steroids. Certainly, I think that we all embrace the ability clinically to do post hoc analysis, as they may allow us to tease out patient populations who may derive benefit in future studies. And certainly, I think you have heard, as the applicant have heard loud and clear that we're all embrace new treatments for multiple myeloma, and we're encouraging you to work with the FDA to see if you can actually identify the right patient population for the right clinical trial with the right statistical design if you really believe this agent has promise in multiple myeloma. With that, and before we adjourn, are there any last comments from the FDA?
Yes, this is Nicole Gormley. I'd just like to say thank you for your discussions and considerations today.
Well, thank you all for a robust and active discussion. We will now adjourn the meeting. Thank you again for all your participation. Have a great night.
The meeting is now over. All the participants have been disconnected.